Today in Chandigarh, it is raining heavily.
Oh, is it? Is it?
Yes,
Yeah.
lot of rain. Lot of
Yeah,
rain.
we hear a sound.
Yeah, there is rain.
In Ahmedabad also from yesterday evening it is raining, not heavily,
Right.
but constant raining.
Okay.
Okay.
Very nice to see all of you.
Oh, once again, after a long time.
After long.
I think we met in the biological water. Yeah.
We met there. Yes. It was very interesting.
Yeah, it was interesting.
Good morning, everyone. Soon we will be starting with the webinar. Let us wait for a few minutes for other participants to join.
So, hello, Retno, how are you?
We'll meet actually.
Has Dr. Deshmukh joined? Dr. Deshmukh?
I'm fine, Dr. Soranji. Sorry.
Yeah, so Retno, I am in Bali on 1st of December.
Okay.
Oh, you will come to Bali?
Yeah.
Yes. Please contact me.
Yeah, I am there for 4 days.
Yes.
Yeah.
And December.
Yeah, December 1st week.
Okay. Please contact me, please. Yes.
Yeah, surely. I need your number.
Okay.
Yeah.
Okay. I will meet you.
So, which department or college or university you are there attached?
At Department of Biology, Faculty of Math and Sciences, Udayana University.
Udayana University, okay.
Yes.
Science knows no country because knowledge belongs to humanity, and it is the torch which illuminates the world. Science is the highest personification of a nation because the nation will remain first, which carries the farthest the works of thoughts and intelligence. Wishing you all a very pleasant morning. I, Ms. Sonal Singh Shrivas, Assistant Professor at Sri Shankaracharya Institute of Professional Studies, Raipur, Chhattisgarh, and your host and mistress for today, welcomes you all to celebrate International Microorganisms Day, organized by the Microbiologist Society India, St. Xavier's College, affiliated to Ranchi University, Botany Department, St. Xavier's College, Ranchi, and IQAC. Let's explore the microscopic marvels that shape our world. It gives me an immense pleasure to be a part of today's webinar and host today's event. Divided by borders and yet united by technology, here we are all celebrating today's International Microorganisms Day to commemorate the contribution made by Anton van Leeuwenhoek, various researchers, scientists, scholars, and microbiologists. And whether we accept it or not, we know that microorganisms are really very essential part of our lives. From food to health to every sector, they are contributing much to the human needs. And I congratulate the Microbiologist Society India and St. Xavier's College Ranchi for taking the initiatives and organizing various competitions such as speech, art, Olympiad, mime, and various presentation and motivating the young minds to showcase their talents and put up their thoughts amongst all of us in the Microbial Fest. We all know that India is a land of culture and tradition where we believe in seeking the Almighty's blessings before beginning or doing any other work or ceremony. I therefore invite Ms. Sneha Kashyap, an assistant professor in the Department of Sociology, St. Xavier's College, Ranchi, Jharkhand, for the prayer service.
Good morning to all, one and all present here. Let us pray. Dear Lord, we thank you for this day as we gather here in your presence for the international webinar on International Microorganism Day. We thank you for this opportunity that you have aligned us together from different zones to hold this webinar. We ask you to bless and guide us. We ask you to bless all dignitaries, the guests present here, provide them with knowledge and wisdom to contribute towards this webinar. May the participants and the presenters be blessed with a fruitful discussion. May this webinar be a huge success and contribute towards your glory. We put everything into your mighty hands. In Jesus' name we pray. Amen.
Thank you so much, ma'am. May- By the time our beloved guest, Dr. Uma Shankar sir, is joining, may I know if Mr. Ranjan Garge has joined the webinar or not?
I am there. I am there.
Okay, sir. Okay.
Yeah. So you want me to start?
Yeah. So, please give me a moment. Actually, due to the technical issues, I just
Okay.
Dr. Ranjan Garge, as we are very excited and very happy to have you amongst us. Sir is a microbiologist, science journalist, adventurist, environmentalist, and management expert. He was head microbiology department, Government College Aurangabad, and he was a director pre-IAS Training Institute Aurangabad, retired as the principal, Government College of Arts and Science Aurangabad. He is now the director, Cosmos Outward Bound Pioneers Aurangabad from 2011 to onwards. He is a corporate social trainer, gives motivational speeches, and conducts workshops. His core training areas are outdoor management development and faculty improvement program for teachers. He is a science propagator. He has been awarded a silver medal in mountaineering at the hands of the Presidency and Best Maharashtra Award at the Science Propagator Award by Marathi Vidya Parishad, Mumbai and Indian Physics Association Pune. He is the recipient of Rising Personality of India Award instituted by Penguin Publishing House and the Bharat Gaurav Award of Indian Economic Forum of India. Dr. Ranjan stands listed in the world's WHO. He is a member of Academic Council of MGM University, Aurangabad. We truly are blessed that we are getting the opportunity to meet you, sir. And now I would like to request you to please kindly put up your thoughts and encourage the audience.
Okay.
Thank you, sir.
So thank you very much, Sonali, for saying so high about me and introducing me. I must congratulate the Saint Xavier College for organizing this beautiful seminar on recent advances in life sciences. And I'm also happy to be the opening batsman of this seminar. I don't know whether the first speaker has arrived or not, but ultimately I am there. So I will just switch over to I will share the screen. So please bear with me. So, do you see the slides? I don't know what is the problem.
No, visible.
Okay, 1 minute. I think is it allowed from the admin side?
I think they have to permit
Yes.
you. They
Huh.
have to allow you.
I think I shall request Sonali to please permit the sharing.
Okay. Yes. Yes.
Yes, sir, I'll ask the coordinator to kindly do it as soon as possible. Please give us a moment. So there are no restrictions from our end. Can you please try presenting the screen once again?
See, I'm trying.
Okay.
It comes, but it doesn't. The slides don't appear. I don't know what is the
So
problem.
your screens are, yeah, the screen is visible. Now you can move to your PPT and we'll be able to
Yeah,
see the PPT
yeah,
presentation.
that's what I'm doing. But
Okay.
You open
Yes,
PPT.
sir.
You
The
open.
slides
Yes.
are visible.
now
I,
is it visible?
yes, it is
Yeah.
visible.
You can
Make
put
the
it
full
on the
screen.
presentation mode.
Uh,
Yes, sir.
So should I
Yes.
start?
Yes, sir. Sure.
Yeah. So the topic is recent trends in life sciences. And I'm just going to highlight some 3 to 4 technologies which are now the current trends in life sciences. Let us now focus on this very interesting kind of a uh, cartoon. So it says, the GMO brinjal, it says that, Professor, we want to discuss certain flaws in your scientific theories. And hearing to this comment, one professor tells others that that's it, we have taken the genetic engineering too far. And therefore, I think in genetic engineering, the scientists themselves are now afraid that whether they are taking this engineering part too ahead or what. We talk of synthetic biology. Synthetic biology is an engineering counterpart to biology. Whether it is building a novel genome or editing an exciting one to add a new function or to remove a problematic pathway. So, the whole idea is to improve the performance. And this synthetic biology got its first push in the year 2000. So, in short, it is a genetic toggle switch that let the researcher turn the gene on and off. See, the major paradigm shift that we see in this particular area of life sciences, that we are shifting from the body parts to genetic parts. That means instead of organ transplant, we are now switching to the biobricks transplant. That means transplanting only the defective genetic part and to achieve that particular recovery or repair. So this paradigm shift basically is the one which is coming up as a new trend. The first technology that I'm going to highlight is IGEM BioBricks, that is International Genetically Engineered Machines, that is IGEM. Registry of Standard Biological Parts is a growing collection of genetic parts that can be mixed and matched to to build synthetic biology devices and systems. So this is what is all coming up as a new trend. Today, there are about 2,000 parts, biological parts or biobricks, which are available in the repository. So this registry is to be made with the standard biological collection center. Biological part is a sequence of DNA, basically, that encodes for a biological function. For example, that part can be a promoter, that can be the protein coding sequence. So, at its simplest, it's a basic part of a single function unit that cannot be divided further into a smaller functional unit. That particular part or genetic part becomes a biological part. The first attempt to create a list of standard biological parts was done in 1996. This team actually introduced a cloning strategy for the assembly of short DNA fragments. However, this early attempt was not widely recognized by the scientific research community at that time. But in 1999, Arkin and Andy, they realized that the heterogeneous elements that made up a genetic circuit were lacking standards, and therefore they proposed a list of standard biological parts. So initially, the standard biological parts were listed, and accordingly, those were put into the repository. Basically, how exactly this biobrick works, it's very interesting. See, most biobrick parts in the registry, they are maintained and propagated on plasmids. So plasmid is a backbone, and the construction of biobrick parts, devices, and usually requires working with the plasmids. Now you see in this picture, if you focus upon the red line part is a plasmid part, and this bluish part is a biobricks part, that is BBA underscore B0015 is a specific biobricks part which has been inserted into this plasmid, and this whole plasmid now is actually preserved in registry. So this is how exactly it works. Now biobrick part is basically, as I told you, is a coding sequence. It can be a promoter or ribosomal binding sites, or it's a terminator. This basically was introduced by Tom Knight at MIT in 2003. And since then, various research groups have utilized the biobrick standard parts to engineer novel biological devices and systems. Biobrick can be thought as a building block that can be put together in different ways to assemble larger synthetic biological circuits together and have a unique function to perform. Examples of biobrick parts include coding sequences, promoters, and all that, as just I told you. The second important area of life sciences is in silico research. Basically, the pharmaceuticals, cancer research, and the stem cell research is very much into this in silico research today. A 3D cell structure is used to study the biometabolism, and accordingly, the solutions are actually found out. It encompasses the theory and application of computational approaches to model and predict and explain biological functions at that particular molecular level. So it is 3D bioprinting basically. This 3D bioprinting is performed in three ways. The first is spheroids. The spheroids are the spherical cellular aggregates. That is one type of studying the 3D bioprinting. The second part is organoid. Organoid means these are the tiny self-organized three-dimensional tissue cultures that are derived from the stem cells. And such cultures can be crafted to replicate much of the complexities of the organs. So, not an entire organ, but a small part of the organ is actually brought into the bioprinting. And the third actually system is the organ on chip. That means liver on chip or the lungs on chip. So that is how entire liver three-dimensional structure is studied and its metabolism is understood. So 3D bioprinting is the technology that has come up recently. The third technology that I'm going to talk about is the CRISPR technology. I don't think this is quite new to the students of life sciences. And in fact, the students must be studying about and learning about this CRISPR technology. But this is a fantastic technology. So it is the clustered, regularly interspaced short palindromic repeats. Genome editing is a group of technologies that give scientists the ability to change the organism's DNA. And these technologies allow genetic material to be added or maybe to be removed or altered at a particular location in the genome. And that is being done with this particular Cas9 enzyme. And these are the different palindromic repeats. Now, CRIS19 or Cas9 was adopted from a naturally occurring genome editing system in E. coli in 1987. So it is actually a editing system, gene editing system that has been taken from E. coli. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments. They are known as the CRISPR arrays. So CRISPR array is exactly a DNA segment that we must understand. Now let us focus upon this figure. This figure is very important, showing you how the DNA editing by the CRISPR-Cas9 enzyme is carried out. This particular left side figure, as you see, there is a cell, and inside the cell you see a nucleus, and there are chromosomal pairs inside. Now you see a chromosome with a black circle. Exactly, there is a problem. The defect in the chromosome is at that particular point. So in this case, what happens is a cell is actually trans-infected with an enzyme complex. This enzyme complex contains three elements. The first is a guide molecule, which is in blue color. The healthy DNA copy, now which is going to be replaced, that is in green. And the third is a DNA cutting enzyme that is shown in the star. Now, what happens is in the first picture, the second picture, what you see is a specially designed, synthetic guiding molecule that is in the blue color. It approaches towards the defective part, and that defective part is shown in the that is target part is shown in the red color. Now, since the guiding molecule recognizes the red part, there the DNA cutting enzyme comes, and DNA cutting enzymes cuts that particular red part, that is a defective one. And that is how the defective DNA strand is removed. Once the removal part is over, now it becomes the ligase enzyme which comes here, and the healthy DNA strand is now replaced, and red defective standard or the DNA strand is removed. So that is how the entire DNA is now repaired, and that is nothing but DNA editing. So this is basically a CRISPR technology, and this technology has actually not yet been used on a large scale, but it has been practiced in few examples which I am going to cite here. In 2014 and 2015, scientists, they have reported successfully using this Cas9 mice to eliminate muscular dystrophy. This is possible. Second is cure rare liver diseases. The third is make human cells immune to HIV. Now, how to make these cells immune to HIV, that I am going to come later on. Then CRISPR has been used to experiment with gene editing mosquitoes to reduce the spread of malaria. This technology is also used very widely in engineering the agriculture to withstand climate change, and in human clinical trials to treat a range of diseases from cancer to a rare protein disorders that actually devastates nerves and the organs. So there's a wide range of area where you can use this technology. HIV cure through genetic engineering is possible if you focus on CCR5 gene. See, CCR5 negative people are always immune to HIV infection. And CCR5 usually codes for a receptor on the surface of a white blood cell, and it plays a role in normal immune responses. HIV co-opts CCR5 as a way to get into the white cell. So, to block the HIV, we have to really play with this CCR5 gene. And how to do it, let us see in this particular picture. In the picture A, you find that the virus is approaching, HIV is approaching towards the the surface part or the cell membrane of the WBC, and there you find that there are two receptors. Now in B figure, in the first B figure, you find that there are two receptors. The first is the CCR5 receptor, which is called as a co-receptor, and the normal one is CD4 receptor. As we know, these two receptors are actually responsible for attachment of the virus to the surface of the WBC, and that is how it gets introduced into the cell, and that is how the cell gets infected. But now what exactly you have to do is the CCR5 gets mutated, and in the right hand figure you will find that the CCR delta 32 is a mutated CCR5. Because of the mutation, it actually falls down from the surface inside the WBC, and it doesn't remain as a receptor at all. And therefore, this virus is basically searching now for the co-receptor, but it can't get that co-receptor at all. So it gets attached with CD4 receptor, but because there is no availability of CCR5, that particular entry of the virus fails there. And that is how that particular individual is saved from this HIV infection. So those people not having CCR5 receptor by birth, they are very lucky, because they are not likely to get infected with HIV throughout their life at all. But if at all you have CCR5, if you are CCR5 positive, then this kind of genetic treatment can actually bring about the mutations in CCR5 and save you from HIV infections. Next is, and the last technology is transproteomics, that is chip technology. The Affymetrix is an American company which has come up with the genetic chips in the form of a glass slide. And a glass slide is coated with the DNA, which recognizes the messenger RNA sequences of different cells under the test. And therefore, you have the sequence of mRNA molecules, maybe recognizing 60,000, maybe recognizing 1,700 these sequences. And that is how these kinds of mRNA are very important in recognizing it. This particular technology has been very widely used nowadays for detection of life on the extraterrestrial planet, that is either on the Mars or maybe on the moon. Therefore, this particular picture is of Dr. Monaco and other researchers at NASA's Marshall Space Applications or Marshall Space Flight Center at Ella, and they are customizing the chips to be used for many space applications, such as monitoring microbes inside spacecraft and detecting life on other planets. So, a lab on a chip by Dr. Lisa Monaco is doing this. So there are a lot many scientists working in NASA basically on identifying the life on the extraterrestrial land. So Affymetrix, an American company, therefore it has come up with such kinds of genetic chips and for identifying the counterpart of it. Protein treatment for cancer is also possible through this particular chip technology. See, National Cancer Research Institute USA is studying cancer-related mRNA molecules so that they can have the counterparts. They have discovered 5692 genes causing cancer, of which 277 genes are related to breast cancer. This is also possible, and they have done. And the Melio Pharma, which Bayer is developing more than 225 proteins of human treatment. So, human treatment is possible. Identifying the genes for cancer, it is possible, and that is all is because of the chip technology. So, in short, in the last slide, what I want to try to tell you is this life sciences is merging with many different branches like this. For example, biology, then physiomix, then the cellulomics, then biotech, then evolution, then infotech, then ontology, then proteomics, then molecular modeling, then mathematics, then metabolics, then proteomics, then genomics and statistics. So to have the current trends in or research in the life sciences, probably one has to be a versatile one, or maybe it should be a group of these experts together to have these technologies to bring them into a reality. So thank you very much. This is one of my books which has been recently come in the market, available on Amazon. Two sides of the same coin. And this book actually speaks about 45 paradigm shifts in varieties of areas in the field of science, where we were thinking of something, and now because of the new attempts or new technologies, now our thoughts have been totally changed. So these kinds of paradigms are changing very fast in the area of science, in all areas of science, and that is how science itself is known as ever changing and never ending. So here we mark the end of my talk. Thank you very much for giving me this opportunity. Thank you.
Thank you so much,
Yes.
sir, for wonderful presentation and
Thank you.
imparting and sharing your knowledge with us. We really got to
Thank
know
you.
some of the new facts regarding microbiology and the emerging trends in it.
Yes.
Thank you so
Thank you.
much.
Thank you very much.
Sir, can you please turn off the screen sharing?
Yeah.
Thank you so much.
Thank you.
Introducing our next speaker is really a matter of pride and fact for us, all of us. Dr. Dra Rechno Kavuri. She is a professor in microbiology, head of the microbiology laboratory, biology department at Udayana University. She has given various lectures on microbiology and virology and soil and waste microbiology and biocontrol microorganisms for the way during the tenure of her studies and the research period. She has been involved in various programs from the University of Bandung to the University of Udayana, Bali. She has been part of various national and international conferences, webinars, and seminars and workshops. We are truly honored to have you, ma'am, amongst us. Now, I would like to request ma'am to please give her presentation and share her views with all of us. We
Thank
welcome
you.
you, ma'am.
Thank you, Madam Sunar Sharif. Could you send
Thank
my PowerPoint from you? Because I already sent it.
Okay, ma'am. I'll just ask the
Okay.
coordinator to present it. Please give us a moment.
Thank you.
Miss Preeti, can you please present ma'am's presentation?
Yes.
Yes, I'm doing that. Just a minute.
Okay.
Okay.
Ma'am, is my screen visible?
Yes, could you make it enlarged?
Yes, ma'am.
Okay.
Now it
Okay,
is
thank
visible.
you. Thank you very much. Yes. Okay. Uh, good evening, but in Bali is already afternoon. Yes. Today I would like to say thank you for the organized by Microbiology Society of India, Dr. Desmond, and then Saint Xavier College Ranchi affiliated with to Ranchi University, and then Department of Botany Saint Xavier College Ranchi, and then Internal Quality Assurance Cell, and then all the committee who arranged this webinar. Thank you very much for invite me for international webinar, International Microorganism Day. And the title, what I want to share knowledge is about the biodiversity of Streptomyces species bacteria in the Alas Purwo Forest Area, Banyuwangi, East Java, Indonesia. And my name is Professor Retno Kawuri. And then my colleague, which is Professor Made Parmawati, who joined at this moment. And then also my student and Dinda Nurmalik Insani, Nada Karunisa, and Amara Nadim Ayu Putri Karisa, and they already joined in this moment. I'm from Biology Program Study, Faculty of Mathematics and Natural Science, Udayana University, Kampus Bukit Jimbaran, Bali, Indonesia. And then in the slide, this is my email, retnokauli@unut.ac.id. Next, please. And then for introduction, exploration of Streptomyces in the forest area needs to be carried out to find species of Streptomyces that are rare and capable of producing new antibiotics. The habitat of this bacteria is often found in soil in forest area because it has not been touched much by human activities. One of the tropical rainforest areas that has quite high diversity is Alas Purwo National Park in Banyuwangi, Indonesia. Alas Purwo Forest is a lowland forest area located at the eastern tips of Java Island. This forest area is divided into several types of vegetation, namely savanna or grassland, bamboo forest, coastal forest, natural forest, or lowland wild forest, and mangrove forest. Next, please. Exploration of Streptomyces in the forest. With the above condition, the Alas Purwo area has great potential for various exploration, including research of certain bacteria. Then, the purpose of this study was to explore biodiversity of Streptomyces species in the Alas Purwo Forest, Banyuwangi, East Java, Indonesia. Next, please. And then the first location that I then to collect the sample is grassland area at namely Sadengan in the jungle zone of Alas Purwo, and make a sample LA1 till 5. I took the 5 sample with different coordinate point, and I used the Google Earth at the end of last year. Next, please. And for the result, in Sadengan grass area, I found 15 isolate of Streptomyces species. We can see that the morphology of the colony, the size is different, and the morphology different, and then the color a little bit similarly, white to creamy and chocolate. But interesting here is they all have produced soluble pigmentation, and they can see from the we can see into the media. I use the media G-Smart agar. Next, please. Still continue at Sadengan grassland. We can see the age of the colony difference. Sometimes is wavy, sometimes it's round, and then the surface is different. Actually, septomyces have not flat area in the surface. Next, please. And then the second location is the tropical rainforest. I took 5 samples with different coordinate point. Next, please. Interesting. In tropical rainforest, I only found 5 isolated Streptomyces species. Why? Because I took only the sample beside the street. Why? Because if I goes through the center of the tropical rainforest, this a little bit, not a little bit, but it's too dangerous because a lot of wild animal there. So maybe sometimes I will go there if I brave. So maybe that reason, so I only found 5 isolate of streptomyces. This 5 isolate of streptomyces is different from the Sadengan city. Thank you. For the last sample, location sample is a Pedul mangrove forest. This is the mangrove forest at the jungle zone. At that moment, the water recede, so I can took more sample there. Then I took 10 location, I think. So with the different coordinate point. Next, please. Next, please. Next, please. At the Bedul mangrove forest, at that moment is receipt water, and then I found 28 isolate Streptomyces species, but in here the colony different color in the Streptomyces before I found in the 2 location, but a lot of the Streptomyces is same color. Next, please. Here, we can see that the morphological of Streptomyces I found in the Bedul mangrove forest is different with the two location before, and then the color is more variated, more variated. And then we can see in the plates number 2 that they produce soluble pigmentation yellow, and then the others is different color. You can see that the age of the colony is more variated. Next, please. And we can see also that this continue the Streptomyces I found the Bedul mangrove forest. Here, if we see that it look like same, but if we look more detail, it different each other. And then interesting for me to find this Streptomyces in the Bedul mangrove area. Next, please. This is still in Bedul mangrove forest. The morphological of the colony is different. All of the Streptomyces found is different characteristic morphological age and surface with different location. Next. Already as a conclusion, total many as 47 Streptomyces species isolates were found in Alas Purwo National Park, Banyuwangi, East Java, which spread over 3 exploration areas. Actually, I done all of the Streptomyces, totally 47. I done antagonistic activity with several pathogenic bacteria such as Staphylococcus aureus, Staphylococcus epidermidis, E. coli. then Ralstonia solanacerium, Pseudomonas aeruginosa, Salmonella, and they give interesting inhibition zone. Some produce lots of strong inhibition. That's interesting, but I cannot show in here because I still make a draft for publication. And then some of the Streptomyces which has potential continue with the identification molecular with Professor Parmawati. Acknowledgement, thanks go to the unit of research in Udayana University, Bali, based on the letter of assignment like in the slide. Thank you very much for the opportunity for today. And then this is the picture that the Alaspuro forest. This is the enter. If you enter the Alaspuro forest, we can see the gate, and then at the bottom a Sadangan grassland, and then in the center in Bedul mangrove forest, and then this Professor Madi Parmawati who wearing a hat and when yellow shirt, and then me with the pink hat, and then the others is my student Amara Nadine, Dinda Nur Malik, and Nadia Khairunisa. And then maybe if you have a time, please show the video, maybe only 2 minutes or 1 minute. This is a site of Bedul mangrove forest. At that moment, as the water is receded, and then we can took the temple move into the center of the Bedul mangrove. This is dry, not too dry the soil, but a little bit wet. Okay. Yeah, this is also the Bedul mangrove area. Okay, thank you very much for today for inviting me.
Thank you so much, ma'am.
Thank you very much.
Thank you, ma'am. Moving towards our next speaker and guest of the day, Dr. Prasenjit Mukherjee. He is the head Department of Botany, KKM College, Parker. He has 24 years of professional teaching experience in various levels with his active participation in the environment and education conservation. 5 years of experience with NGOs as expertise in imparting training on sustainable waste management and environmental issues. He has published around 93 national and 11 international academic papers. He has a strong communication and instructional skills with special expertise in organizing events, team management, and imparting training on environment education. Excellent leadership and organization capabilities with empathic oratory and public speaking skills. He has been felicitated as Best Teacher for Promotion of Science and Environment on 5th September 2005 by Directorate DAV Public School, Ranchi Zone. He has been honored with World Environment Excellence Award 2017 at Mumbai by World Achievers Foundation and many more. I am very pleased to invite you, sir, to please put up your thoughts and give a presentation to all of us.
Thank you, Sonal. First of all, I would like to thank you for your introduction. And I would like to thank the Microorganisms Society for organizing such a wonderful event, and especially the student of Sanjay College. Right, the students, I'm telling, beside the faculty, that you people are really doing wonders and in organizing event. And it's a kind of revolution kind of thing that every day just I saw that there is a group that you people used to send uh, the activity event list or whatever that. Just I would like to say that I'm unable to share the slide actually. Uh, can it be done? Just a minute. Okay. Uh, Actually, there is a problem. Just I'll show the things. Can it be seen from this? Are you able to see my laptop?
Yes, sir.
Hello.
Yes,
So
sir.
just
Yes, sir.
I'll so
Yes, sir.
so accordingly I'm unable to share, but actually just I'm speaking from my mobile and it is in the screen. There is some problem. So anyway, just I'll be sharing, and I'm not actually telling related to the subject. Actually, just I'm working on the various issues, but solid waste management is one of the important aspect which I have worked, and I have produced a PhD on this in solid waste management. And you know that we are talking about the microbiology, and the microbial degradation of waste is very important. You know that how much waste is being generated and what are the impact of the waste. You know the micros play a very important role in managing this waste, and especially the decomposition of the waste. But I'm not talking about that especially, but I'm talking about the environmental impact with special reference to solid waste management. So is it visible? Hello?
Yes, sir. It is visible.
Okay. So, okay, next, just. Yes. So, major environmental problems are the climate change, the global warming, greenhouse effect, energy, water crisis, water pollution, water logging, scanty rainfall, biodiversity loss, conservation and land use, effluents, chemical, toxic, and heavy metals, air pollution, soot, burning of fossils, industrialization, waste management, ozone layer depletion, ocean and fisheries, deforestation, mining, loss of flora and fauna, invasion of alien invasive species, habitat loss, man and animal conflict, soil pollution, excessive use of chemical fertilizer and pesticide. These are some of the what I jotted down the major environmental problem, and the solid waste management is one of them. You see, I'm just telling about the statistics of solid waste in India. Why? What is the importance of the microbial degradation? You know, just the annual waste generation is 62 million tons every year. Out of that, plastic waste is 5.6 million tons, biomedical waste is 1.7 million tons, hazardous waste is 7.90 million tons, and e-waste 1.5 million tons. And you see just the per capita, just we'll see the decadal growth and decadal generation of the solid waste. So per capita waste generation in Indian cities ranges from 200 grams to 600 grams per day. So 43 million tons, right? 43 million tons is collected, and 11.9 million treated, and 31 million is dumped in landfill site. And you know that it will go to, it will see that 85% of the waste is the kitchen waste or the biodegradable waste that can be easily composted, and the ones the role of the microbes is very important in the decomposition of the waste. So you see just the waste generation will increase from 62 million tons to about 165 million tons by 2030. And it is projected that by the year 2031, the municipal solid waste generation shall increase to 165 million tons and to 436 million tons by 2050. If the cities continue to dump the waste at present rate without treatment, it will need 1,240 hectares of land per year, and with projected generation of 165 million tons of waste by 2031. The requirement of setting up of landfills for 20 years of 10-meter site will require 63,000 hectares of land. So there is a big problem. So I'm skipping some of the slides. So waste, also known as the rubbish, trash, refuse, garbage, junk, litter, and ORT, is unwanted or useless materials in biology. Waste is any of the many unwanted substances or toxins that are expelled from living organisms, metabolic waste such as urea and sweat. Well, according to Environmental Protection Agency regulation, solid waste is any garbage or refuse, municipal solid waste, sludge from a wastewater treatment plant, water supply treatment plant, or air pollution control facility, other discarded material, and solid liquid, semi-liquid, or contained gaseous materials from industrial, commercial, mining, and agricultural operation, and from community activities. Well, biodegradable and non-biodegradable, everyone knows. And another classification, waste can be classified as hazardous waste, non-hazardous waste. So in Ranchi, we conducted a survey, a scientific survey that Ranchi per capita waste generation is 287 grams per day. So what are the impact of the waste? If not managed wisely, affects our health, affects our socio-economic condition, affects our coastal and marine environment, affects our climate. And greenhouse gases are accumulating in Earth atmosphere as a result of human activities, causing global mean surface air temperature and surfaces of ocean temperature to rise. Rising global temperature are expected to raise sea levels and change. Sorry. So changing regional climates could alter forest, crop yields, and water supplies. This could also affect human health. Just a minute. See, changing regional climate could this could also affect human health and many types of ecosystem. Activities that have altered the chemicals composition of the atmosphere, building up greenhouse gases, primarily carbon dioxide, methane. CO2 is released to the atmosphere by the burning of fossil fuels, wood and wood products, and solid waste. Methane is emitted from the decomposition of organic waste in landfills, the raising of livestock, and the production and transport of coal, natural gas. NO2 is emitted during the agricultural and industrial activities as well as during combustion of solid waste and fossil fuels. Well, these are the some point of the context how the solid waste get inside our body: soil absorption, storage, and biodegradable plant uptake, ventilation, runoff, leaching, etc. How you can use this waste, resource recovery, composting. What I told that what is the importance of the microbial that I have incorporated on the day of this microbiology. Vermicomposting, energy recovery, incineration, pyrolysis, gasification, biomethanation, or anaerobic digestion. These are some of the impact of the nausea and vomiting, increases in hospitalization of diabetic resident living near hazard waste site, mercury toxicity from eating, Stavanos suggest. Old books exchange, we must exchange or donate old books, old clothes, old computers, excess building material, all equivalent to local organizations. Develop an office recycling procedure packets. So this is the project just describing in brief. There was a solid waste management project called the Clean Jharkhand project, and there was a component of community action, awareness generation, and environment education for school children, capacity building, policy and licensing, resource recovery. So these are some picture of the Clean Jharkhand project in Ranchi. This is awareness generation. 5 components. There was first was the community action. This is the picture of community action. The second one is the project component, awareness awareness generation, and this is about the environment education for the school children. The several training were imparted, the talk show, etc. These are capacity building training. The several training were imparted to a different set of stakeholders, and this is the policy and hygiene, and these are the major issue addressed. This was the way that the doorstep collection. It is the compliance of the solid waste management of the class 1 cities in India, and it is the solid waste management rule 2016 too, that where the doorstep collection must be ensured, and this is the primary storage of waste. This is transport of waste, and this is transport of waste again, and this is the storage of waste primarily. Just I want to say that Jiri dump site, some of the Ranchi people, they might have visited. This is the waste generation. You see the two pictures are here. One picture you see is the Jiri dump site we have started. This was initiated by our team actually, and this is 2004, and this is the landfill site, the present fill site. I compared and taken the photograph. So this is, I took this photograph in the year 2003, and this is 2021. This again, you can see this how this waste generation, and this is impacting in the landfill. So this is the picture. This is again, you can compare the composting process and how this the decomposition or degradation. So the microorganisms play a very important role. This is the same picture, the different location, the picture, and this is the current picture. Well, this is the just the one very sad part is that the fire, they are putting fire in the dump site, and this is the I caught this photograph during the when the fire was set in the Jiri dump yard, and this is again the same landfill site. How the Rakvikas, this is the composting option, the various windrows composting we initiated in the year 2005 at Jiri dump site. Now these are not there. Now the dump site has been completely filled with garbage. And this is the windrows composting. We were selling it at rupees 1 per kg, this waste. The windrows composting waste, this saving. This is Nadex composting. And this is the vermi composting process. And this is still existing in Chutu, near the BIT Mesra. This is actually, and this is a drain cleaning. And this is the collaboration of GeoNGO with the eco clubs in the schools. And this is environmental club in the community. And this is Toxo in the institute. And this is presently this picture. This is Urban Kheti. It's a new initiative by a group of people. I'm also there. So this is Urban Kheti. We are distributing. This is jar, and it is composted at the home itself. And this is again, I can say that this is microorganism plays a very important role at household level. And so one picture I want to show. This is waste bank. This is my model actually. Earlier I was in KK College Pakur. Now I'm in KO College Gumla in Ranchi University, under Ranchi University. Earlier I was under SKM University Dumka. Now at last November I am transferred to Gumla. And this was the model I prepared, and this is waste bank I used to call it. And this is replicating in the colleges. So thank you very much for giving an opportunity. And sorry, just I couldn't share the slides in a proper way. Thank you so much.
Thank you so much, sir. No issues. Technology is somewhere or the other creating problems for us also as we are growing. Since childhood, we have been told to mold ourselves. So somewhere or the other, it is okay. It was a wonderful presentation, sir. And thank you so much for giving us your valuable time. Thank you. Moving towards our next guest of the day, Dr. Prakash M. Halami. He is a chief scientist, professor ACSIR, and head microbiology and fermentation technology department, as well as head Department of Food Protectants and Infestation Control, also a nodal officer, CFTR COVID-19 Testing Center at CSIR, Central Food Technological Research Institute, Mysore, India. He is also serving as an expert advisor. Akshay,
Excuse me, ma'am, आपका mic mute हो गया है.
Okay. I am really sorry. Okay. Just give me a moment. Yeah. Now am I audible?
Yes, we can hear you.
Okay. I'm really sorry, sir.
No issues.
Okay. Dr. Prakash M. Hilami. He is a chief scientist, professor at ACSIR and head microbiology and fermentation technology department, as well as head Department of Food Protectants and Infestation Control. He is also a nodal officer at CFTRI COVID-19 Testing Center at CSIR, Central Food Technological Research Institute, Mysore, India. He is serving as an expert advisor at Lakshya Patra Research Lab, Bangalore, and he's also a member of Advisory Board, Institute of Excellence, University of Mysore. He has nearly 3 decades of research and development experiences besides HRD activities. He has so far guided 80 postgraduate dissertation students and 12 doctoral students. He has 123 publications in peer-reviewed international journals, where 4 Indian patents have been granted to his credential. He has presented 180 papers at national and international conferences, winning 22 Best Paper Awards. We are really honored to have you amongst us, sir.
Thank you.
I really welcome you for your presentation.
Thank you very much, Sonal, for a nice introduction. I hope you can hear me.
Yes, sir.
Wonderful. So, first of all, thank you very much for all for organizing this event, Microfest at St. Xavier College, Ranchi. In fact, this is the second time I am giving a webinar. 2 years back on the same day, I was one of the guests to talk on the International Microorganisms Day, and that time I spoke actually on food grade microorganisms. So this is very nice that Microbiology Society India, in collaboration with St. Xavier College, has taken up this day to celebrate as a Microfest. I really salute our your organizing team as well as the Microbiology Society, Professor Deshmukh, and I could see most of our seniors are there. Professor Shailesh Dave is there, Swaranjit Singh is there, as well as Dr. J.S. Virdi is there. In fact, I have not prepared the slides because I thought to address Microbiology Society, I don't think I should have a real slides to prepare or present. So I thought I will just share my views and opinion of last 30 years in working in a microbiology rather than touching with life sciences. I thought how microorganisms are really contributed in understanding, and then ending with what type of research actually we do in a microbiology department in CFTRI, which I am actually heading from last 3+ years. As we know that microorganism we use, for several long time for the several scientific studies as a tool, which facilitates because of their much more simpler lifestyle, easy to propagate, the breeding, obtaining the mutants, and then several other applications. And this is how actually microorganism has been used for the several scientific studies in a biology from a very long time. In fact, this month beginning, I was actually, in fact, in the Pasteur Institute in Paris, and it was really a wonderful experience how the Pasteur contributed in a microbiology in general and in a fermentation technology in particular. So this is what actually the next point which I want to cover is about the industrial production of several metabolites. There are several organic acids, the enzymes which we look, the amino acid which we use has revolutionized from the industrial microbiology perspective. And we look for the several food grade microorganisms for this purpose, right? Similarly, the microorganism has been used as a tool, as a treasure for several useful genes, whether it is the E. coli or thermophiles or the extremophiles or from the any biosphere microorganism we use, and then see how their enzymes are, whether they are mesophilic, extremophilic, thermophilics, and then we actually bring them in our industrial production in a different ecosystems. Okay. Similarly, the microorganism used as a model system to study several metabolic pathways and processes. As I told initially, because of their simplicity in the life, the biosynthetic pathways, engineering the microorganisms, and looking them as a cell factory specifically, which is not that easy if you use, suppose, the higher organisms, right, plants or the animal system. Microorganisms facilitates much more in a better way, and that is what actually one can use them as a model system to study several metabolic pathways, and that's why actually in food microbiology or in industrial microbiology, we will be dependent on the several microbial processes to produce such metabolites, whether it is the enzymes, the carotenoids, the food additives, the amino acids, and then several, or even if it is the engineered microorganisms also. Recently, though nanotechnology is not as a frontier area today, but then definitely bio-nanotechnology, where we use actually microbial systems to synthesize several nanoparticles for this purpose, right? What actually we are looking in a microbiology department or our present research activities, perhaps that can be more interesting. So one of the area which we are looking is the clinical trial of the probiotics. And how this actually matters, because we know that several of the probiotic microorganisms are being obtained or taken from the overseas laboratories, whether it is a Lactobacillus rhamnosus GG or Lactobacillus casei strain serota, or even the Bifidobacterium in animalis, right? BB12 culture. Why we don't have the native microorganism for this purpose? And this is what actually the microorganisms are the probiotic bacteria, if they can be obtained or isolated from the Indian gut, they will have better adaptability with the native populations, and they can actually perform better with reference to giving the various health benefit. As we see, the increasing number of the lifestyle diseases, the increasing number of the diseases of the civilizations, the only way is to combat them is through the microorganisms or through the probiotics, in other words. And once we have such a probiotic organism, especially their industrial production, this is actually the major challenges these days, because once we say the probiotics, it is naturally the question comes with reference to the viability of a probiotic bacteria, except the bacillus group, is a question, and especially with reference to the bifidobacteria. Not many laboratories in India actually are working on the bifidobacteria. If you take a bifidobacterial culture also from any culture collection centers, viability as well as their authenticity or purity is a big question mark. What are the formulations which we are available in hand are only through the drug or in the form of capsule. And the bacteria which we get from the capsules, the viability is really the question mark. And as I said, that for the probiotic efficacy, one has to have the minimum therapeutic viability of 10 million cells. And this is what actually most of the probiotic that do not meet this criteria. The next one is within the probiotics, there are health benefits. In recent past, several of the probiotics has been blacklisted by FAO and IPSA, European Food Safety Authority, and FDA, Food and Drug Administration, USA. The very simple reason, because of insufficient income health claims. Most of the probiotics for the health claim has been given by the authors will not be able to fulfill those requirements. And that's why actually we have to look for their industrial production, for their large scale production to meet our domestic demand. That is what I can say. In this context, we have several such pilot scale facilities. Unique Biotech is one such laboratory based in Hyderabad, produces a probiotic culture in a fermentation in a large quantity. Several such facilities are there around the country also. The newer biopreservatives, and this actually aspect is very much related with the food microbiology or in the food technology. The newer biopreservatives is concerned. Why? Because right now, the Nisin is the only preservative available for canned and dairy products. But we know that Nisin can work only in the acidic pH and can be used mostly again in the fermented food. Where in a fermented food, actually because of their, suppose if it is acidic fermented food, you basically don't need much the preservative, because the natural acidity itself prevents such contaminating microflora. But definitely, if it is the processed food or if it is the alkaline fermented food, we actually need several such biopreservatives, because the conventional food preservation processes where we use chemical preservatives, they are carcinogenic, mutagenic, and in general affects our health also. So looking for such a biopreservative from microorganism is very important aspect. I remember that other previous speakers covered very quite intensive areas, especially Dr. Ranjan Garge. But one of this area which actually we are looking, or the whole world is looking from the microbiology, is the gut microbiome. And then food, and as I am from the Food Technology Institute, it is the gut microbiome and food host interactions. What is actually the response of the human genes when we consume the different food. Now, this being a international year of millet, when we are actually consuming the millet, how our digestive system reacts, how our gut-brain axis actually works, and with reference to the millets, which are actually found to be very suitable, especially for the diabetics, for reducing the blood pressure, minimizing the cardiovascular strokes, then including the malnutrition deficiency, right? And this actually millet-based product provide a very good nutrients first for our gut, right? And that's why actually another day, one of this MBSI conference, I was in fact telling that the type of food which you eat, actually our gut actually act as a one petri plate to grow microorganisms, allows to grow the bacteria or the microorganism based on what type of food you eat, and that's why millet consumption is a very healthy idea and to be encouraged and to be practiced, in other words. Development of high scale up of alcohol production in the indigenous fruits and vegetables as well as the grains, because the energy now, or the bioethanol production or the biofuel, is one of this important aspect. Looking such yeast cultures, which will help us to produce the ethanol, is a very vital, because of the natural fuels are getting exhausted, and bioethanol in otherwise also is being produced and is being used as a in the vehicles, and from that angle, utilizing the several our fruits or the vegetables or even the grains. Sorghum actually is used even in the Maharashtra for production of the beer. So several such grains can be brought into the practice for the production of alcohol, so that our need of the alcohol or the fuel gets fulfilled. Machine learning and artificial intelligence and machine learning are the other two important areas which actually can be brought in the life sciences, and particularly in the microbiology, with reference to screening the microbial cultures, optimizing the parameters, harvesting the product, downstream processing, and even at the using the product more efficiently. And this is how actually AI and ML is playing a big role, along with the 3D printing. Another important area actually in the microbiology we see is the microbial metabolites like microbial pigments. Again, the food grade pigments, especially from the rhodotorul and several other fungus, that actually can be enhanced or produced in a large scale in the fermenter, so that we actually minimizes the use of the synthetic pigments. Use of the metagenomics in the microbial systems, several of the hotspots, hot springs, has yet to be characterized. Our gut microbiome, our gut microbiota has to be characterized, where the metagenomics will play the important role. The Western Ghats or the marine ecosystem or the several naturally fermentation processes even, that has to be actually investigated for the purpose of identifying especially non-culturable microorganisms. And this large number of non-culturable microorganisms, especially establishing their role, what they do, and how is their occurrence actually. And in this regard, through the metagenomic approaches, several of the microorganisms which has been detected in our gut microbiota has been considered as a next generation probiotics. Enhancing the functional properties of the fermented food using the defined starter cultures. So this is another area of interest, because most of our fermented food, which we are actually rely only on the natural fermentations, very few cases are there. We do not have the defined starter culture, just like what we have the defined starter culture from the yogurt or for the sauerkraut or for the kimchi fermentations. We do not have such a starter culture for the idli or the dosa or any other fermentation. If you go to the northeast part of India, you will see that several vegetables are being fermented naturally. The bamboo shoots are fermented naturally. The soya products are fermented properly. Perhaps if we can look at defined starter culture or processes of fermentation will enhance, we will get the uniform consistency in the finished product. We will have a better quality of the product with the guaranteed safety and shelf life. We will have a product, a labeled product with the with the non-nutrients contents. So such processes where we have to actually depend on the defined microflora. In my laboratory, we actually works on the additional other two aspects. One is on the genome editing and the genomics of the probiotic bacteria, where we are looking for the genome editing of few of these antimicrobial peptides for their iron production. And this is actually, in fact, covers the area which I was talking with relation to the biopreservative. Now here, through the genome editing, as Professor Ranjan Garge also has told, how genome editing actually has the impact and being used in several of these plant systems. We are looking actually in the probiotic bacteria, whether genome editing can help to make the engineer the microorganisms to produce required metabolites or the given metabolites in the large amount, and then we know that the product which we obtained through the genome editing tool is not considered as a GM. So their utility in a food will be definitely is useful or will not have much constraints. Similarly, the antibiotic resistance, which we are actually working in the lactic acid bacteria, because lactic acid bacteria being a natural microflora of our gastrointestinal tract, they found to have, they are actually intrinsic resistance, and they harbors actually, or they are actually called as a natural reservoir of antibiotic resistances. So we are actually looking how this antibiotic resistance is evolved in a lactic acid bacteria, how they actually transfer the genes in a different pathogenic or among the lactic acid bacteria, how the antibiotic resistance actually is being spread or coming in our food chain or in the food system. This is another area which actually we are looking. I hope my time is over. Otherwise, this will be never ending. I actually look forward to listen the other speakers and participate in an interaction. Once again, I thank the organizer for giving me this opportunity, and I look forward for hearing the other speakers of the MBSI society as well as the other colleagues, those who are joining. To interact, I will be actually happy to interact with them. Yes. Thank you. Thank you, Sonal. Are you there?
Yes, sir, I'm there.
Wonderful.
Thank you so much, sir, for giving us a precious time. And it was like nice to hear you. We were just waiting. We were on the sync that we'll be listening to you continuously. But thank you so much for sharing all the knowledge that you have gathered through your experiments and your experiences. Thank you.
Yeah.
I think Ranjan sir has something to say. So please.
No, I'm okay. I was just listening. Yeah.
Okay, sir. Ranjan sir, actually, I have texted you.
Yes.
Reply to my message.
Yes.
Okay. Thank you.
Okay. Did you like it?
Definitely.
Thank you, sir.
Thank you, sir.
Thank you so much, sir.
Thank you.
Moving forward with the webinar and the session, it's now time to invite Dr. Girish. He is the Dean of Krishna University of Allied Sciences, Krishna Vishwa Vidyapeeth, Karat. Having more than 40 years of experience in teaching and research, he owns more than 175 research papers which are published in peer-reviewed national and international journals. 15 PhD students have been awarded PhD under his guidance. Dr. Girish is a member of various academic bodies and has been part of many conferences, webinars, national, international workshops, and various functions as well. We are delighted to have such an eminent personality amongst us, sir. We welcome you all with all the love and the gratitude from our side. Please enrich the gathering with your powerful words and the session. Thank you, sir.
Thank you, madam. May I request, madam, to share my slide from your side?
Yes, sir, sure. Ms. Preeti is on it,
Thank
sir.
you.
Yeah.
Hello. Am I audible?
Yes, sir, you are.
You are sharing?
So Ms. Preeti is sharing the PPT. Is it fine?
So is the screen visible?
1 minute. Can you see the slides?
Yes, slides visible.
Slides are visible. Okay, then I'll start.
Yes, yes.
Yeah. First of all, I congratulate the organizers, especially Microorganisms Society India has made the paradigm change in the awareness among the students about the microbiology. Dr. M. Deshmukh and team. And secondly, the organizers of this particular event from the St. Xavier College, Ranchi, Department of Botany, Srivastav sir, Sonal and Prityan, all whole team. I congratulate you all. And in addition, I express my sincere thanks for giving me this opportunity. Here, I listened to some speakers, Ranjan Garge and others. Sir Garge talked about the advanced field that is in the molecular biology aspects. Mukherjee talked about the applications of the microorganisms, especially in the waste management. And Dr. Halami sir, he talked about the probiotics, that is the applications of the microorganisms. I thought that I should select such a topic that there are amazing microorganisms in the nature, and these microorganisms probably some of the students may be knowing some of the groups, but some students may not be knowing some of the groups. So I thought that I should discuss these topics so that the awareness among the students as well as interest in the students in the field of microbiology. When we talk about the microorganisms and their groups, you will find that. Hello. When we talk about the groups of microorganisms, you will find that the whole world is teeming with the microorganisms, and there are different groups. There are different groups like microbacteria, yeast, molds, viruses. Next, you are changing the slides, madam. You are changing the slides?
No, sir. Right now, this was the second slide.
See, the participants are requested to
Just
keep their mics off.
Yes,
Keep the
just
mics off.
Hello.
yes. Just a second, sir.
Huh.
Now I'm presenting, sir.
Second slide.
Yes.
Please.
This one,
World of
this one, sir. Yeah.
World of microorganisms.
Is it
So,
visible, sir?
For me, it is visible.
Hmm.
Okay. In the world of microorganisms, you'll find that common groups of microorganisms, that is bacteria, viruses, fungi, that is yeast and molds, algae and protozoa, they are the commonmost microorganisms. And if you find these microorganisms, they are present, they are ubiquitous and they are present everywhere around us, in us, inside the water, soil, everywhere in air, they are there. Even in the stones also they have found the microorganisms. And I have shown here one just to show the microorganisms which are found in a single drop of water. All kinds of microorganisms are found in a single drop of water. I was my intention was to show that everywhere microbes are teeming around us, and generally there is a wrong notion in the society that the microorganisms means they are dangerous, they are harmful, but that is not the case. 99% microorganisms are useful, and only 1% microorganisms they are involved in some damages to the mankind. And these microorganisms, if you learn the language of these microorganisms, then you will find that there can be a happy coexistence between the microorganisms and the human being. Can I have next slide, please?
Yes.
Is that?
Yes, sir.
That is the potential of microorganisms. Now, just I showed the groups of microorganisms, but what is their potential? Louis Pasteur has said that never underestimate the power of microorganisms. Microorganisms are far more powerful than the king, the human being, considered the king of the planet. These microbes are far more powerful than the even human beings. If we know that microorganisms, if you talk about the origin of life on this planet, as we know that origin of life has taken place as a single cell in the water body and in the form of, say, microorganism. So it has evolved. It has originated as a microorganism, then it is evolved to the multicellular, up to the Homo sapien, the human being. So when time will come in the future about the extermination of the life from this planet, if we go with such a hostile conditions we are creating in the environment, then the higher forms will go first. Microorganisms, they will go at the last. So that indicates that how these microorganisms are very powerful. Second aspect of these microorganisms about their potential, they are highly adaptable to the rule of nature.
Excuse me, sir.
Smaller in size.
Excuse me, sir. I think there's a disturbance. Let me remove that person. Excuse me, Rashika, ma'am. Can you please turn off your mic?
Thank you.
Rashika, ma'am. Yeah.
Thank you.
Just a minute.
Microorganisms are highly adaptable in the nature. If there are drastic changes in the environment, still microbes, they easily cope up. You will find that the microbes, if the temperature variations are there, or any type of the variation, drastic variation in the environment, there are still microorganisms, they adapt. It is the rule of nature. As the system becomes smaller and smaller, they are highly adaptable. And a human being is the least adaptable. This is the potential of microorganisms. Microorganisms, they are involved in the elemental transformation in the nature. All these basic elements, they are as carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, and other micronutrients. They are constantly immobilized in the form of the living system. And after the death of the living system, it is necessary they should be recycled, they should be mobilized. And that role is played by the microorganism. That is a tremendous potential in the microorganisms. Then they are involved in the production of variety of compounds. Halami sir also, they have told that they are involved in the production of antibiotics or organic solvents, biofertilizers, biopesticides, and pickles and food products. Everywhere these microorganisms, they can be used. Only thing is that you should learn the language of microorganisms. These microorganisms, they are involved in the damages. I am not saying that they are causing damages. They are involved. And in the disease causing also. Microbes are not directly responsible for disease causing, but two things are very important. The number of microorganisms, their virulence, and most important is the body resistance. If your body resistance is very strong, then microbes will not cause the disease. If they are present, still you can resist these microorganisms. And if you look at the potential of microorganisms, you'll find that if the microbes, they go on strike, what will happen? The situation will be like this, that everything will be stuck and no living beings, higher forms will survive on this planet. Can I have next slide, please?
Yes, sir, it is.
Now I want to talk, as I told, that I want to talk about the amazing world of microorganisms. There are such amazing microorganisms that many times the people, they don't know. First group is the smallest bacteria, smallest microorganisms and largest microorganisms in the size wise. The smallest microorganisms in the size, they are we call the Mycoplasma group of organisms. One of the examples is the Mycoplasma genitalia. Size is just 100 nanometers in size. While the largest bacteria which are there, we say microorganisms means they are not seen by naked eye. We require microscope, but that is not the case. There are some microorganisms which are large in size. For example, Thiomargarita, this bacterium, Thiomargarita namibiensis. This bacterium's size is 2000 micrometers. 2000 micrometers largest, and the smallest one is the 100 nanometers. This range is there, and these microorganisms are found. It is said the Thiomargarita size is like eyelashes size is there. And these microorganisms are very, very important in the nature. They are found in the sediments in the mud near the seashore. Second group is the bioluminescent bacteria. Beautiful. If you look at Tare Zamiper, when these microorganisms, they grow on the plate, they shine in the dark. They shine like stars. And these bioluminescent bacteria, they are there. They have got tremendous power because this bioluminescence, this capacity is there only with a few microorganisms and few animals also. They are there where they have got the typical enzyme that is called luciferase enzyme. This luciferase enzyme is it is owned by the animals, but in bacteria they have got their own luciferase enzyme, and for production of 1 photon of light, about 8 ATP molecules are required. Tremendous input of energy is required, and only some of the microorganisms they have got capacity. And these microorganisms mostly they are found in the marine environment, and these microorganisms they should be studied further. They have got tremendous applications in the different fields. Environmental field is there, medical field is there. It's the Lux gene that can be used in the genetic engineering also. Next group of microorganisms is the nitrogen fixing bacteria. That is very common. We know Azotobacter, Rhizobium. But why I am showing these organisms as example, amazing microorganisms? Because nitrogen fixing capacity is there with the microorganisms, and that is not with the plants. Why it is with the microorganisms, and that to some of the microorganisms only. Another thing, the energetics. You'll find that the ATP required for the single dinitrogen to convert to the ammonia is about 12 ATP. Huge amount of input of energy required, and that is not affordable to all microorganisms. So only few microorganisms they are evolved to have this capacity. Again, these microorganisms they should be studied, they should be exploited. Next group of microorganisms is the autotrophic microorganisms, autotrophs. These autotrophs are the extreme synthesizing microorganisms. We know that most of the living beings, they utilize the organic compounds, and from these organic compounds they take the energy and the nutrients, and finally they convert that into final product CO2 and water. But these autotrophic organisms, they use CO2 and water, and from that they produce the compounds, and they are found in nature, and they should be used in the nature. Next slide, please. Next group of organism is the extremophiles, organism growing in the extreme environmental conditions. For example, temperature extremities. Some organisms they can grow at the minus, that is sub-zero temperatures. That is they are called saccharophiles. Some of the organisms they grow at higher temperature. That is the extreme thermophilic organism beyond 100-degree temperature also they grow. Some of the organisms they grow at extreme pH ranges. That is a pH of 2 pH, for example, Thiobacillus. There are some organisms which can grow at below, that is pH around 11, for example, Bacillus pasteurii. Then there are microorganisms which can grow at high salt concentration. Microbes that are found in the high saline concentration, that is in the marine environment or others. So saline soils also, for example, Halobacterium is there, Halococcus is there. Then they are found in the some organisms like 1 minute, requiring high sugar concentration. They are called osmophiles. and organisms which are growing at the high pressure. For example, in the marine environment, tremendous pressure is there. In that environment, they grow. Their cultivation in the normal laboratory is not so easy. Organisms which are growing at when they are exposed to high radiations, and that is another organism which to be tapped and studied very well. For example, Microorganisms radiovorans and such organisms. And there are some protozoans also like Bodo, where they resist tremendous dose of radiations, and there are some microorganisms which can tolerate toxic compounds like pesticides, and they degrade these microorganisms. And the last group which I want to just enlighten is the Delovibrio. That is called living antibiotic, and this microorganism has a tremendous capacity to destroy that is gram-negative bacterial pathogens. So these are some microorganisms which are amazing type of microorganisms having tremendous capacity, and I wanted just to enlighten the students that they should study these microorganisms in the laboratory, and that will create the interest among the students. Take into consideration the time factor, I had to go a little bit faster. Thank you, madam. Thank you, Sondal madam. Hello.
Yes, sir.
Thank you.
Thank you so much, sir, for the wonderful presentation. Thank you. Moving towards our next speaker, and I now invite Dr. Rajendra Kumar Roy.
Excuse me, ma'am, आपका mic mute हो गया है.
I don't know. It's getting clicked again. Now am I visible and audible?
Yes, ma'am.
Okay. Moving towards the next speaker, I now invite Dr. Rajendra Kumar Roy, Head of the Department, Zoology, St. Xavier's College, Maharo, Dumka. He has been teaching here for last 12 years, and he holds specialization in ichthyology and areas of research in ecology. We are happy to have you amongst us, sir.
Respected Father Novar Lakra, Principal of the St. Xavier's College, Ranchi, and patron of this fest. Dr. Deshmukh, co-patron of this fest. Dr. A.K. Srivastava, Head of the Department of Botany, St. Xavier's College, Ranchi. All the resource person, chairperson, and participants coming from different parts of India and abroad. Myself, Dr. Rajendra Kumar Roy, Head of the Department of Zoology, St. Xavier's College, Dumka, standing here in front of you to congratulate the International Day of Microorganisms. First of all, good afternoon to one and all. Today is the day of International Day of Microorganism. Microorganism is very important and effective organism in this world. Just before me, some sir told that some microorganisms are useful and some microorganisms are harmful. But in these days, from few years, we are seeing that different diseases are arised by the microorganisms. In last week, I have also seen in the news that some virus developed in Kerala and created disease. So study of microorganism is very important. Some microorganisms also found in boiling water and in fire also. These are the temperature resistant microorganisms. So its study is very essential. Early, the biology or life science was only limited to the botany and zoology. After some times, microorganisms become added in that group. And these microorganisms, this microbiology deals with the microorganisms which are very useful to the people. It creates maximum diseases in plants and animals, including man. Now, in school level, the study in the life science is limited to the structure and function, matter and energy in the ecosystem, their life cycles, their evolution, and their heredity. But in university and colleges level, the life science become a broad branch. It includes anthropology, which deals with the human race and human development. That is, next, zoology deals with the animals, botany deals with the plants, microbiology deals with the microorganism, ichthyology deals with the fishes. So in university level, master level, many branches become developed which are useful to the students. Now, during these days, we are seeing some amalgamation in the subjects. These amalgamations, just like the biophysics, this biophysics deals with the instrument developed by the physics, and it is useful in the biology or life sciences in the diagnosis and test of different diseases. Another amalgamation is the biostatistics. This biostatistics helps us in the studying and computing the animals and plants. Another amalgamation is found that life sciences with the pathology or healthcare, and it also includes some pathological instruments, some medical insurance companies, some healthcare management also. So this branch gives help to the other branches also. Now, in 2023, new technological trends are developed. These trends are artificial intelligence and machine learning, robotic processing and automation, edge computing, quantum computing, virtual reality and augmented reality, blockchain, Internet things. These are the recent trends in 2023. Climate change and new mobility and digitalization are the major challenges for the life sciences. They will shape our future life and make it unequivocally clear to us that a fresh way of thinking will be inescapable in many fields. The new technology in life sciences are artificial intelligence, clinical trial management, population healthcare management, and diagnostics. In these years, in colleges and university, it is my experience, I am seeing that the mental status of the students become decreases. Why? I don't know. It may be the effect of the society, it may be the effect of the culture. But it is a matter of research and study. A seminar on the topic Emerging Trends in the Life Sciences, organized by NMKRV College for Women, Bangalore on 4th November 2022. In that seminar, large number of professors and students were participated. Long discussions and long lectures delivered by the professors, but finally they came on conclusion that we have to include a new branch in life science, that is the mental health of the students. So we have to think about this. The mental of the students is decreases and it needs to include our life science subject. Now, if you will see the commercial point of view, in commercial point of view, life science is a fast growing market in India. It will develop in multifold growth by 2030 because the population increase rate is very high. So we have to think about these things and we have to implement. It is my short lectures about the recent trends in life sciences. Thank you. Thank you all. Thanks.
Thank you so much, sir. Introducing our next guest is really again a matter of honor and pride for all of us. I feel privileged to introduce him to everyone. He is currently the assistant professor in botany department and he has been part of many orientation program, seminars and workshops. He has been the organizing secretary of the 11th National Seminar in the year 2012. His various papers have been published in the national and international journals, and he has been also awarded with various honors. He has been the state president of the Microbiologist Society. Yes, we are honored to have Mr. Avdesh Kumar sir with us today. I invite sir to please kindly share his experience with all of us.
Thank you, Sonal. Am I audible?
Yes, sir, you are audible.
Thank you very much. Can you share my slide from you? I have sent it on your mail.
Yes, sir. Ms. Preeti, can you please share sir's slides?
Just give me a minute, ma'am.
Okay. So please bear a moment with us.
Thank you very much. I'm Dr. Avdesh Srivastava from Chhattisgarh. And it is a matter of proud that a student has organized this. As yesterday I've talked with Mr. Ajay Srivastava sir. He told me that students are organizing. This is a very good, wonderful experience. Thank you. Yes. As my first slide shows, is starting with tobacco mosaic viruses in viruses up to COVID-19. And in lower portion, I want to say that the products starts from dahi, that is curd, to co-vaccine or Covishield. We have passed a very dramatic pandemic since last 2 years when this trend comes into existence, that we are on webinar mode. Previously, we are organizing seminar, but now it is converted to webinar, which were created by the multinational companies. Next slide, please. As per the scope of microbiology is concerned,
As our earlier speaker has told that a single drop of water can be an ecosystem. So microorganisms are omnipresent creature. They are present in human, animal, plants, and all other living creature. And three layer of the atmosphere, that is soil, water, atmosphere, and microbe can multiply in these three habitats, and they also present throughout the planet, because in seminar we have studied that in different layer of this atmosphere they were also reported. So microorganisms are relevant to all of us in a multiple of ways, because it influences our life directly or indirectly, that is beneficial as well as harmful. Next please. Next slide please. Next slide please. Next please. Because yes Preeti, yes. As the ecosystem is concerned in unit area, the living interaction between living and non-living, and they are broadly considered as micro producer, decomposer, because they are responsible for the recycling of the ecosystem, that is mineralization, energy cycle, everything is created by the microbes. Next please. So, what are the scope of microbiology nowadays? As our earlier speaker has told that so many branches are emerging from microbes: genetics, chemical technology, biotechnology, dairy industry, agriculture, medicine, and so others like nanotechnology, water industry, immunology, food technology, etc. Next, please. Uh, just our one very wonderful speaker has told so many things about the probiotics, fermentation, the food intestine, microflora, probiotics. So this slide is not need more explanation, as our earlier speaker has already told. So we move for the next slide, please. Uh, Starting from dhokla, dosha, including vinegar and so many wines and the beer, rum, vodka, everything is found by their products. So they are not only restricted to our food industries, rather they are also included our recreation industry. Next, please. Yes, they are the different microbiology branch, which is already told by so many speakers, like CRISPR technology. And nowadays, CRISPR technology is also a type of gene therapy type of branch, on which in 2020 Nobel Prize has also been given. And the person who was working on HIV is in prison. That was also very important. Next, please. Yes. So industrial microbiology, the natural product, the secondary metabolite, the primary metabolite like citric acid, acetic acid, lactic acid, vitamins, ethanol, acetone, butanol, and biopolymers, biodegradable biopolymer like PHB, the products of the bacteria is now producing biodegradable plastic, and the several type of food products are also being produced by them. Next, please. Next, please. Yes. So future of microbiology is not a limit, rather it is expanding in such a way that they were present since Earth was formed, the cyanobacteria, the conservators, and they are still surviving, and they will survive even in future, as last 2 3 years back everyone has felt. And again there is alarming situation in the Kerala. One known again no new virus has not new, but rather it was reported in 2018. Again their strain is coming. So the future of microbiology, because if you see about the economic pattern of the USA and China, nothing is more to say about the future of microbiology. Everyone is stand about this one. Next, please. The cyanobacteria, they are very important because they are considered as one of the most important organism for sustainable management, sustainable development, because they fix nitrogen, they decomposes waste material, they also detoxify heavy metals, pesticide, and other xenobiotics. they catalyzes the nutrient cycling, they suppresses growth of so many other pathogenic microorganisms. Rather, they are considered as PGPR, plant growth promoting rhizobacteria, a very emerging new branch explained from the microbes. And lot of work has been carried in foreign, including the Philippines. There is a lot of papers are coming on PGPR, even in India, Banaras Hindu University. Peoples are working like Basam Panzer. And next is hormone production, enzyme production, uh, and so many other things. Next, please. For suppressing them, there is a word which is commonly used because most of your students, including the speakers, so I'm basically confined to myself to the students, that infection refers to an invasion or a body by harmful microorganism or parasite, and the severity ranges from mild to fatal. uh, and what are disinfectants? So those chemicals which destroy the disease-causing pathogen or harmful microorganism are considered as disinfectant. Next, please. Next, please. Infection are very common, as we have seen in the list: viral, bacterial, fungal, parasitic, like mycoplasma, actinomycetes, etc. Next, please. There are 7 causes of the infection. It may be airborne, bloodborne, sexually transmitted like HIV, fecal, oral, environmental, and few are, spread by the animals. Like bat is now spreading a new strain of virus, and COVID-19 was also considered that they were also spread by the bats. Next, please. So this is a droplet and how inoculum is being spreading. This is a model. Next, please. So besides so many other type of disinfectants like biocide, germicide, sanitizer, disinfectant, sterilization, and detergent, nowadays we are using a new thing which is known as plasma. Next, please. Which these are the biocides and germicides. We have a limited time, that's why I'm escaping. Next, please. Next, please. Sanitizer. Everyone is familiar with disinfectant. Is everyone familiar with you? And next, please. Next, please. So I'm coming to that final product, that is everything is being used for the sterilization, detergents, because the first year student usually use the detergent for sterilization and washing of the utensils in the laboratory. Next, please. Nowadays you are using plasma, some physical like microwave, gamma radiation, freezing, um, and even the cryopreservation. So the physical disinfectants are so many types. Next, please. Yes, they are classified into several groups, and the classification of chemical disinfectant, they may be unique for, because some of them are organic matter, pH, and water hardness, and they are the different type of disinfectants. Next, please. Yes, this is the new technique for sterilization, plasma, because it also reduces the, and I think all of you are familiar with the plasma, because in next slide, I have, in next slide, uh, there is 3 stages of the matter, usually considered as solid, liquid, and gas. But when a solid is heated, it is transformed into liquid, and then it forms into gas. And when of energy, whenever applied to the gas, it is ionized as gas and considered as plasma. And the fourth fundamental state of the matter is the plasma. So plasma is very important because it contains reactive chemical species such as electrons, ions, neutral molecules, and atoms, as well as charged species. So in addition, emission, radiation, ultraviolet, and other infrared region, they are usually developed by the plasma generation. And in next slide, there is a model for plasma generation. And the next slide, how we can use this plasma is being illustrated. So they are the different type of disinfectant sterilization process, which is very needed, because when COVID-19 was spreading, we were very much afraid with them, and we are using sodium hypochlorite and so many type of disinfectant to save ourselves. but this is one of the most authentic technique which may be used when again such type of diseases comes to you. Next, please. Yes. Uh, Everyone makes the house, and we have seen that after 25 year it again requires some repair. So nowadays we are developing bioconcrete, and the bioconcrete are working on the principle that the carbon dioxide and the calcium carbonate crystals, they have such type of microbial solution that never permit carbonate participation or precipitation. So they blocking the pores and cracks. So if they blocks the because nowadays we are using when I was just a child like you, the students, at that time my papa brings a calcium oxide and pour it into a bucket, and that is converted to calcium hydroxide, bhatra chuna, kali chuna, or where the term which is used for them. And after mixing with some needle or tin type of thing, that was given in the houses. And whenever we touch with them, our total cloth was looking white. So that was a situation. And nowadays, so many paint companies are making such type of chemical which can last for 10 year, 15 year, even in 20 years. And so many atmospheric weathering rocking has been created by, I mean, they can prevent the natural weathering and cracking of the concrete. So it is a very important alternate approach for existing concrete. And very soon we will in position to make such type of bar concrete. Next, please. Next, please. Yes. The fungal infection, most of the fungus are beneficial, rather few are very infectious. And if you think about the principle of self-healing nature and production of calcium carbonate crystal with the whole help of microbial solution, they have calcium yes. Yes, please, next slide, please. Okay, perhaps you have escaped that slide. Okay, no problem. Yes. So we have about 1.9 million patient which develops infectious invasive fungal infection. This is the comment of Benedict in 2019, and these their secondary metabolite may control so many type of because fungus produces a toxin, and everyone is familiar with mycotoxin. And mycotoxin was like aflatoxin. Aflatoxin is also type of mycotoxin, and these mycotoxin can be prevented, can be stopped by the help of the products which is obtained from the fungal infections or from the fungus. So we have a challenge, and we have to go for the diagnosis of the fungal infection. Next, please. Now I'm coming to most important part of my this lecture, that biofertilizer production, because we know that we are using chemical fertilizer, and that chemical fertilizer goes to the water after runaway, contaminate the water bodies, and causes eutrophication, and eutrophication result in the formation of enormous production, and adversely or negatively they produces algal bloom, and due to algal bloom the plant life diminishes, abnormal growth of insect develops, and production of enormous other amount of aphids or insect are being used. So chemical fertilizer like urea, in next slide you will see this, why cyanobacteria or why few fungus and few bacteria. We know that in atmosphere most abundant gas is the nitrogen, that is 78.01, and the least amount is for the carbon dioxide, that is 0.03%. Why a plant can absorb the carbon dioxide, not the nitrogen? Because they have the genes for converting carbon dioxide into uh, 3-phosphoglycyl aldehyde or 3-phosphoglycic acid, the first stable ribitol-like production. So they have genes to produce such type of synthesis. And why we are unable to absorb nitrogen? Why we are getting nitrogen in the form of nitrate, nitrite, or ammonia? Because the three bond which is present between nitrogen is not easily broken. So what happens? There is a genetically determined discrimination use of the carbon dioxide and nitrogen by the plant. So we are using a new technique that is not new technique, that is nitrogen fixing genetic engineering. Most of the cereal pulses, oilseed fruits, vegetable oil is the only answer to this junction. Next, please. What happens in nitrogen fixation? Uh, in nitrogen fixation, actually nitrogen is converted into of their oxidized form, rather either nitrate, nitrite, or ammonia. So the synthetic nitrogen fertilizer, if you are going to form like urea or any other, so you need 450 degrees Celsius temperature to break this three azo bond which is present in between two nitrogen. So we have to use enormous non-renewable fossil fuel, and reserve is spent to create such a high temperature. So what happens? The fossil fuel is going to escalate their prices day by day, and simultaneously 75% of the diverted to the plant removes vegetative growth, and only 25% is used in the grain filling. The grain may be millet or cereal, whatever it may be, because nowadays we are moving to the millets. So this magnified plant body consumes majority, that is 103 element from the soil, and represses nitrogen fixation by the microbes. So the soil fertility is also diminishing, or inversely, day by day. So what will be required? In next slide, we will see that. In next slide, please. Yes. In some cyanobacteria or in bacteria, this nitrogen and hydrogen combines to form ammonium, and they needs few genes like NID gene, NRD gene, and these genes are responsible for nitrate reductase, nitrite reductase, and then only nitrogen oxide can be converted into ammonium. And the different ADH gene, DH gene, GOGAT gene, and GS gene are responsible for conversion of these ammonium into glutamine, glutamate, alanine, and other amino acids. So on the basis of these amino acids, we can form the different type of fertilizer. So they are the genes which are present in different type of microorganism, like in cyanobacteria and few bacteria. So we have to use such type of genes for enhancing the production of, and that is possible for the genetic engineering which is needed and which is being used nowadays by our scientists. Next slide, please. Uh, some bioactive enzymes and marine mangrove fungi also produces some secondary metabolites. They can be also used in our coming generation in their genomics, proteomics, and combination of biochemistry and bioactivity. So the pharmaceutical industry will work against this novel bioactive compound production. And in next slide, yes, next please. Everyone, I think the students who are gathered here are their favorite food is mushroom, and you know that the life of mushroom is only 1 to 3 days, and very soon the tannin is the phenols which was present in their fruiting bodies, their basidiocarp, is converted into tannin. So we need some special precaution to keep them for a long time. The shelf life can be enhanced if they are being placed at 4 degrees Celsius. That's why you have seen that the shopkeeper are usually keeping the mushroom in their refrigerator, and by this you can enhance only the life by 4 to 7 days. So not only for the mushroom, rather almost every vegetable need such type of technique, how they can be kept for the long duration, how their respiratory rate, how their the storing metabolism should be reduced or minimized, so that, because in 2025 again the food price, the crop price and the mushroom price will again going to very high, because availability of fresh mushroom throughout the year, microbial and fresh products should be present. And if we are using the conventional package for marketing, we will use a very important, I mean to say that the microbial infection being reduced, because when we are using desi tomato, the thin layer of the pericarp was very important for rhizopus, and nowadays flavor saver have come into the market, and you can see the tomato can be placed for 1 month or more than that in normal room temperature. Next, please. So this is the need of our, and probiotics has already said much more, so many other the curd producing microbes, and the probiotics are the live microorganism. They are present in our intestine like Escherichia coli, and they have health benefit and consumed and applied to the whole body. And they may be yogurt or fermented food, and they are considered as dietary supplement or beauty products. So these are few about the present scenario and the future scenario of the microbes. But I am worried for the next slide. Please come. Recently, this Nipah virus in Kerala, and this was taken by me on 15th September 2023. Today is 23. And 11 more sample test negative is Nipah in Kerala. Think about this. In next, please. Nipah outbreak number of cases rises in 6 in Kozhikode. And so many other countries are also suffering from this. Next, please. Why Nipah is very dangerous? Because from active cases, so many soap has been closed, school has been closed in Kerala. And in next slide, why it is very dangerous? Because Nipah has high mortality rate. COVID was only saying about 2 to 3% mortality, but Nipah says about 40 to 70% casualty rate. You just imagine. The society president, Dr. A.M. Desmukh, has worked tremendous in COVID-19. When that was infected, he has written so many letters to PMO office. So now again, this is the time to aware all the microbiologists, be ready and be prepared for the next strain, which is coming very soon to us. And in next slide, why it is very dangerous? Because it causes mild respiratory issue to fatal encephalitis. It is transmitted from animals like bat or pigs, and human to human transmission is also possible. So this is about Nipah virus. Next, please. The initial symptoms is fever, headache, muscle pain, vomiting, and sore throat. Severe respiratory problem in advanced stages, and encephalitis, seizure, and even people goes to the coma. When India was not ready, at that time we have vaccinated 95% of the peoples are now vaccinated. But to see whether this Nipah virus will give immunity to that or not. Next, please. What is the treatment of the Nipah virus? No specific medication, no vaccine available for the Nipah, and primary treatment is only intensive support care. We can provide ventilator, we can provide oxygen. So think about this. Next, please. Nipah virus outbreak in Kerala, all about the deadly virus symptoms and treatment. Such types of news are coming in Times of India, and all the news channels are alarming. So, my friends, this is the time to see and be precautious for this type of viruses should not spread from Kerala, from that specific district to any other district, so that we can save ourselves. So, your wonderful virus today, our uh, Singh sir has given a very important comment in the group. He, I have liked this, and in Jharkhand group of the he has given a very good, important, because in microscope microbes are how is spreading and how they are creating. This is a very challenging thing. So next, please. Our planet is such a beautiful, because Jai Johar is also called in Chhattisgarh as well as in Jharkhand, because I was also done my honors from Jamshedpur Cooperative College, uh, and I am shifted in Madhya Pradesh after selecting in MPPSC. So this type of things are also available even in our Chhattisgarh. So our smiles should be like that important novel lady, and this is all about microbe from Sanatan Yug to this present 2023. And nothing is more to say which is more important and which is less important. Microbes are very important, and every student, whether he or she do not belongs to the microbiology, should be very aware, should be very precautious for the microbe. And the new strain is also very alarming. So I want to give a type of alarming to all of us. And this platform, especially Ajay Srivastava sir, and all the faculty members of the St. Xavier's College and all the students, thank you very much on this World International Microbial Day. And thank you very much to giving me this opportunity. If you have any question, then your questions are being welcome. Sandhirani has raised her hand. Yes. No.
Can I say something, sir?
Sure, ma'am. Sure. That will be my pleasure.
This side is Dr. Swati Soren, sir. Means I'm thankful to you. Means today, as being as the resource person, my 12th number it is. So I'm really thankful to you for your enlightening lecture, sir. So I'm also looking forward, because I just discovered my alumni, because I'm the alumni coordinator in my college. That's why I'm really happy to see you.
Thank you, ma'am.
Thank you so much, sir. Thank you so much. It was very enlightening.
Thank you.
you. Thank you. Thank you.
So even I'm very happy to see you here, because I too belong to Chhattisgarh state, and being in the same state, I live in Raipur district, and we are meeting here in the online platform. So thanks to the St. Xavier's College and Microbiology Society again for giving me the opportunity to be the host for today's event, so that I could meet you and
Okay, so
everyone
then
else
whenever
here.
you come to Chhattisgarh, please, I am in Bhilai.
I am in Chhattisgarh.
Okay,
Yeah, okay,
I
I
am
am
in
in Raipur, right?
Okay,
Okay,
neighbor
surely I'll
city.
contact you, sir.
Okay, okay. Thank
Okay,
you.
okay. It was
Nice
nice
to
meeting
talk.
you. Thank you, sir. So moving towards our last chairperson of the day, Dr. Shama Afroz Beg. She is an assistant professor and head of the department at Swami Sri Swarupanand Saraswati Mahavidyalaya Bhilai, and she has also been the head of microbiology department in Government Digvijay Autonomous PG College Rajnandgaon, Chhattisgarh, and assistant professor in Government VYTPG College, Durg. So she was also a JRF, SRF, and RA in Pandit Ravi Shankar Shukla University, Raipur, Chhattisgarh. She has been honored the 12th MP Scientist Young Award in the year 1997, Award of Excellence in the year 2011, and Best Designer Award Nav Bharata 2009, and qualified KJRF. She has a lifetime membership in various professional bodies, including MBSI, Advances in Life Sciences, Trends in Biosciences, and many more. She has a membership in around 20 committees, and she has attended more than 45 conferences and 33 webinars at national and international levels, and presented paper there. She has published 22 research papers and 3 patents, which includes 2 national and 1 German patent. We are very pleased to have you, ma'am, amongst us.
Good afternoon,
Welcome,
Sonal.
ma'am. Good afternoon.
Please share my
Welcome.
slides, and good afternoon
Yes.
to all the dignitaries present in the webinar. And thanks to the organizers, President Dr. A.M. Deshmukh sir, MBSI, Department of Botany, St. Xavier's College, Student Unit Rachi, and the principal of the college. They have provided me the opportunity to speak in the present topic, which itself is very interesting. That let's explore the microscopic marvels that shape our world. Please share my slides.
Rajeshwari, could you please share ma'am's slide?
Till then, I will be saying that the topic is very good because microorganisms are at the top, right from the producers to decomposers. They are ruling the world, as we have seen during the lockdown. In the world surrounded by visible, beautiful, great mountains, rivers, oceans, beaches, and landscapes, which we see, the microscopic world is easily overlooked. But when we explore them, the incredible beauty and complexity of these microorganisms is marveled, and they hold secrets that has shaped our world. So from prokaryotes to eukaryotes, and from eukaryotes to higher microorganisms, these little tiny microorganisms, I always say to my students that these tiny microorganisms are wonderful and they have made this world. Actually, they are the rulers of the world. We are just using them for us, and we have yet to use, we have yet to explore them. Because how can we explore? Now the time has come that we have to explore them to shape the world, future world. The application of science and technology to the living organisms, as well as parts, products, and models, which many of the speakers have told you about their uses, as well as to alter the living or non-living materials for the production of knowledge, goods, and services is yet to be done. And from probiotics to gerobiotics, Halami sir has said very nice about the gut microbiology, and this is the stage where my previous speaker, Dr. A.K. Srivastava sir, has said that nowadays Nipah virus is there. So this virus, actually this is ruling the world, which is not is on the border of the living and the non-living one. So we have to reveal them. Not even more than 95% of the microorganisms have been known by us. We have just studied about the 5% of the microorganisms. The 95% of microorganisms are yet to be researched, yet to be known, and they can be used for our benefit. We can use them for our benefit. Is it possible to share my slide? I want to show just one picture that how much microorganisms can be, how beautiful these microorganisms are. Rest I can say without that slides also, if you can.
Yes, ma'am. Rajeshwari, kindly turn on enable editing, or just put up the slides in presentation mode and change the slide accordingly as ma'am asks.
So what are the microscopic marvels? The microscopic realm isn't confined to the natural world, but it plays a pivotal role in various scientific disciplines and industries, as being said. In medicine, understanding the behavior of cells at a microscopic level leads to breakthroughs in disease, their treatment, and the drug development, mostly the vaccines. For all, we are using the microorganisms, which is we are exploring them and we are shaping our world, which should be useful for us. The microorganisms are tamed to make human beings lead their life in a very sustainable manner. And to use microorganisms, nanotechnology is the latest technology to harness the power to the small to create innovative materials and devices. We can transform the industries from electronics to energy with the help of these microorganisms. We can study the ancient artifacts at a microscopic scale even, and they also unravels the historical mysteries, shedding light in our own past. 3 or 4 days before, I was just reading in newspaper that scientists have used introduced the genes, some of the genes in the bacteria to photosynthesize a special type of protein which can be used for treatment of various type of diseases and which is environment safe. It is making the environment safe. Now, how can we use these microorganisms? These microorganisms can be used with the help of biotechnology, and 3 types of biotechnology are being classified, and these are the green biotechnology, the white biotechnology, and the red biotechnology. What is the green biotechnology? The use of genetically altered plants or animals to produce more environmental friendly farming solutions as an alternative to traditional agriculture, horticulture, and animal breeding process is the green biotechnology, where we can use the microorganisms, as Srivastava sir has said, that use of microorganisms for fixing of the nitrogen instead of using the urea. White biotechnology encompasses the application of biotechnology based tools to traditional industrial processes to make intermediate and end products more efficiently with less environmental impacts by making use of renewable resources. The factory products which are harmful for us and which are being polluting our water can be made safe by using by the use of these microorganisms, and that can be transferred into a new renewable products. The red biotechnology refers to the use of organisms in genetic engineering processes in order to design and manufacture pharmaceutical products like antibiotics and the vaccines. So these 3 biotechnology, the green biotechnology, white biotechnology, and red biotechnology, are impossible without the help of the microorganisms, and these will lead to the sustainable development. And if you can show the slide, I would have shown the images of beautiful microorganisms. If not, I will be and the breathtaking intricates that shape our world. So let us journey forth with curiosity as our guide, ready to be amazed by the hidden wonders that await beneath the lens. And as the theme of the International Microbiologist Day 2023 is from health to food to the environment, I have given the solution in the form of green biotechnology, white biotechnology, and the red biotechnology, for which we can use green biotechnology for the food, white biotechnology for the environment, and red biotechnology for the health. So, thank you very much for providing me this opportunity. As we are having short of time, I will conclude my session by thanking to the organizing secretary, Ms. Preeti Kumari, and all the other organizers who have made this very wonderful webinar, international webinar for exploring the microscopic marvels that will shape our world, and you, the youth, you, the youngsters, are being given the chances to explore this world. Thank you very much.
Thank you so much, ma'am. I really apologize from our end. Might be due to some technical issues, Rajeshwari is not able to share the slides properly.
Okay.
Thank you so much. I thank all the guests. All the resource persons are now welcome. So now I invite Shifa Ansari, a student, to continue with the program. And from my end, I would like to thank all the chief persons, co-patrons, and and all my fellow organizing team for giving me the opportunity to start today's event. So Shifa, please can you take up the session now?
Yes, ma'am. Thank you so much, ma'am, for your support. Girls and boys, ladies and gentlemen, warm good afternoon to one and all. This is Shifan Sari, student of BNN College and your host for the event. It's a great privilege for me to ask for your most awaited words through this remarkable journey. Now, I would like to call Anjali to please introduce Mr. Sudhanshu Shekhar Pandey, sir. I request Preeti to introduce Mr. Sudhanshu, sir.
Yes, thank you, Shifa. Just give me a minute. Okay. Good afternoon, ladies and gentlemen. Dr. Sudhanshu Shekhar Pandey. It is with a great pleasure to introduce Dr. Sudhanshu Shekhar Pandey, sir. Dr. Sudhanshu sir is a biomedical scientist. He has earned his PhD degree in biotechnology from National AIDS Research Institute, Pune and Savitribai Phule Pune University. His PhD work was focused on molecular evaluation of HIV. His PhD work was resulted into many research articles published in journals of international repute. After completing the PhD, he worked with Ternacia Biomedicals Limited and Tenor Performance Materials India Limited. Dr. Sudhanshu sir is having vast experience in development of molecular biology, ELISA and CLIA-based immunoassays, clinical chemistry, hematology, test kits, etc. Currently, Dr. Sudhanshu sir is working as a project leader and heading the R&D and production department of IVD division in Trivitron Healthcare Private Limited. The responsibility involves planning, designing, and execution of new R&D projects to supervise the manufacturing of clinical chemistry and hematology parameters. Dr. Sudhanshu sir has submitted more than 300 genomic sequences of viral and bacterial genomes into the Gen Bank and published many articles and research papers and is reviewer of many international journals. He has been awarded with prestigious Forgetry Fellowship from NIH, USA Scholarship from Spain, and many other awards. So please join us to welcome Dr. Sudhanshu sir, whose journey will inspire us in a great way. Thank you. I request Sudhanshu sir to please join us and enlighten our audience.
Is my slide visible to all of you?
Yes, sir, it is visible.
Yeah. So thank you, Preeti, for such a wonderful introduction. And I would like to thank all the organizers for organizing such a wonderful event. This is very informative and it is a pleasure to present my work here. Initially, I thought to present recent advances in vitro diagnostics, but since the event is on microorganism, so I thought to present the subject which is very close to my heart, that is microbiology only. So here I am going to present migration and spread of HIV-1 subtype A, that based on evidences of phylodynamic and historical support. So as we all know that HIV was HIV is one of the most studied and most dreaded disease we all know. So when scientists track back the introduction of HIV to human, actually they found it to early 1920s. So here is one very early paper which was published in 2000. Here they found that based on earliest sample available, which was from collected in 1959. So they backtracked the timing when it was introduced to human. So from where HIV came to humans. So it is based on multiple zoonotic transfer. HIV came into human. So here we can see that HIV-1 came into human from chimpanzees and HIV-2 from Sivian macaques. And after getting introduced into human, it went to multiple evolutionary processes, and ultimately it got classified based on molecular data. So HIV is basically classified into HIV-1 and HIV-2. HIV-1 is further classified into M and O, and recently identified group P. HIV group M is the largest and is responsible for the most of the infections worldwide. HIV-1 group M is further classified into A, B, C, D, F, G, H, J, K, and one more recent is L, but it is not yet established since very few infections are found for that. So again, evolutionary trends are there among these subtypes also, and further it is classified into further subtypes. So here I am going to focus only on subtype A, since we are talking about going to talk about evolution of subtype A only. So subtype A is classified into 6 subtypes. Now, few scientists are also suggesting that there is one more subtype, which is subtype A7. So maybe it will be also well established and will be accepted as a subtype. So subtype A, why subtype A I have chosen for my study. I did study on subtype C also, and then I went to subtype A because in India subtype A is also found. And while I was doing that research, I found that there are unique recombinants between subtype C and subtype A. So I got fascinated how subtype A came into India, and since then subtype C is largest predominating in India, 97%, but subtype A is not even not even 1%. So what could be the reason? So I got into the genomic details of these, and further I went away with the study. So the subtype A is mainly found in Africa, Europe, and Asia. Among at that time when I started, it was 5 only. So further study, it will be seen as 5, and subtype A6 should be there. That was also identified by our group initially. That in results I will show. So 5 subplates at that time. Now it is considered as sixth one. So the sixth one is earlier classified as subtype A1 only, and which is majorly from former USSR. So although reports for last 2 decades, HIV-1 subtype A represents a small proportion circulating HIV virus in India, till date very few sequences are available. So when I did this study, only 113 strains or 113 sequences were available, and none of the full genome was available. So I was fortunate to work at National AIDS Research Institute, where we got isolates. So I sequenced 6 different HIV subtype A isolates. So here we can see these are the isolates which we sequenced, and here it is clustering with 100% bootstrap. 100% bootstrap suggests that it is monophyletic lineage. That means one HIV-1 subtype A strain came into India, or multiple strain may came into India, but it was established, that population was established because of one single introduction only. So this was based on a full genome. Then I found more envelope, gag, and Paul genes. So I wanted to check with that also. Here also we can see this 100% for when we tested with gag gene, we found 100% bootstrap value. Here also monophyletic lineage, and here we can see this because of this more similarity is with Kenyan sequences. So here we can see along with this Indian cluster on left side and right side, both sequences from Kenya are there. So it is suggestive of the hypothesis that it came into India from Kenya. Then we did the same study with envelope gene. Here also we found monophyletic lineage, and again it is surrounded by Kenyan sequences or East African sequences. Then we went for Paul with larger sequence size and larger number of sequences, but lower length of the genome sequence. Here also we found monophyletic lineage and closeness to Kenyan sequences. So what we identified so far was HIV1 subtype A in India belong to subtype A1. That was the first finding. HIV1 A1 epidemic in India show monophyletic lineage. That means introduction from a single strain. Third thing we find based on results, we hypothesize that HIV1 A1 most probably introduced into India from Kenya or East Africa. Because in East African sequences, if we will see that mixing kind of thing we can see. And to prove the hypothesis, more further study was required. That is called timing of most recent common ancestor, TMRCA, and phylogeography analysis. So this, whatever I have described so far, was published in 2016. Then this molecular clock analysis we performed. So for this, one Bayesian Markov chain Monte Carlo approach is there. Softwares are there for this kind of work, and we took out around 4,000 full genome sequences from Los Alamos database, which is sequence database for HIV, and from there we zeroed down to 233 sequences from 1998 to 2010, and we tried to incorporate sequences from each country available. Then for this molecular clock analysis, we performed the phylogeny and divergence time under relaxed molecular clock model, and software Beast was used for this thing. So here we can see that this is the phylogenetic tree which was there. Here, because we cannot read the sequence name, so I have given color. For different color denotes different geographical region. Here we can see that Indian cluster is again monophyletic with this method also, and it is clustering within subtype A1. So to reduce it and summarize it, here we can see that subtype A1, origin of subtype A we backtrace to 1950. And from here it got evolved, and from that major A group only, that A4 was evolved in 1967. From here in 1972, another group one another cluster we can see evolution of Central Africa sequences, that was in 1960s, and from here only we can see different clusters. So within subtype A1, that this A1 was here also, and A1 USSR was outclustered here. So at that time only we questioned how it can be outclustered if it is subtype A1. And in between subtype A1, how A3 can come. So this question was further taken up, and reclassification was done. So now it is reclassified as A1 from former USSR is reclassified as A6, and rest of the things are reclassified as A1. That was remained same as A1. So here we can see that introduction of subtype A into India was 1979. Here is another way to show that with the confidence, because when clustering is there, that clustering should be more than 80% to give the confidence. So here we can see that origin was giving 99% confidence. Central Africa sequences 80% confidence, and so on. It is going. Here for India, it is 100% confidence, and it is clustering along with East African cluster. Here we further enlarged it to show the same thing. So here we can see which subtype emerged at what time. So the root means origin. So origin was in 1950, and we can check say the limit is 1942 to 1957. So that may be the range. Subtype A1 in 1960, so 1955 to 1965. A2 evolved in 1972 and ranges from 1967 to 1977. A3 was newer than others. So A3 emerged out in 1973 to 1981, and mean we can take 1977. Subtype A4. So it was emerged out in 1967, and again range is there, and so on. And here we can see at the last that Indian subtype A. So introduction into India is in late 1970s. So how it can. So here we have identified with phylogenetic study and molecular clock analysis. To support this, we have some historical evidences that how and why it is spread at different time point. So to start with this, we all know that origin of HIV is Central Africa. So in 1895 to 1920, there was one railway project was ongoing to connect Central Africa to East Africa, majorly in Kenya and Uganda, where one big lake was there. This was under British colonies, so the Britishers took laborers from different parts of the world to Africa to get the work done. So they took people from India also, or Indian subcontinent also. So after completion of the that population settled down in that area only, in East African area. Alongside that, big lake was there. So that was the one of the best place to live, because in African conditions, that was almost considered as grave. During that rail line project also, millions of people died for that only. So after the independence, so they got settled in that area, and people from migrated people from different part of the world, they got large chunk of economy. They did good work, and they get settled very nicely there. And they continued to stay there till 1960s, at the time of African revolution and independence. So in 1960s and 1970s, this socio-political instability was there in Africa, and people migrated from India, or people migrated from Indian continent or different part of British colonies, were having British passports. So they had the chance to move to the different countries. So some came back to India, some they spread from East Africa to different part of the world, that we can see in next slide, how at the same time HIV subtype A got spread. And the latest, we saw the breakdown of USSR. That is why that USSR cluster was very close, and that diversity is also not very high. So here we have done this study with envelope gene also and Paul gene also. And here we can see that with envelope gene, we have found it is 1950 origin at Central Africa. From Central Africa, it went to different part of the world. So in 1966 to Europe, in 1972 to different part of Africa, and in 1966 only from Central Africa to East Africa, and from East Africa to India in 1979. Here in Australia, we can see it migrated from in 1982 from Europe to Australia. And when we did the same study in Paul gene, we found slightly different result because of availability of sequences. So different sequences are available from different countries. And here also we found almost similar kind of data, only that dating was slightly different. So here we can see that origin, it is showing in 1936, and from Central Africa, it went to different part of the world, and in 1960. So in an envelope, we saw 1966. Here with with Paul, we have found 1960. So that is why I have told that in 1960s and 1970s, it got spread to entire world. If we consider Paul, it came into India in 1983. So finally, we concluded this study that if not only a subtype A, if we will consider other subtypes also, which is spread into world, slight variation we will get, but eruption will be in 1966, near about 1960s and 1970s only we will get. So one study we have done on subtype A, and people are studying on another subtype also. So this is the conclusion from my side. And any questions, if there, I'm happy to answer.
Sir, what is the actual host for HIV virus, by the way?
Actual?
Host. From where it actually emerged. Is it a human being only, or it was in some other vertebrates or any other animals?
The nearest we have found that that is chimpanzee, gorilla, and macaques only. So multiple zoonotic transfer we can say. And here in that earlier slide I can show.
Actually, your slides were not moving, so it became actually invariably difficult. But then you could explain properly, so we could make out.
Sure.
You are working in Nari or which institute?
No, right. I worked at Nari. Now I am working with Trivitron Healthcare Private Limited.
Okay.
So here we can see that different animals are there. So multiple things are there. This we can consider this center point. We can consider as origin. But till that point, since no known sequence is available, so we cannot conclude. That is why the nearest which was there, that was only considered. So in case of HIV1, it is considered chimpanzee, and in HIV2, it was considered as simian macaques.
And they also have the similar symptoms like what we see in the human being, or they are more immune to the HIV?
Different. It was different, and that is not very much conclusive study. But still, they had symptoms, and but not exact similar symptoms.
And you get this often mutation like our COVID virus, with reference to the
mutation of COVID virus is not so much studied, and the evolutionary rate of COVID is initial stage it was slower. Since it is very new virus, it is very early to conclude on evolutionary rate and classification, because when HIV enters into certain population, we talk about Indian population. Immune system of Indian population is very much different from European population or Chinese population.
Exactly.
So even within person to person it varies. So evolution of virus from person to person also varies, and from ethnicity also it varies. And getting stabilized, it takes time. In HIV, so many groups and subtypes we are seeing. It is result of hundred of years. So for COVID also, it will take some time to classify based on phylogenetic analysis. Right now, it is few mutations we are considering, and we are naming those mutations only as of now. We are not classifying it as a group or subtype.
Exactly. Very interesting study. Thank you.
Thank you so much, sir. Now I would like to request Anjali to please introduce Dr. Kumar Rakesh Ranjan.
Thank you very much.
Now let's gladly introduce our next resource person, Dr. Kumar Rakesh Ranjan, Assistant Professor in Department of Chemistry in Amity University, Noida. Pursuing his love for science, he went to Ranchi University to do graduation and masters in chemistry honors before furthering his PhD in inorganic chemistry at Banaras Hindu University. He also has teaching experience in BHU as teaching and professor assistant and has been awarded more than 2 times for the best poster presentation at BHU and Tejpur University. Besides these, he have also contributed his fellowship in project at SMST IIT BHU and attended various conferences at the national level. Throughout his journey, he has publications in synthetic characterization and magnetic properties of cyanide-based and new homo and hetero bimetallic assemblies and have co-authored essential oil extraction method and application published in the Journal of Fluorescence. So now to dive into the ocean of knowledge he have, please let's welcome Dr. Kumar Rakesh Ranjan sir.
Thank you, Anjali. Thank you so much for the nice and brief introduction of mine. And good afternoon to all of the dignitaries and the attendees of this international webinar. And it always gives pleasure to when we will get the opportunity to connect with our alma mater. And first of all, I want to introduce myself as I have did my BS intermediate ISc and the BSc from the St. Xavier College. And now currently, as Anjali told, that I am Assistant Professor of Chemistry at Amity University, Noida, Uttar Pradesh. Okay, so with the permission of you all, I want to share my slides here. Okay. Just a second. Okay, is it visible to all of you?
Yes, sir.
Okay. As Anjali introduced me, I am Dr. Kumar Rakesh Ranjan, and it's an international webinar on the day of the microorganism, organized by the St. Xavier College, Ranchi. So my topic of presentation is like recent trends in life sciences, a nanotechnology approach. We have already heard about 2 or 3 speakers who have just introduced the word about the nanotechnology. And currently, I am working in the field of the nanotechnology as the development of some biosensors. Okay, so let introduce me, the nanotechnology field, with all the attendees here of this international webinar. So why nanotechnology? Okay. So the answer is that in nanotechnology, when we move to the nanotechnology from the bulk one, okay, so it is just a manipulating of the atoms in a way that the properties of that particular atom will change drastically. Okay, so it is the manipulating atoms and the molecules at the nanoscales. And what happens after that? If we look into that, it says that the the nanomaterials or the nanoatoms or the nano configuration has the different properties and new effects compared to the same material which was earlier into the larger size. This is because of the high surface to volume ratio of the nanoparticles. So there are the different types of the arrangement of the nanomaterials here. As you can see into the image here, these are the zero-dimensional fullerene or Seonian, or it is a one-dimensional as a nanotubes or nanohorns. It is a quantum dots as and nanoribbon as a 2D material. Okay, these are all the carbon materials are this. Okay, so pillared graphene and the nanodiamond. These are the 3D materials. Okay, so the basic properties, or you can say that the enhancement in the properties of the nanomaterials is because of the high surface to volume ratio as well as the porous nature of these materials. Okay. So various fields of the application, as we are talking about the life sciences, so I have to put my focus on the application of the nanotechnology in the field of the life science. Okay, so these are the various fields where nano can work. Okay, whether it is the metrology, whether it is the sensor, whether it is electronics, whether it is the materials, and nano biotechnology. Okay, where it can mimic the sum of the compounds. Okay, like the sum of the hormones, it can be used in the diagnostic, tissue engineering. Okay, implantable devices, drug delivery, drug discovery, and myself, I, along with some of the research collaborators, I am working in the field of the sensors, which can work at a environment and medical. Okay, as in the form of the biosensor. So here you can see one of the thing what I want to mention here very specifically, that the nano in food packaging, how it can work. Like if I have or scientists have developed a food packaging material. So what we are observing that we have to mention the date of the manufacturing and the date of the expiry. Okay, now look into the way. Okay, what will happen? If the date of the expiry will be represented just by looking into the you can say the graphics of that package. Like if some like microorganisms will develop into that particular food. Okay, so there must be the release of some gases, and if that gas will interact with that you can say the packaging material, and that packaging material, because of the interaction of the gases, if it will change in the color. Okay, the packaging material, if it changes into its color from someone to the another, like from red to green or something else, okay, or from green to red. Okay, by simply just looking into the packet, you can say that, okay, no, that packed food is not at all good for intake. Okay, so it is the edible and biodegradable packaging, active and intelligent antimicrobial packaging. Okay, intelligent nano coating, that's what I am explaining here. Intelligent nano sensors, okay, and the smart nano composites. So nanotechnology in the food industry has tremendous scope, and the scientists are really working in that field. So now let me introduce you all with the trajectory of the nanotechnology advancement over the years from 1970s to 1990. It has been the nanoparticles so scientists were moved from the bulk to the nano, and they found that, okay, because of the increase in the surface area, okay, nanomaterials are showing tremendous characteristics. Okay, and from 2000 to 2005, you can see that it applied research. Okay, cell nanoparticle interaction, targeted delivery systems, and like that. From 2005 to 2010, it's the early adaptation. Okay, adapters in diagnosis of the disease. Okay, and the imaging, like I can say the CT scan and all. Okay, MRI and all. So now from here to 2010 to 2020, now there is the advancement. Okay, as the researchers more and more researchers are working in the field, more and more funding has been released by the government. Okay, so there is the rapid advancement in the field of the application of the nanomaterial into the biological system. Okay, and now we all have to work for, after 2020, we all have to work for the mass marketing. Okay, that is the anti-aging medicine, cancer treatment, infertility treatment, regenerative medicines, and like that. So one of the application I want to put your focus here, nanopore technology. So what is a biological pores? So it is a biological pores. You can see into the image that biological pores and the pore forming peptides, it can generate a pathway. So here you can see that there is a pathway for the flux of ions. So through the lipid bilayer, the ions will move through that pore forming the pore forming proteins, and sometimes that pore formation can be done by some toxic substances also. Okay, because of this, some foreign particles can get the chance to enter into the cell, and it will destroy the cell, and it starts damaging the cell also. Okay, so if it is the nanopore technology, so after the biological pore, we are going to look into the solid state pores. Now we are ourselves preparing some pores into the cell to damage the particular cell if that cell is the cancerous one. Okay, so the passage of individual molecules through nano-sized pores in membrane is central to many processes in biology. Okay, previously experiments have been restricted to naturally occurring nanopores. We are looking into the nanopore. Okay, we are just looking into the what are the naturally occurring nanopores. Through that, we are transferring some of the chemicals from the outside of the cell to the inside of the cells. But because of the advancement in this field, now we are allowing artificial solid nanopores. Okay, just you can see the image here. This is the solid nanopore here. This is the solid nanopore here, and just a second. Okay, this is a solid nanopore here, and this is the protein nanopores. By the combination of that, we have made this channel, and now we can transfer any of the required chemicals into the cell through this pore. Okay. So nanomedicines, this is the medical application of the nanotechnology. It helps in the detection, treatment, and prevention of the biological disorder at the molecular level. Now we are not going to look into whether we are at the stage 1 and stage 2 or stage 3, that for the detection of any cancerous thing, we have to wait till stage 2. No, we can do at the single cell level only. Okay, as soon as the problem started, okay, in the body or any of the cell getting damaged, starting damage of any of the cell, okay, so because with the help of the nanotechnology or the nanomedicine, we are going to treat that particular cell. Okay, so some of the drugs are very toxic and they can cause the harsh effect or the side effects. Okay, when like if we are taking some of the medicine, okay, like 500 milligram or 650 milligram we are taking, so how much effective amount of that our body is consuming? Okay, like if you have taken the medicine, so that get some of the amount get dissolved into the mouth only, okay, through the saliva only, and rest amount can go or reaches up to a certain particular point. Okay, but when we use a nanomedicine, so it can say that if they decomposes during the delivery, okay, they put the harsh effect or side effects. But in case of the nanomedicine, the drug is delivered to the site it is required, thereby reducing the side effect. Okay, so rather than taking the 500 mg, our work can be done by the 20 mg only. Okay, because effectively the 20 mg is only working, and rest all are getting you can say that reacted with into the mouth or lots of the body parts, small intestine and everything. Okay, so here you can see that it is these are the monomers one. Okay, these are the monomers here. Here is the cell. Okay, so application of medicine in the diagnostic and the therapeutic, like imaging, quantum dots, microscopic sampling, and in therapeutic it is delivering medicine to exact location, targeted drug delivery. It says about the targeted one. Okay, killing of the bacteria and virus at the cancer cell. Okay, repair of the damaged tissue, oxygen transport, skin and dental care, and brain enhancement. So very advanced techniques are these, where only some of the works or very few works are going on currently at this stage. The microscopic machines, okay, we are putting the microscopic machines into the blood. Okay, now that machines will detect any abnormality, okay, that will through the bloodstream. Okay, and it will provide you the signal. Okay? Like through the blood is different compounds. Okay, so a single inhaled nanorobots, okay, so reaches deeply inspired into the lung, enters in alveolar duct and attaches to the tissues surfaces. Okay, so when we use the nanotechnology for the drug delivery, so first what we are going to do, we are going to encapsulate the drug. So one major class of the drug delivery system is materials that encapsulated drugs to protect them during the transit into the body. So we are polylactide as encapsulating material. So now we are doing the functional drug carrier. We are putting some of the material into that. Okay, so nanostructures can be controlled to link with a drug, a targeting molecule, and an imaging agent. Then attract specific cells and release their payload. Okay, they will find out, okay, at this point I have to release my payloads. Okay, So they do so. What are the materials we are taking as a transport system? You can say. So just look into the image. This is the mesoporous silica nanoparticles in which we have loaded the drug, and that drug has been encapsulated, okay, through the stable encapsulating substances that is called as a surface locking. And when it reaches up to the targeting ligand, it delivers the medicine there or drug there. So another the protein corona shield for targeting. So this is the encapsulation. Now, therapeutic application, nanotechnology also theoretically allows the mimicking of the natural biological process. Example, repair of damaged tissue. Actually, the tissue is not getting repaired, but nanomaterials itself work as that, okay, that portion has been repaired by the nanomaterials. Okay. So, and these are the biocompatible materials. So using nanotech, I will provide you a list of that one. Okay, so what are the biocompatible materials into my next slides? So using nanotech to build scaffolds of the artificial molecules that bone cells soften, okay, that bone cells soften, adhere to, and grow bones on the broken bones would heal much faster. Okay, the rate of healing get increased. So the transport of oxygen within the body by creating an artificial red blood cells. Okay, so basically we have to rely on that. Okay, first we have to increase the hemoglobin percentage or amount of hemoglobin into the any of the patient, then the oxygen level will slowly will get increased. But no, we are putting the artificial RBC into that, which will help into the transport of the oxygen. So to cure skin disease, that is to remove the right amount of the dead skin. Okay, so removal of excess of oils and missing oils, etc. Now, so just take the exam, just graphically, I want to represent that how the cancer cells will be get detected into the body through the quantum dots. Okay, it is the quantum dot, one of the nanomaterials here, through the UV light. So first look into that. These are the quantum dots, and the cancerous cells has been represented by the green one. These are the healthy cells. Okay, these are the healthy cells. Now this has become cancerous. We have the quantum dot beads. So we'll give time to the cancerous cell to interact with the quantum dot. So once the interaction has been done and we irradiated that interaction with the UV light, so look into the example. Okay, so the UV light, cancerous cell, all the quantum dots. So this is when quantum dots are working as a sensor, it get adhered to the cancerous cell only, not with any of the normal cell. So okay, so we got that the location where the localized cancerous cells are. So now it will be easier to burn them or to destroy them. Okay, just through a small video, I want to present something, and I think this is the best way to understand anything, like through the videos or through the graphical cartoons. Okay, just allow me to play this one.
Institute for Nanobiotechnology. In the Jordan Green Lab at the Johns Hopkins School of Medicine, researchers are using nanoparticles as a mini delivery vehicle. This vehicle will allow more of the therapy, whether it be a drug, gene, or an antibody, to reach the site of the tumor more efficiently. In the Jonathan Schneck Lab, also at the Johns Hopkins School of Medicine, researchers are using nanoparticles to activate immune cells. This treatment would be similar to how a vaccine would work, where immune cells would only kill cancer cells. This would eliminate many of the off-target side effects with current treatments and provide a long-lasting immunity. In the Margarita Herrera Lab at the Whiting School of Engineering, researchers are studying the kinetics of how polymer brushes assemble into these nanoparticles, so they may try to better control the size and increase the efficiency.
Okay, so what I want to represent it through this way, here you can see that these are the drugs which are hydrophobic in nature. Okay, and these are the materials which has two parts. It is the hydrophobic part here and the hydrophilic part here. So you can see that this is the higher water here. Okay, so the hydrophilic, it is a sometimes missiles type of thing. So the drug has been loaded inside this area. Okay, the very localized loading of the drugs, and it has been kept you can say bounded with the hydrophobic part. Okay, now you can we can transport it through any of the targeted places. Okay, because it will never get released into that till the environment is aqueous. Okay, so this by this way we can use a minimum amount of drug to do a targeted drug delivery and targeted therapy. So as I told you that these are the biological nanoscales with respective natural as well as man-made things in that range. So these are the biomedical application of the biocompatible nanoparticle, iron oxide, the metal in the female long-term concentration, gold, okay, nanocells, quantum dots, cyanide dyes, Okay, so there is a complete list of the biocompatible nanomaterials, okay, and its biological applications. So, and because of its property, they are working like that, like nano iron oxide. We all know that the iron has the magnetic property. So it has the magnetic field mediated targeted drug delivery. Okay, so we can guide that loaded, can say that the encapsulated or loaded drug to a particular place through the magnetic field, and we can release the magnetic field, and at the same time, at the same place, the drug will be released into the body. Okay. So I want to conclude that one because since it's a long topic, okay, and we have crunch of time here. So I want to just conclude here. So nanotechnology has great scope in the field of the life sciences, and smart enabled material can show a remarkable application in the field of the medical field, whether it is a device or a medicine. Challenges, okay, the big challenges that scientific community has to work in this. So synthesis of the nanomaterials, handling and disposal is a great challenge. Okay, like if we are synthesizing some of the nanomaterials, because it's very difficult if we are trying to, like, if we are looking for that water pollution. Okay, so if that water is polluted with some insoluble material, then it's very easy to filter it out. But if it has been contaminated with dyes, then we have to take some molecules, like as I published one of my paper in fluorescence. Okay, so we have to design some of the molecule which can effectively work for particular dye or particular metal ion. Okay, so nanomaterials, because we know that its size is in the nano range, so it is very hard to filter it out if it get transferred into the ecosystem. Okay, mass scale production, it requires a large number of skilled person, and its effect into the body is still under study. Like we have taken the fluorine, we have loaded the drug has been delivered to a particular target. Okay, no, after that, what happens to the fluorine? Will it come out, or will it remain sustained into the body? What will be the can say the harmful effect of that deposition of that fluorine on some of the carbon nanotube into the body? Okay, that has to be studied. Thanks to all of you for your patience and listening me. If any question is there, I'm very happy to answer that one.
I guess nobody have doubt.
Okay.
Very good, sir.
Yeah. Thank you, Safra.
Now I request Anjali to please introduce Dr. Jaya Kumaran Nair.
With great pleasure, let's introduce our next resource person, Honorable Professor Dr. A. Jaykumaran Nair, Emeritus Professor of Department of Biotechnology in University of Kerala. Holding a diverse and impressive educational background, Sir have done PhD in biotechnology from the National Institute of Interdisciplinary Science and Technology, Thiruvananthapuram, and MSc in Botany from Mahatma Gandhi College under University of Kerala. Sir has been in the research field for the last 32 years and have undertaken and completed more than 8 projects in the field of science. Throughout his professional journey, Sir have held various roles in the academic field, from being the professor and head at Department of Biotechnology in University of Kerala to being a senior research fellow in Regional Research Laboratory in Kerala. He has also shared his expertise in the administrative field in various designations, from staff advisor lecturer in the Department of Biotechnology to the chairman and member of board of students in biotechnology. Despite immense contributions, Sir have also organized and presented various webinars and workshops, published textbooks, and supervised researchers. He has been honored with more than 7 awards, which includes the Award of the Youngest Scientist in the Department of Science and Technology by the Government of India, and 22 PhD awards, and many more. Currently, sir is working as the chairman in Board of Studies in Biotechnology and also as member in the faculty in Studies of Applied Science. His accomplishments makes him an invaluable resource for today's webinar. So please give a warm welcome to Professor Dr. A.J. Kumaran Nair, sir, whose journey and experiences will undoubtedly provide us immense knowledge.
Thank you. Thank you, Anjali, for a nice introduction. I am very happy to be here to participate in the International Microbiology Day organized by the St. Xavier's College. And I appreciate the organizers for arranging this lecture series in a very organized conditions, and it's very nice. And I am also at the outset thanks all the organizers for giving me this chance of giving a lecture on microbiology and biotechnology. So these slides are visible? Hello? Hello?
Yes, sir, we can see your slides.
Okay, thank you. So my talk, I think I will talk about the involvement or the importance of microbial world in the field of biotechnology. And I selected the topic as a perfect blend for sustainable development. So as we know, we take the resources of today's and that is for meeting the requirements of today's without compromising the needs of future. That means the resources has to be sustainable, because when we use the resources without sustainability, it will create a lot of environmental problem and related so many other problems like a reduction in the resources, and we have to make the things that is needed for the tomorrow's world. So that is the main thing. By working in biotechnology, we can understand. So taking the issues of climate change, then environmental protection, then the health and education, poverty challenges, all these are some of the issues that is present. There is the present situation is needed, and we have to eradicate all these by taking new technologies. And by taking the technologies, we have to see that it is not producing any bad effects on the environment as well as in the society. So we know that the technologies are available. Basically, the industries are available. Most of them are chemical based, and that results a lot of pollution in the environment, and it results in the degradation of environment with serious climatic changes as well as ecological disturbances, reduction in the biodiversity, and also extinction of a larger number of biological resources. But an answer to this, if you take the methods of sustainable biological alternatives, it will result in the green technologies. Particularly, biotechnology can be considered as a green technology which protects the environment and other related things very well. So there are environmental biotechnology which clean the environment, and also the clean technologies that develop out of biotechnology will be having less impact on the environment, and environmental pollution can be minimized. And with that biotechnology, or with this thing in our mind, we can make new products as well as processes which are needed for the development of the country and as well as the society. So we know that biotechnology, the major component, or about 80% of biotechnology involves microbiology or microbial world, like bacteria, fungus, etc. And if you consider the other branches like plant biotechnology or pharmaceutical biotechnology, nanobiotechnology, animal biotechnology, environmental biotechnology, all these areas cannot exist without the involvement of microbiology. That means about 80% of the biotechnology is the direct impact of microbiology, but the other things are there which also cannot exist, which cannot sustain with the involvement of microbiology. So that is what, in that case, about 90% of biotechnology is run by microbiology, or you can tell that the soul of biotechnology is microbial biotechnology or microbial technology. And there are some the products or the outcomes of the microbial world or the microbial biotechnology, which includes medicinal compounds, biofuels, microbial enzymes, other microbial compounds, agrochemicals and biopesticides, biopolymers, and for environmental applications, compounds for environmental applications, and food and dairy biotechnology. So all these are some of the areas where microbial technology can contribute. So we know that there are a series of microbial enzymes which are involved in industries, particularly food industry as well as other industries, which include direct microbial involvement or the microbial enzymes like amylase, lipases, proteases, etc. They are very much involved or which very much needed for the food biotechnology as well as dairy biotechnology. Then glucose isomerase, penicillin acylase, cholesterol oxidase, restriction enzymes, glucose oxidase, lactases, pectinases, fibrinolytic enzymes, all these alginate lyase, all these are very much important in the clinical aspects or in the medicinal fields. Then in addition to that, there are direct medicinal compounds from microbial world like antibiotics, then alkaloids, secondary metabolites from funguses and plants as well as from the bacteria, then therapeutic enzymes, hormones, vaccines, vitamins, amino acids, veterinary medicine. So the list is very long. In the food and dairy industries, we can see that food additives, food processing, flavoring compounds, vitamins. Then there are processed foods like processed meat, processed starch, lipids, and other dairy products. Then various types of organic acids, glucose, fructose, and other mono and oligosaccharides, etc. Then cyclodextrins, itaconic acid for acrylic resins, polyhydroxybutyrate, lactic acid polymers, cellulose-based biopolymers, and cellophane, etc. All these are the products from microbes directly. Then we know that biofuels like bioethanol and biodiesel, that also is very important, and there also microbiology has a very important role. Biofuels like biofuel cells, that is for getting electricity, and that is also a new development in the microbial research, and it is in the infant condition, but there is chances of expanding into various aspects for getting electricity directly from biofuels. Then the environmental applications like waste management, detoxification processes, effluent treatment, bioremediations, etc., and also production of value-added compounds from these effluent as well as solid wastes like single-cell proteins and single-cell lipids, which are also having tremendous applications in the food industry as well as in the biofuels like bioethanol as well as biodiesel. Then there are agrochemicals like biopesticides, bioherbicides, genetically engineered organisms for agricultural purposes, and nematicides, which are used in the crop fields, means in the agriculture for the protection of crops. These are some of the examples from our laboratories from the Department of Biotechnology. We worked on certain production of certain compounds as well as certain enzymes like cyclodextrin gluconotransferase, means the fibrinolytic enzymes from Bacillus cereus, alginate lyasase from the Nocardia species, endogluconase from Aspergillus caldocystus, actomycin D from Streptomyces, alkaline metalloprotease from Halophilic Bacillus species, alkaline lipases from Aspergillus fumigatus, lacase from Orthography species, as well as Enterobacter, and also these are some of the enzymes as well as compounds. But there are so many other researches also in our laboratory. For example, there is the solid waste management of Kyer pith. Kyer pith is a recalcitrant material, and it cannot be disposed or it cannot be composted very easily. So we have taken a method in collaboration with our Kyer Research Board in Kerala and produced a nitrogen-rich compost from this coconut pith, and now they are marketing this material to the farmers for various purposes, for horticultural things, as well as for making the soil enriched with nitrogen, as well as to retain water. Similarly, we have produced cyclodextrins. We synthesized cyclodextrin, as well as we produced CGTAs, the enzyme that is responsible for the production of cyclodextrins. Then another important product that we developed is the fibrinolytic enzymes, which also is very important in the medical field. Then we have developed a method by which we can dispose the poultry waste and also produce certain value-added compounds or value-added material from this by using the enzyme keratinase, which is a protease which digests the hair as well as feathers of poultry as well as human. And it has got environmental application as well as it has got application in the food as well as in the cosmetics. For example, the hair remover in the food industry. For example, for the preparation of chocolate, they use certain amino acids, particularly cysteine, and hair is a good source of cysteine, and that can be digested with the keratinase, and the cysteine can be extracted and separated and purified. That can be used for various other purposes. And another important enzyme is alginate lyase, which is an enzyme which can digest the calcium alginate. Calcium alginate, it is an antidote, or it's a medicine that is used for a genetic disease like a cystic fibrosis. Cystic fibrosis is a genetical disease which result in the conjunction of or the blockage of the respiratory passage of our lungs by the formation of alginate. Because when there is a lung infection with a certain bacteria, and it can be eradicated, means it can be cured, the bacteria can be killed, but the things will be there, and genetically the person will have some problem, that is the mucus of the lungs will secrete calcium ions, and the bacteria will produce the alginic acid or sodium alginate, and it reacts with the calcium and forms the calcium alginate. And we know the calcium alginate is like a fibrous material, and along with the DNA fiber that has been released from the pathogenic organism, they can block the respiratory tract as well as fine capillaries, means small air passages of the lungs, and thus the patient will be resulting in the death. So such conditions can be cured, can be controlled by using alginate lyase, which digest the sodium as well as calcium alginate and give some relief for the patient. Then another one is the betulinic acid that has been isolated from Tectona grandis, that is teak, but that is a medicine used for obesity, and that also is one of our product that has been produced in our laboratory. Then, of course, the cellulase and lipase enzymes. The cellulase, as you know, it has got environmental means we can use converting the cellulose into glucose, as well as a material, means as a process for making process for the disposal of solid waste, which is 90% of this solid waste is cellulose, and in that way also we can dispose the material, or we can convert the cellulose rich waste into other value added materials. And lipase, that we know, it has got various aspects in the food technology as well as in the industry, particularly for biodiesels as well as for transesterification, for converting one type of lipid into another type of lipids. And these lipases are very important in these industries. And as we know, I will tell something about the cyclodextrins. The cyclodextrins, they are certain oligosaccharides, the circular oligosaccharides having 6 to 10 glucose units, and it has got an means interior part which is hydrophobic, and the surface part is hydrophilic, so that these compounds can take certain hydrophobic materials inside, and thus the water insoluble materials can be dissolved in water using the cyclodextrin, because the cyclodextrin is a guest material, and the cyclodextrin is a host one, and the guest, which is what is called hydrophobic, it can be easily enter into that cavity, and it will be there. The surface of the cyclodextrin being hydrophilic, that material will be dissolving in water, and it has got a tremendous application in the pharmaceutical industry as well as food industry, and also in cosmetics. In pharmaceutical industry, we know there are certain drug materials which needs to be slow releasing ability, that can be, for that purpose cyclodextrins can be used, and also in the food industry for encapsulating flavors and fragrances, which is needed in the food materials, that also can be easily engulfed in the cyclodextrin, and the food material have that flavor as well as its fragrance for a very long time without any problem. And the same thing is there in the cosmetic industries also. So that is the importance of cyclodextrin, and there are some bacterial population which can synthesize the CGTA, cyclodextrin gluconotransferase, which convert glucose or starch into these cyclodextrins. There are 3 types of cyclodextrins based on the number of glucose that is present. There is alpha, beta, and gamma series. Then similarly, there is the fibrinolytic enzymes, which are produced by certain bacteria, and we have isolated a Bacillus cereus strain, and it has the ability to digest the fibrin that is responsible for the formation of clot in the capillaries of our body, particularly in heart as well as brain, which can lead to the serious clinical problems. And there we can use these fibrinolytic enzymes, which directly goes and dissolve this clot and release the give relief to the patient, and thus it can be developed into a clinical compound. Already there are these fibrinolytic enzymes, but it has got certain disadvantages. It randomly digests the other proteins also, so it has got side effects. So we have to develop a protein, a fibrinolytic protein, which selectively goes and bind to the fibrin and digests the fibrin and the clot, so that without affecting any other proteins of the blood. So that is the importance of these type of enzymes, and we have isolated enzyme from this Bacillus cereus, which is having this particular property. In addition to these microbial things, we have some other work also that is based on nanobiotechnology. We have developed a nano-based active food packaging film, which is used for covering or giving a coating for the food materials which are easily damaged, like fruits as well as meat. If you give a coating with this particular nano-based active food packaging material or food packaging film, this fruit as well as meat can be stored for a very long time without any bad effects on its structure as well as in its quality. So the figure shows the packaged fruit as well as the meat with the other. For comparing, we have put this material without this packaging film. Similarly, we have developed a nano-based carotene incorporated ice cream as a standard food material to show that this material, in this way, we can make food materials enriched with a different type of vitamins. And this ice cream we developed, and that also has been checked its flavor as well as the taste with selected persons, and also that has been passed. And in addition to this, we have developed a film of nano-based film incorporating certain antibiotics, so that can be used for means covering the wounds, so that it can be cured very rapidly or without any other problem. So that also is one of the major work that is going on in our laboratory. So with this, I'm coming to the conclusion. The future aspects of industrial microbiologists as well as biotechnologists, the exploration of natural sources for new potent microorganisms with new properties as well as new abilities and products, design and standardization of new bioprocess technology conditions for maximum product output. So that is one of the major activity that is going on in various laboratories. In our laboratory also, the product enhancement, product output, to maximize the product output. So then only the product can be marketed very easily without any financial problems. Similarly, genetic engineering of microbes with the novel genes for the production of high-cost products in fermenters. So that is also another important area where the biotechnology, means microbial-based biotechnology, can go further. So I am coming to the end. With this, I express my gratitude to the organizers for giving me this particular chance, for giving me to deliver this lecture. I express my gratitude to all the participants as well as the organizers, and our Halami, Dr. Halami is there. I am personally indebted to him for his help giving to me during whenever I approach him. He is very generous to help me in various ways. Thank you very much for all of you.
Thank you so much, sir. I request Anjali to please introduce Dr. Sunita Borkar, ma'am.
Now, the next resource person is Dr. Sunita Borkar, the Head of Department of Microbiology and MSc Microbiology Coordinator at PES RSN College of Arts and Science, Pamaguri, Ponda, Goa. She has qualified BSc and MSc with first class from the Bombay University. Her PhD is on the Alka files from Department of Microbiology of Goa University. Following her passion for microbiology, currently she is the head of Department of Microbiology and working as the IQAC and MSc Microbiology Coordinator in the PSS RSN College. Ma'am have worked on many research works focused on alka files, halophiles, phosphate from the marine and coastal ecosystem. With her vast experience, she has also guided many research works which has been awarded as well. Holding diverse knowledge and experience in the field of microbiology, she has been invited a lots of times as a resource person and presented on the bioprospects of coastal eubacteria, characterization and formulation of trichoderma, and also to speak on her journey in the field of microbiology. Utilizing her knowledge and pursuing her interest in microbiology, she has participated and presented posters and gave oral presentation on the alka files and many others. She has authored more than 15 publications at the national and international level journals. Ma'am has been honored with the award of Mother Theodosia Prize, Best Poster Award, and MSI Best Teacher. So to gain the knowledge and inspiration from her journey, please welcome Dr. Sunita Borkar, ma'am.
We would be honored to invite Dr. Sunita ma'am to pay tribute to our distinguished audience with heartfelt words. We welcome you, ma'am.
Okay. A very good afternoon to one and all. At the outset, I thank the organizers for giving me this opportunity to be a resource person at this international webinar on advances in life sciences, recent trends in life sciences. Thank you, Anjali, for that very elucidative introduction. I would like to share my presentation now. Yes, can you see my screen? I've shared the screen.
Yes, ma'am, it is visible.
So what I'm going to present today is related to bioprospecting pigment-producing bacteria from mangrove ecosystems of Goa. Goa is the smallest state in India and is located on the west coast with a geographical area of 3,702 square kilometers. This state is known for its famous beaches, temples, and churches, including, of course, a lot of activities, fun activities that go around. It has a coastline stretching along the Arabian Sea, and it hosts a diversity of ecosystems. We have areas where land and water join to create an environment with a distinct structure, diversity, and flow of energy as coastal ecosystems of Goa, which vary right from mangroves to khazans to sand dunes, salt pans, estuaries, bays, and wetlands. Mangroves, these are an ecological group of halophilic shrubs and trees and are also called as wetland forests with very high pneumatophores above the ground as the breathing roots, and they have alternate fresh and saline water runoffs. These ecosystems are nutritionally rich due to the continuous shedding of the foliage and support abundant and diverse microorganisms. It's because of this reason that we have this ecosystem that can be used for the isolation, identification, characterization, and for the biotechnological applications of various metabolites of microorganisms, where this ecosystem also is known to have slight high salt concentration and high pH. Pigments, as you all know about, that we have no world without pigments, and they are chemical substances which can selectively absorb light of certain wavelengths while reflecting the others. And we have a number of types as synthetic and natural. And when we talk about microbiology and microorganisms, that these pigments that we think of isolating the organisms which can produce pigments and utilize them for their biotechnological applications. So natural colored substances which are produced by microorganisms are actually produced in order to protect themselves from ultraviolet radiations, and they are also known to act as antioxidants and protect the organisms from heat, extreme heat and cold, function as antimicrobial and anti-cancer agents, in addition to acquisition of nutrients like iron, nitrogen, and carbon. They have a number of beneficial properties, like they can be used as anti-cancer, agents, as anti-proliferative agents, as immunosuppressors, as antibiotic producers, and for biodegradation of different compounds. As we know about the Vibgyor, that we have the entire range of colors which are produced by these organisms, right from a violet, the violacin, to blue as indolin, to green as produced by Pseudomonas like pyocyanin, to yellow like by Sarcina, Streptococcus, Staphylococcus, and orange, of course, by a halobacterium, to red, as we see with respect to the production by Serratia. In our study in this particular work that I'm presenting to you, we thought of collecting samples from the mangrove ecosystems, because this ecosystem is really a very potential system for the isolation of microorganisms, and isolate and characterize pigment producing bacteria, optimize the conditions for pigment production, extract the purified pigment in order to study the biotechnological applications. With respect to isolation and characterization of this pigment producing bacteria, we have collected samples from different mangrove ecosystems, like from very common spaces like Durbat, Adkoi, Rasai, and from Panchavadi, and plated the sediment samples on nutrient agar, on sodium chloride tryptone yeast extract agar for isolation of halophiles, and polypeptone yeast extract glucose agar at pH 10 for the isolation of halophiles. Dear participants, these extreme environments are really very important ecosystems for the isolation of different types of microorganisms, as they themselves can produce different metabolites at extreme conditions of maybe temperature, pH, salt concentration, absence of oxygen like anaerobes. So I would advise the student participants, the young researchers, as to work on these extreme ecosystems, as you can really obtain very novel organisms. So during our study, we isolated organisms on nutrient agar, on antibiotics, sodium chloride tryptone yeast extract agar, and polypeptone yeast extract glucose agar, and we obtained a number of pigmented organisms, not only at neutral pH, but also at high pH and high salt concentration. So we have the organisms producing all different colors of pigments like pink, red, yellow, and they were really very fascinating isolates for us. And we cultured them in the flowers and saw that they could produce the pigments in nutrient broth. Looking at the colony characters of a single colony, we found the two organisms that we selected were PES-AB3 and PES-AB4, where they were found to be gram-negative rods. PES-AB3 was found to be gram-negative rods, and PES-AB4 as gram-negative cocci. So observing the single colony, PES-AB3 had very viscous mucoid layer around the colony, whereas PES-AB4, we found it to be having a circular margin. We cultured these organisms in different media, and through biochemical characterization, PES-AB3 could be identified as belonging to the genus Pseudomonas, and PES-AB4 belonging to the genus Serratia. Biochemical studies are now not the ultimatum to identify the organism, and hence our 16S ribosomal RNA identification showed that these two organisms, PES-AB3 was Pseudomonas chloroamphes orienticum, and Serratia was identified as Serratia nematodiphylla. Scanning electron micrographs showed Pseudomonas to be having gram-negative flat rod morphology, whereas Serratia were oval shaped cells. On optimization of conditions for pigment production, we found that Kingsby medium was the best for both the isolates that gave us very good pigment formation. During our screening, we used nutrient broth, lurea botany broth, polypeptone yeast extract, glucose broth, and Kingsby medium. Further, going into the addition, as we know about that the addition of glycerol can enhance the pigment production, but then to our surprise, both the cultures produced less pigment in the presence of glycerol, as a result of which we avoided glycerol in our medium, and the temperature of production was found to be best at room temperature of 25 degrees C in the presence of 1% salt concentration. High salt concentrations inhibited the pigment production by these two isolates. With respect to their time relation and the pigment production, it was noted that Pseudomonas species produced the pigment after 120 hours of incubation, whereas Serratia nematodiphylla could produce the pigment, maximum pigment production within 96 hours. With respect to the UV survival curve and to study the effect of ultraviolet, as you know about that ultraviolet pigments usually protect the organisms because of the ultraviolet radiations that they can produce the pigments, and it is a very protective pigment. The ultraviolet radiations resulted in the formation of two types of colonies, that is pigmented and pigmented. Pseudomonas was found to produce a water-soluble pigment, whereas Serratia produced a pigment that could be extracted in different types of solvents like acetone, ethanol, and for the extraction of that intracellular pigment from Serratia, we obtained the pigments in the different solvents and performed a UV visible spectroscopy, a UV scan of this pigments of the extracellular pigment produced by Pseudomonas gave a peak at about 404 nanometer, whereas the extraction of the pigment, intracellular pigment, by using different solvents resulted in 3 different peaks, where the best maximum the peak was at 523, 534 nanometer. These 2 different peaks were related to the production of a fluorescent pigment by Pseudomonas and prodigiosin pigment by Serratia. These pigments were further purified by carrying out the thin layer chromatography, and these purified pigments were again further scanned for their absorption, and the purified pigment produced by Serratia was found to be prodigiosin. As all of us know about that, prodigiosin is a red colored pigment that is produced by this organism, different species of Serratia. And a test that was done to identify this pigment by using hydrochloric acid to oxide agents, and the prodigiosin pigment produced by Serratia was found to give us good red colored candles. As you can see in this picture here, the candles were colored with the acetone extract of the pigment, with the ethanol extract of the pigment, with the methanolic extract of the pigment, and with the butanol extract. Methanolic extracts gave us the best color of the candles. We also tried dyeing the fabric as well as filter papers, and the pigment was very much absorbed by the filter paper as well as the fabric. As we know about that, most of these pigments are used in the textile industry, and most of them, of course, are chemically synthesized. But looking into these pigments which are produced by microorganisms should be our choice, as they are very safe to be used as compared to the chemical dyes, because different environments. Analysis, that is, in Goa, we have a number which the biotechnological applications. We have a book that is Bioprospects of Coastal Eubacteria, that is comprising the work of all different organisms which have been isolated from coastal ecosystems of Goa as 15 different chapters. So in your library, if it is possible, you can get this book from Amazon, as well as you can buy through your dealer, the book dealer. And this book is really very precious book for us, as we have many different, all the parts very. I thank the organizers for giving me this opportunity, and MBSI, Dr. Deshmukh, who's our president, and we are having national
of Gujarat University, Ahmedabad. He has 40 years of teaching and research experience in the field of microbiology and environmental biotechnology. 30 students have awarded PhD and 50 students have completed their postdoc, biohydrotechnology, etc. There are more than 350 international seminars on his name and contributed articles as chapters in several books. He has handled 19 research projects funded by various funding agencies. He is expert member in several national and international committees and a member of Executive Court and Academic Council of Gujarat University. He has been awarded the Hariom Ashram Award. He was PhD recognized guide for 4 universities. Under his guidance, the bioleaching process and acid mine remediation technology are developed in Gujarat Mineral Development Corporation. Presently, he has been awarded the Lifetime Devotion Award in the field of microbiology in the January 2019. He has filed 4 patents, 2 of them are already awarded and 2 are waiting for the final decision. With that, I invite Shailesh R. Dave sir to our esteemed gathering.
Hello. I am audible?
Yes, sir, we can hear you.
But
yes, sir.
Give me the sharing option so I can share my slides.
Give me a minute, sir.
I'm not able to share the slides.
Sir, have you sent us your PPT?
No, I have not sent.
Okay, sir. Just me check, sir. So there is no restriction in screen sharing, but I don't know why I'm able to do that. So your screen is visible to us.
Screen means what is visible?
Yes, sir.
It is my only mail is visible.
Yes, sir.
My slides are not coming. Somehow, I am audible. My slides are not there.
So you are audible, but your slides are not visible.
Okay, so I think I...
So send me to the microbiologist Jharkhand email ID. I will present it.
I don't know why I'm not able to say.
Don't know, sir.
Is it coming?
No, sir. Only your mail ID is visible.
No.
Yes, sir. Now it is visible.
Is it come?
Yes, sir. Yes, sir.
Okay. Okay. So first of all, I would like to thank the the JVS College Ranchi for inviting us for this seminar. I am very much thankful for giving me the opportunity. Now, you will find something surprise by looking to the title of the talk I am going to give. I have selected it M3. Now, what do you mean by the M3? Now, the second slide is visible to you. Hello?
Yes, sir.
I am audible?
Yes, sir. Yes, sir. You're audible.
So I'm going to talk about the microscopic marvel of metals. Now, if you look at the microorganisms, then definitely we have to look to the history, because the first microorganism developed on planet Earth are nothing but microorganisms. No, and we are using microorganisms without knowing about the microorganism long back. The second thing what I want to talk during this talk is the microscopic marvel in metal leaching. The second topic which I am covering is role of microorganisms in metal removal from the aquatic waste, and how the microorganisms are playing significant role in metal detoxification and metal transformation. And what are the future aspects of this type of the thing? If you look to the since 6000 BC, then in Babylonian and Sumerian, they're using the microorganism without any knowledge of the micro or brewing the beer. Even in 4000 BC, Egyptians have used microorganisms for baking the bread. In case of the present day 21st century, we talk about the microbiota. But the origin of microbiota study was started with Antonio Leeuwenhoek. And even with the simplest microscope, he has described various animal kingdoms. And till today, the same what he has described, no, they are quite useful to us for morphological differentiation of this organism. In case of the topic, what I'm going to talk regarding the role of microorganisms in extraction of matter. Of course, this scientific study of the involvement of microbiology and microorganisms in the metal extraction, it is as long as 2000 years ago. And there are data that in Spain, China, and India, the natural recovery of the copper and other base metals were carried out without our knowledge regarding the involvement of microorganisms. So this very fantastic organisms which are playing significant role in the extraction of metal, remediation of metal, and detoxification of metal. I am going to talk about these organisms today. So microbial leaching is the mobilization of a immobilized material by the involvement of either microorganisms or their metabolites from low-grade ores, mineral, concentrate, sludges, e-waste, and nowadays we know that the natural resources regarding the metals are depleting, and accumulation of the e-waste is increasing at an alarming rate. The e-waste and the various sludges, they can play the second resources for the metal availability for any country. And these organisms are playing very significant role for the extraction of this metal ion from e-waste as well as from sludges. When I have started my career in the biology, that is 1975-76, we were knowing only the two types of organisms. They are involved for the extraction of metal ion, that is thiobacillus thioxidans and thiobacillus pyroxidans. But in this 21st century, if we look at the types of organisms which are involved with a metal extraction or metal transformation, which belong to the wide group of organisms such as chemolithoautotrophs, iron and sulfur oxidizers. They belong to acidophilic and neutrophilic group of organisms. They also diversified in meso and thermophilic environment. Apart from chemolithotrophs, even the heterotrophs are also found to be playing important role in the extraction of metal. And archaea is not the exception. So, wide range of microorganisms are involved in this process. If you look to the some diversity of only specific group of iron and sulfur oxidizers, then it includes acid thiobacillus pyroxidans, acid thiobacillus pyroxidans, which is mesophilic in nature, sulfobacillus oxidotroph, sulfobacillus metallurgs, leptospirillum pyroxidans, and many more iron and sulfur oxidizers. Now, this will give you some idea regarding the morphology of these organisms. Unfortunately, the scientists and the students are afraid of working in this field because, as compared to the heterotroph, to work with this acidophilic autotrophic organisms is bit more difficult. The first difficulty we will face, that is very difficult to cultivate them in a solid media. You have to try liquid media, and the preservation is also a problem. While some heterotrophic organisms which are producing organic acids, which are also used for the extraction of metal ions from oxidized ores, which belong to Bacillus group, even Saccharomyces cerevisiae, Erogia, some of fungi like Aspergillus niger, Penicillium simplicitium, and in overall, if you look to the metabolite produced by these organisms, they produce citrate, gluconate, and oxalate. And due to the production of this organic acids, oxidized ore and even the carbonate ore, they can easily solubilize and you will get this metal ion in the soluble form, from which it is possible to recover this metal ion. If we look to the production of particularly precious metal, mainly gold, silver, palladium, and platinum, in this group of metals, the production of a hydrocyanuric acid, which is playing a very crucial role, and cyanogen microorganisms, they are involved in the production of HCN, and it is working like a nanotechnology because cyanide is quite harmful to the environment and microbiology as well as human beings. But these particular organisms are producing at a slower rate gradually. The organisms involved, they include the Bromobacterium violens, Pseudomonas fluorescens, Pseudomonas aeruginosa, Pseudomonas leptospiraceae, Cytobacter, Polyporus, and they work at alkaline pH, and the solubilization of metals were in the decreasing order of gold, silver, palladium, and platinum. Now, this will give you that what is the habitat of this acidophilic organism which are oxidizing ferrous and sulfur. So, Yellowstone National Park you can see the reddish color, the precipitation of a xerocyte, which is due to the natural activity of acetylthioacetylose peroxidase and acetylthioacetylose thioxide and acetylthioacetylose leptospirillum. So this is the main habitat from which these organisms were isolated. The other habitat from which these organisms were isolated and they are used for the extraction of metal ion, which is a Rio Tinto mine. And you can see that at the what large scale the mining activity and the extraction of metal ion can be carried out by this iron and sulfur oxide organism. And these organisms were reported not in the form of a culture known, but the activity in 1725. What other activity these organisms are responsible and which are you can see at any sulfide mine or gold mine, and you can see the beautiful color. Of course, it looks beautiful in terms of a visibility, but they are responsible for the environmental pollution, and we have to take care. These are some of the figures where we have got upon. Now, sulfuric Chitradurga, lignite Rajpadi, and millions of the gallon of water which is acidified due to this organism. And this slide will give you some idea regarding the pH is decreased in the acidic mine, which is as low as 2. And any water which is having less than 6 pH is not permissible to be discarded any water system. But if you train this organism, then the same organisms are responsible for the metal recovery as well as metal detoxification and metal remediation. And this is one of the examples where we have worked upon these organisms and we have given solutions. So you can see these two slides. So here vegetation was created by the solution which is given to the Rashpadi mine at Maljipura pumping stations from our department. The other aspects of the organism bioremediation of metals, I will not go into the detail about how the mechanism takes, but mobilization and immobilizations and this particular characteristics of a microorganism, either it may be intracellular, extracellular, either it may be the metabolic dependent, metabolic independent, maybe the precipitation of the organism, precipitation of metal ion, complexation of metal ions, and by this, these organisms are responsible for the removal of metal ions in the aquatic waste. There are organisms, some of them we are having in our laboratory, which are degrading the cyanides. We have the Bacillus pseudomonas group of organisms isolated from the Hatti gold mine, and they are showing the degradation of the cyanide. We have worked with various organisms for the bioremoval of silver, platinum, palladium, and gold. And these are also some of the data where you can find that many other scientists, they are working with a various group of organisms such as Lactobacillus, Cyanobacterium, Aeromonas, Neurospora. We have worked with Aspergillus and various species of the mucor. But similarly, we work with the Aspergillus mucor for the gold recovery. And platinum group of organism also, we have now more than 100 cultures which can work with the platinum group of the metal for the separation from the phase. Some other biotransformation of arsenic 3 to arsenic 5, because arsenic 3 is more toxic while the arsenic 5 is less toxic. A similar way biotransformation of a chromium 6 to chromium 2, even the biotransformation of a manganese and detoxification of mercury, cadmium, and copper. several autotrophic, heterotrophic organisms we were able to isolate from various mining sites as well as from the various other ecosystems. And we have studied the significant role of these organisms for the conversion of these metal ions into the less toxic. Now, this gives you some of the idea. Characterization of the arsenic resistance, arsenopyrite oxidizing, this autotrophic organism, acidic pyroxidans, what we were able to isolate from Hatti gold mine. And the beauty of this organism is it is not only tolerant to the arsenopyrite and arsenic, but it is utilizing arsenopyrite and arsenic for that growth. We have also have organisms which were isolated from the chromium 6 reduction and detoxification from various industries, and we got that Pseudomonas aeruginosa, giving the excellent activity. And we have also studied some of the cocci, which are very highly resistant to the arsenic, and that can be used for the sequenciations of the arsenic from the have also the organisms and very role from organism isolated from marine ecosystems and for the bioremediations of various organisms. One copper remediation was studied by the combination of bacteria and the Econia species, and we got more than 99% of the removal of this. So now this is my group because since 1977 we are working on the various aspects of the metal ions, metal remediations, metal leaching, and detoxification. So all these students, they have contributed for one or the other topic. So apart from this, in our laboratory, we have with various aspects where the microorganisms are playing important role. But as I cannot include everything, but we have worked with a high degradations, organic solvent tolerant organisms, therapeutic organisms and their application, biosurfactant producing role of microorganisms, role of various types of bacteria, and some of this actinomyces in the microbial exopolysaccharide repression, biosurfactant production, biodiversity of the marine life. So, with this presentations, I am thankful once again for the organizer to give me the chance to present. Thank you very much.
Thank you so much, sir. I request Kashish. Now I request Kashish to please introduce Professor J.S. Virdi.
He is formerly a faculty in the Department of Microbiology of University of Delhi South Campus. Is a well-recognized expert and mentor in the area of microbiology. His research work on molecular epidemiology of Yersinia enterocolitica, a food and waterborne gastrointestinal pathogen, is well recognized as an original prize of scientific contribution. In recognition of his work, he was conferred the Y.S. Narayan Rao Award by Indian Council of Medical Research. His team has been awarded as the first AMR Ideathon Prize by BIREC, Discovery Award by BIREC NESTA. He has received Science Hero Best Lab Mentor Award by InVitrogen. He has served as President of Association in Microbiologists of India, SAP coordinators, NAC coordinators, and member of Learning Outcome-based Curriculum Framework of UGC. He has published more than 70 research papers in peer-reviewed international journals, as well as he has also served as an expert member, nominee, and chair for several national and international committees. He is one and only J.S. Virdi sir. We welcome you to share your insights and perspectives with us.
See, somehow yeah, somehow I'm not able to share my slide. So if you can present the one I have sent you.
Uh, I think Dr. Vidhi, no,
Yeah.
I face the same difficulty. So you go to the screen, and there they are saying the window and the no on the left sides.
Yeah, yeah.
So there we have to first you know click to the window, then the
Okay.
slide will.
Let me see.
Yes.
Yeah. Okay, first window. But even then the share is not highlighting.
Now first you click to the cell, then you find a white screen. No, then the second option is window.
Think, sir, you can open your presentation first.
Yeah, yeah.
Then go to the screen window.
Yeah, I have opened my presentation.
Okay,
Then
so
I
you will
have
go to the window.
then go to window.
Click onto the window. Yes.
Yeah.
And click onto your presentation.
Oh, I see. Okay.
It's slightly change, I think.
Yeah. Got it.
And share.
Got it. Got it. Can you see my slide? Not yet.
Not yet. We can see the screen, but not the slide.
Yeah, yeah. Sorry, I'm not able to.
Sir, send me your presentation to me. I will present it to.
I have already sent it to Preeti.
Just a minute. Yes,
I have
I'm present. Okay.
Yeah, I have sent to Preeti.
Give me a minute, sir. I'm presenting it.
sorry, sir. Yeah. Is
Yeah, let me know it is visible or not.
yeah.
Okay, sir.
Is it visible now?
Yes, it is visible. It is visible. It's okay, perfect.
And you can hear me also. Can you hear me?
Yes.
All right. Shall I? So that means shall I start?
Yes, sir. Yes, sir.
Okay. See, I'm sorry for the this very small glitch, but anyway, see. So the our topic of discussion is developing a point of care test for the detection of antimicrobial resistance in bacteria. Now, what is this point of care test? May I have the next slide, please?
Yes, sir.
Yeah. See, a point of care test is a simple and a rapid test. See, it's written here less than 30 minutes. No, 30 minutes is only for antimicrobial testing. Every point of care test is not for 30 minutes. This 30-minute limit for the point of care test has been kept by some international funding agencies for the antimicrobial testing only. See, before they give funds, they want that the test which you are aiming must be less than 30. Okay, they give huge funds only when you present them with an idea or present them with a rationale that the test which you are going to develop is less than 30 minutes. So that's why I have written here 30 minutes. Requires minimum manipulation. A test requires minimum manipulations. Result can be interpreted unequivocally. That means there are no, you can say, controversies about the test, or there are no, you can say, ifs and buts about the test. They can be taken out, they can be considered, the results can be considered as the final. Do not require much expertise. Even a person with some technical knowledge will be able to interpret those results. Gives actionable results. That means when a test is done, a point of care test will give you directly actionable result. What action is to be taken? It will not require any other test or manipulations to be done further before you start taking action. Probably can be performed near the bedside, see, or preferably, not probably, preferably near the bedside or even very close to the patient in a side lab. Preferably, see, this green color, the simple sample in, result out. That is one format. That means you simply put your sample and within minutes or within hours, simply the results come out. So that is what we call as a sample in, results out format. So that kind of it is cost effective, scalable means it can be produced in very large quantities. So this is what a point of care test is. Next slide, please. Now, just to orient yourself that a point of care test, these are some of the very common, I'm sure most of you have seen these tests. These are point of care tests which have been developed and they are routinely being used almost all over the globe. For example, the first one which you see in the panel is a pregnancy test. Presence, pregnant or not, okay? It can be interpreted. Anybody can interpret it with this very little knowledge or instructions. It can be done at any other place. Second in the panel is of a glucometer. That's also on a strip itself, you can find out how much is your blood or urine glucose. Then there's another oximeter, another, all these we have seen very recently during the COVID. This is the fourth one is the temperature. Okay, simply you point out this gun kind of a instrument onto the person or towards his forehead, and then you'll be able to see that what is the temperature. So these are all what we call as a point of care test. Now this is the kind of a simple test is what we want for antimicrobial resistance or for antibiotic sensitive to be tested, so that any patient which is to be prescribed a medicine or an antibiotic must be prescribed a proper antibiotic immediately. But then it is a very difficult thing. It's a very difficult thing. Why? Because when we want to know the sensitivity of the microorganisms to the antibiotics, we always have to culture them. That means if you have to culture them, how can you develop a method for in 30 minutes or in couple of hours? You have to culture them overnight and then do the sensitivity testing. Next slide, please. So let's see, I give you how the antibiotics they are used in clinical setting. See, this is, for example, this is a patient in the first picture shows there is a patient in the ward. A sample is collected, and then after culturing the and sensitivity testing, the results go back to the doctor and he will prescribe the antibiotic. But many times he won't have that much time that the sample should go to the lab and then he should see the results, because after all it is an infection. It could be a very fatal infection. So he has to prescribe an antibiotic. That's what we call as an empirical antibiotic prescription. Some antibiotic has to be prescribed at that stage. Of course, he may change the antibiotic once the results come and we know what kind of a sensitivity to which antibiotic the microbes are, or the infectious microbe is sensitive. Similar, if you go to a patient or if you go to a clinic, see, most of the time you will see what should be the ideal thing. Ideal thing is that your sample of your infecting material or your body fluid, it should be taken, goes to the lab, to a centralized lab, and then it comes back with the results, and you start yes, Professor Dave, is some problem? All right, see, but here also you must have seen when you go to a doctor, you are immediately given an antibiotic. We never wait for the sensitivity testing. Ideally, it should have been done that, but then it takes a long time before the sample goes and then comes back, results come back. A physician has to prescribe an antibiotic. Now, what is all? Now, this is a practice which I'm talking all over the globe. Don't think that this is I'm talking about India or some less developed countries. No, this is a practice all over the globe. This picture which I'm telling you is all over the globe. Antibiotics are prescribed like this. Then they are changed accordingly after the sensitive test comes in. Many times even no sensitivity test is done, and the antibiotics are prescribed. Without that, person goes second time to the physician. If he is not cured, another antibiotic is prescribed. But many times the person gets cured at the first instance. Why? The reason is that the physician is prescribing a highly broad spectrum antibiotic. May I have the next slide, please? See this patient with a suspected infection, which it may be in this green color you can see. Okay, patient with a suspected infection, which is the patient is in the ward or OPD or clinic. It goes to a doctor, who prescribes him an antibiotic empirically. That is at that time, because he has prescribed an antibiotic without any antibiotic sensitivity and testing, he will try to give an antibiotic which is highly broad spectrum. although there may be second generation or third generation antibiotics which are effective against this infection, but he is giving a highly broad spectrum, a new antibiotic, a recent antibiotic, and that is the problem. See, those new antibiotics, those highly effective antibiotics must be preserved for life-threatening infections. They should not be given for throat infection, for skin infection, or for ear infection. Certain several low on the low, you can say, rung of the antibiotics in terms of their spectrum, they will equally do well. So when we are prescribing such a large amount of such a broad spectrum antibiotics, microbes are becoming resistant. The more and more antibiotics we use like this, microbes are becoming resistant. And the same antibiotics, which is basically to be used for highly, you can say, surgical patient, heart surgical patient, some orthopedic surgical patient, they are being prescribed for throat infection. So microbes are becoming resistant to them. So this is something which is one of the major, this is one of the, I'm just talking about only one reason. There are several other reasons of the rise of antibiotic resistance, but I have just taking only one point to highlight this. Now, ideally between these two, no, yeah, please go back. Yeah, you can see on the left side, there are culture antibiotic testing. See, there is a Kirby-Bauer method, there is a minimal inhibition, and then you can see this is E-test. This is called as a E-test, which will directly give you MICs. On the lower panel, you can see Vitec, you can see Phenex, or you can see Microscan. These are very, very sophisticated instruments for antibiotic sensitivity testing. That means you can do the testing, have the sample of the patient, do the antimicrobial sensitive testing, and then prescribe the antibiotics. But this again takes not less than minimum of 18 hours on these equipment. So we are back to the square one here. Whether you are doing a automatic or a culturing, it's remain the same. Now, that is why this need for a point of care test is to prescribe the proper antibiotics to save the broad spectrum antibiotics, and in the long run, how to save our antibiotics. Otherwise, the kind of the type or the you can say the progression at which the antibiotic resistance is rising, it is projected by that by 2050 or so, we are going to be in a big trouble when most of our microbes will become resistant to antibiotics. No new antibiotics are being discovered. Okay, these are several. This is a very complex problem. So by 20 or 50, it is projected that this is going to be almost catastrophic kind of a situation. But developing these kind of tests will help us to save our broad spectrum antibiotics. But to address the problem of antibiotics resistance, this is not the only approach. This is a diagnostic approach. There are several other approaches. For example, develop new antibiotics. Okay, don't use antibiotics for the environmental kind of things where they're required. So variety of things are there, but I'm mainly discussing it from a diagnostic point of view. Next slide. See, this is a technology. It's an imaging-based technology. Imaging-based technology is one of the method which is currently being considered to be one of the best method, which is you can say nearing the ideal point of care format. But even then, it is quite far away from that. Can I use my pointer here? No, I think I cannot, isn't it?
Yes, sir.
Can I use my pointer? No, because you are presenting here, na?
Yes, sir.
Yeah. All right. So, can you see the low? Yeah. can you see the in this figure the two growth curve type of a thing? One is the classical, the dotted line is that of the optical density, whereas the upper one, which is a solid line, that is of a single cell. That means, and then you go down, you will be able to see there are two panels under this. One is where the microbes are increasing in a large number, and it is becoming quite crowded. The microbes are becoming quite crowded, and that's just at the lower end, just near this, adjacent to this, under lower, you will see it is when the single cell. When they're still in the single cell, then you cannot detect them optical density. So we have to use these single cells. These no, this is not my work, nothing of us. This I'm telling you is one of the top you can say scientists who is doing this all work in Sweden and has published this work, and as I would say is one of the front runners in this area, who has come very closer to a point of care test principles, and has shown the feasibility of this test. Now from here you can see those colored panels around side, on the other on the one side. See these colors, the inside, the circle inside is the one which contains antibiotic, and outside is the single cell suspension. This outside, that yellow color which you see, is nothing but it is a microfluidic channel. Now microbes, they are put into this, and then they come in contact with the antibiotic, and you culture them only for 5 hours, 4 hours or 5 hours. You need not do a culture of 18 hours to do any kind of a sensitivity, but minimum of 5 hours, that is what they have shown. You take 5-hour culture, single cell, do with this kind of a microfluidic channel, expose them, and then last lower panel you can see these microbes. We do lot of imaging. Millions of these microbes, they are then imaged, and then we see what kind of changes they have come in their morphology. And all this data, using artificial intelligence methods, all this data is collated and then presented whether these 5 hours is the microbe sensitive or is the microbe resistant. So that means we are able to tell, and then you can see all this that can be done in a simple Tarasky plates or our microtiter plates with microfluidic channels. Of course, there will be microfluidic channels, there will be special plates, and with a simple microscope. But then, of course, with some software which will analyze all these millions of the cells, the shapes of these millions of cells, because the shapes will be very minimal changes. There will be some inherent changes, shape changes in the microbe. So there is a lot of, you can say, work, science behind it. And then you will be simply by doing a short exposure, not 18 hours or 24 hours. That is out, totally out. And short exposure and imaging and AI, you will be able to tell whether it is the microbe is sensitive to which antibiotics. Next slide. Now this is in a so that means you go to, that means last line, it says combination of short culture in the presence of antibiotics and bacterial imaging plus artificial intelligence, because you are using millions of the cells, their morphology, changes in morphology, and drawing a conclusion about their sensitivity or resistance. You go to the up, I say, remember the idea is to develop a point of care. That means all the prerequisites of POC has to be fulfilled, which I told you in the first instance. Remember, the result of sensitivity of infecting bacterium to antibiotics should be available in less than 30 minutes. As I said, this is one of the conditions for some of the international funding agencies. Okay, 1 hour. By the American funding agencies, these are 1 minute. They are 1 hour. Whereas these are British standards, the 30 minutes. They want to tell method for 30 minutes, less than 30 minutes. So all culture methods are out. Although in this, we have reduced the culture from 14 from 18 or 24 hours to 5 hours or so, and people they are trying to further bring it to down. Anyway, this is one method. As I said, this is one of the front runner methods in this space. There are several other methods also for point of care tests. People are using nanotechnologies, people are using certain other, but imaging combined with this has come closest. Now let us see what is our approach. Next slide, please. See, our approach is just read the from University of Delhi South Campus. You see the title is written underneath, resistance genes array. We are using is resistance genes array based rapid detection of AMR and algorithm driven therapy. We are not doing any culture at all. We are not thinking of doing any culture in for this. And you can see in the panel some of the members, the major members of this team, but of course there are a lot number of other people involved here. Students are involved here, some other mentors are involved here. Next slide, please. See, this is the one. If you see, this is a flow diagram is the principle. That means from a drop, that yellow colored drop, which you can see on one side, is you take your sample and sample containing your bacterial culture, then do the DNA isolation, do its sharing, all in situ in a very, you can say, small format. That means those kind of format has to be used. And then this DNA, this single red color things, and then you can see those green dots. That means we'll label them. How this labeling is done, that is a different thing. Variety of things they can be done. Depending upon that label, the detection method will vary. Now then you go to the lower panel, from side panel, you go to the lower panel, and then you can see these labeled DNA of the isolated microorganism of the organism, isolated from the organism which has been sheared, they will hybridize with a gene array. All those yellow colored bar like things with a star above them these are all hundreds of small arrays. And then those stars, which means those which hybridize, those arrays which hybridize, these arrays are known to us. So from this highlighting, all those information goes to the upper panel, which will tell us, which will suggest us antibiotic 1, 2, 3. Suggestions are based on a WHO recommendation that you first have to prescribe an antibiotic which is second generation, third generation. You don't have to prescribe the fourth or fifth generation broad spectrum antibiotics, because we are going to save those antibiotics. There are several hundreds of infections which can be cured with a second generation, third generation antibiotics. We need not prescribe the third generation, fifth generation highly broad spectrum antibiotics, recently discovered antibiotics, those containing a lot of you can say beta-lactam inhibitors. We need not prescribe those for every small infection. We need to preserve those antibiotics. So this recommendation is based on that. That means the algorithm incorporates all that data into that. So this is the format. Next one. So you can see there is no culturing of the microbes here. This is all the gene array which I told you. This is a large number of these different circles tell you about the different antibiotics, and underneath the names of the databases, they are there. They give us the list of the antibiotic genes. Okay, next slide, please. This is what we are using to draw sequences. We have made our own in-house database for beta-lactamase basis. But of course, our database now becomes redundant because hosting on the McMaster universities, they have come up with a very, very broad spectrum. But in the process, when we started, we made our own database also, but that was only on beta-lactamase genes. Next slide, please. So this is the work components. That means we have various teams. Resistance, there is a component one. There is a resistance gene array selection. There is an in situ DNA hybridization and melting, labeling of the DNA, resistance gene microarray design, algorithm driven selection of the antibiotics, and then integration of all the components of all these things. So we have various teams in which we are involved, mentors. I have some collaborators. Along with this, our PhD students, postdocs, MSc, not MSc students, PhD students, as well as certain other mentors are involved. And we are working on different aspects, and several things of these things have been finalized. Next slide, please. This is the design. Finally, we have gone. This design, I'm showing you almost the final version, but it has gone iterations, or you can say improvements for, you can say almost this is the 6th or 7th iteration. And finally, it comes out to the size of a lap, not a laptop, I mean the iPad kind of a thing. It's slightly intermediate between iPad and a phone. And finally, the prototype comes like this. Not prototype, I mean a design comes like this. Next slide, please. Here it is. See, this is our panel one. Number one is module one. Then second in the center is module 2 and number 3. Module 1 is the one which will, everything happens in situ. Simply you put a drop of your sample and then DNA isolation, shearing, and then it's labeling. Then the panel 2, it goes into microfluidic channel, microfluidics area, where we are taking a lot of, you can say, information, help of certain engineers. They keep on guiding us. This microfluidic channel is the one which contains the DNA array, where the hybridization takes place and the signals are picked up and then processed. From here, we finally goes to the treatment option 1 and treatment option 2, which, as I said, is based on the WHO recommendations how the antibiotic is to be used. Okay, that means we will be suggesting, the software will suggest only those antibiotics first which are not the highly broad spectrum, and they are either 3rd generation or 2nd generation, which will do the purposes. But if there is another, you can say, grade higher also, but we'll try to save the most broad spectrum antibiotics. So this is how you see we are at a various stages of development. As I said, you can see how much work it involves, how much molecular biology, how much engineering, how much software. So a lot of people I have involved. We are doing all these things, and we are at the same time looking for someone who would develop a prototype for this. So this is what we are at this stage. Next slide, please. So here I end up. This work will continue. These are all the students. See, these are not the students who have contributed. I would like to thank them, because these are the ones who have been developing all genomics in my lab, especially the beta-lactamases, based on their work only, the knowledge, the publication, that we have come to developing this kind of a product-based kind of a thing. They have not done any product kind of a thing. They have barely done basic knowledge thing, and using that, we have come to a stage where now we have a group and we can make a product development kind of a thing. Next slide, please. So these are the various funding agencies. And for this work, the mainly funding agencies is the last one, which you can see, BIRAC and NESTA. Next slide, please. So this is the place where all this work has been done at the University of Delhi at South Campus. And with this, I would like to thank you all.
Thank you so much, sir.
My pleasure. My pleasure.
In the garden of discourse, our professor are the rarest bloom. Let us invite another eloquent gardener to cultivate their thoughts beside them. I request Kashish to please introduce Dr. Swaranjit Singh.
It is with great pleasure that we introduce our esteemed guest, the remarkable Swaranjit Singh. With impressive achievements, he is the chairman of biodiversity and chairman pioneer in microbiology. He is president of the MBSI Chandigarh Tri-City, that is Ladakh, Nagaland, Manipur and many more associations. Not only this, he is member in 2 WFCC committee member task force, National Biodiversity Authority. He has CC and Bank, now the first IDA in India, and contributed to the society and masses by spreading science to the remotest corner of the country. Mr. Swaranjit Singh is listed in top 2 global scientists for his tremendous work in his personal career. His publications were listed in top 70 publications of CSIR in its 70th foundation publication are in high impact factor journals and the articles are highly listed in literature. He is awarded Loyola Environment Awards. His accomplishments and commitment to excellence make him an invaluable resource. So please join us in giving a warm welcome to Swaranjit Singh sir, whose journey of compassion, academic excellence will undoubtedly inspire us all.
Thank you very much for the nice introduction. I'm so happy to be here today with all of you. And I know many people who are attending this webinar. So, Preeti, will you please start the PPT?
Yes, sir, I'm starting it.
Okay. I'm very thankful to the organizers for giving me a chance to talk on this great platform. Yes. Please go to the first slide. Yes. So today I'll talk to you on effective antibiotics and the need to conserve microbial diversity. We spend so much time and effort to get strains for our research. So we cannot afford to lose our strains. So we must preserve the strains very carefully in a culture collection. Next slide. We are very lucky that we have 3 hotspots of biodiversity in India, and they must be conserved. We are fortunate that we have the Indo-Burma region, which comprises of 7 sister states like Nagaland, Assam, Meghalaya, Tripura, Manipur, and so on, and the Western Ghats comprising of Kerala, Karnataka, Maharashtra, Goa, and Gujarat. And we have the rich Andaman and Nicobar Islands. There are a lot of novel genus and species that can be isolated and tapped for making high-value compounds, antibiotics, enzymes, and also use them to keep the environment clean because they can degrade pollutants and transform metals. Yes, next. The world urgently needs new antibiotics which are very effective. Antimicrobial resistance is rapidly increasing and spreading, but only a few large companies do antibiotic research. New antibiotics can be discovered by 1. Improving existing antibiotics. Number 2. Rediscovering old antibiotics which we have forgotten and using old drugs. And the third one is rediscovering new chemical diversity. A lot of chemicals are also very good as antibiotics. There are some organizations like CoAD, IMI-Enable, and CARP-X which help and assist in the discovery of new antibiotics. Next, please. There are so many journals like Nature, etc., asking us to develop new antibiotics because we need them. By the year 2050, if we do not have effective antibiotics, many people may die. So we have to look fast for new antibiotics and save millions of lives worldwide. Next, please. How can we get new antibiotics? The first one is rediscover old antibiotics. The other one is target-based approach. Third one is synthetic chemical diversity. We discover untested new chemical diversity. Then from natural products, we repropose old drugs and improve existing antibiotics. Next, please. There was a Desi antibiotic under trial. Next slide, please. There was a Desi antibiotic under trial in Europe which saved the life of a woman. A Nepali woman flew from Nepal to Lucknow for treatment of tummy tuck. And Desi antibiotic, which is developed by Aurangabad laboratory by an Indian pharmaceutical company, saved her life. So, we have to get old antibiotics out and use them to kill the bacteria which are pathogenic. Next, please. Again, a journal, Microbial Biotechnology, reported mining for novel antibiotics in the age of antimicrobial resistance. So we really have to work very hard because a new antibiotic may take 15 years before it comes to the market. It takes a long time to come to the market. Next one. What are the approaches to antibiotic discovery? There are many approaches for antibiotic discovery. First one is bioengineering, in which there is biochemistry, in which they use high throughput screening, phylogenetics, co-culturing. Then we have bioinformatics, genomics, and bioengineering, in which synthetic biology, genome editing is done. So all these approaches together can help in the development of a new antibiotic. Next, please. This is the timeline of antibiotic resistance. Currently, in the last 2 or 3 decades, hardly any new antibiotic was reported. 80% of the gonorrheal infections are now resistant to antibiotics. There are 440,000 new cases of drug resistance tuberculosis annually. In the future, new drugs have to be discovered to combat drug resistance. Strep throat to scrap knee could kill many people. By 2050, as I just told you, many people may die due to drug resistance. We cannot afford this. We have to look for new antibiotics which are effective. A simple hip replacement would result in 1 in 6 deaths. The cost to treat drug-resistant cases is estimated to be more than double of what it is today. Next one. This is the ecology of antibiotics and antibiotic resistance. Globally, 20 to 80% antibiotic classes are released into the environment in active form via the excretion of drugs in urine and stools. And their release imposes a selective pressure on bacteria, leading to the selection of resistant strains. The strains migrate between different environments, thereby creating global movement of antibiotic resistance, genes, and determinants. Next one. These are the antibiotic classes. The penicillins, the oldest antibiotic discovered, it prevents bacteria from making cell wall. Then we have sulfonamides, which inhibit folate synthesis and also inhibit DNA replication. Then we have tetracyclines, which inhibit protein synthesis. Next one, please. This is discovering antibiotic in a new way. There is an eye chip. This is known as tixobactin, a new class of antibiotic. In the Nature study, tixobactin killed Staph aureus and Mycobacterium tuberculosis without the bacteria developing resistance to the antibiotic. Because this strong antibiotic, tixobactin, binds to the lipids and binds to lipids that builds the cell wall. Next one. These are the many antibiotics in the pipeline and their spectrum of activities. As I told you, a new antibiotic takes about 15 years to come to the market. This is the antimicrobial peptides and the mode of their action. I mean, some of them act on the cell wall, some of them kill the bacteria and make them free. Now, I'll tell you something about some different molecules, which are very important molecules. They are biosurfactants. There is a lot of structural diversity in biosurfactants, and they are potential for novel antibiotics. You can see they are big molecules. First is the rhamnolipid, which is produced by Pseudomonas aeruginosa. It is having two rhamnose units linked to a fatty acid. Then you have the trehalose lipid, two trehalose linked to the fatty acid. Then you have a cyclic lipopeptide, which is known as surfactant. And this is having 7 amino acids linked again to a fatty acid. Fatty acid is very important. Then we have a yeast candida biosurfactant known as sulfurose lipid. This is having 2 units of sulfurose linked to a fatty acid. And then we have a particulate surfactant. Next one. Now, surfactants. Next one. Next one. Next one. Yeah, this I've already talked about. Next, please. Yeah, these are the properties of biosurfactants. You can tap. The biosurfactants are having many activities like antifungal, antibacterial, antiviral, antimycoplasm, neurological, anti-cancer property, immunomodulatory property, anti-adhesive property. So these biosurfactants, I sometimes call them magic molecules because they have so many activities in one molecule. And they can be very good antibiotics in the future. They have antiviral property. Surfactant, which is a lipopeptide, is very effective against virus. Sulfurous lipid also is very active against virus, and it has got anti-HIV property. Then we have viscosine, which is very active against herpes simplex virus 2. This is a US patent 5965524. Next, please. The biosurfactants also have antibacterial property. Preeti, can you go to the next one? one back. Can you go one back? Yes. Rhamnolipid is again a very strong antibiotic. It is effective in very low doses. For Mycobacterium, only 16 mg per liter rhamnolipid is required. Against Staph aureus, only 8 mg per liter is required. And there is a glycolipid made by Candida antarctica, which is showing very strong antimicrobial activity against gram-positive bacteria. Next slide. This is an antimicrobial surfactant lipopeptide, which we synthesized in our laboratory with my student. And we published this work in Trends in Biotechnology. We named the biosurfactant as N1 because the name of my student was Pooja Singh. So Pooja Singh did very good work on biosurfactant, and now she is in Symbiosis Pune as an assistant professor. So you can see that this N1 biosurfactant is very strong antibiotic against Staph epidermis, Streptococcus mutans, and Staph aureus. So this is a very good indication that biosurfactants can be used as very effective antibiotics in the future. Next one. Now the biosurfactants also have antifungal property. There is a molecule known as ytterine, which is antifungal. Ytterine A is effective agent for mycosis. Now rhamnolipid is active against Aspergillus niger in very low concentration of 16 mg per ml. Next. Now these biosurfactants, the rhamnolipids, are very, very helpful in wound healing. They can heal the wound very fast. So when you have a burn on the hand or on the leg, if you apply a cream containing biosurfactant rhamnolipid, it enhances the wound healing and it speeds up the healing of the burn. So this is another important property of biosurfactant. Next one. Even the microbial cell. Today is International Microorganism Day. Even the microbial cell can be considered as a biosurfactant because the microbial cell is composed of hydrophobic and hydrophilic moieties. So it acts like the biosurfactant. Next one. Now, the bacteria generate oxygen. The microbes are very important to us and they provide us pure oxygen to breathe in. And they produce more than 50% oxygen of the world. We think that plants are making oxygen. Yes, they are making. But bacteria alone is making 50% of the world's oxygen for us to breathe. This is the picture of the Baltic Sea, which is having a mat of cyanobacteria, the blue-green algae, on the surface, and they make tons and tons of oxygen for us to breathe. That means microorganisms are very beneficial to us. Only a small fraction of microbes are pathogenic, but most of them are very, very beneficial to us. Now, we have made so many cultures. We have made so many efforts and hard work to get the cultures. Now we must preserve them properly. So let me tell you what is a culture collection. You all must be knowing, but I want to tell a little bit. They conserve microbial diversity for teaching, industry, and research. Each microbe is composed of a set of genes that are unique. If the microbe is lost, the unique set of genes are lost. So we cannot afford to lose a microbe That's why we must preserve in a good culture collection. It is having diverse microbes under the same roof. They may have cultures from Antarctica, from the deep ocean trenches, high mountains like Mount Everest, and isolates from space shuttles may be there in the culture collection. They are having cultures of regions. For example, Himalayan region, some may have of the Indian Ocean, some may have of Antarctica under same roof. Next one. There are 3 types of culture collections. You have gone back. You please go ahead. Please go ahead. Culture collections. Ahead. Please go ahead. More. One more. Yes. There are 3 types of culture collections. The first one is a personal collection. Supposing I am working on oil degrading bacteria, I'll have only cultures of oil degrading bacteria. But when I take up a new project on production of alcohol, all my cultures will be lost. The oil degraders will be lost. So it is very important that the personal collection is deposited to a culture collection which can maintain your microbes for many years. Then we have institute collection, which caters to the need of hospital and institutes. They may or may not make catalogs. Then we have a service collection. This is very important. In India, we have 3 big culture collections, which are also International Depository Authority, IDA. They are very well equipped, professional culture collections with all facilities of preservation and maintenance, and they publish catalogs. Next one, please. These are some of the activities of culture collection. They supply cultures to the research people. They identify cultures by biochemical test and also by molecular sequencing. They give pure cultures to you and they compare it with the type strain. Now, supposing you ask for Pseudomonas, you must get Pseudomonas. If you get Bacillus, then your work will suffer. So the culture collection will give you the right culture for your research. Then you can deposit your cultures. You can also make patent deposit in the culture. Next. Next, please. These are some of the methods of preservation by a culture collection. The first one is under mineral oil in -70 to -80 freezer, then freeze drying or lyophilization, active slands, which is not very good method. Then thiobacillus cultures cannot be freeze dried. So they have to be in liquid culture, which is transferred every 1 month. Then you have acidophiles growing at pH 3 or 4. They are also in liquid culture. They cannot be lyophilized. Now, for sending cultures abroad, you can send on paper disc or gelatin bead. You can take out 1 gelatin bead and retrieve the culture. Next. These are some of the methods how they preserve cultures. The first one, 55, is a LN2 canister in which you can pour liquid nitrogen and store your culture. Then you have a deep freezer, -70, -80, and also lyophilize your cultures. Next one. These are some of the cryoprotectants used for, cryopreservation. Skim milk and glycerol are very cheap but very effective. So when I was working in MTCC and Gene Bank, we used to preserve the cultures in glycerol 10% and lyophilize in skim milk. Serum is good but very expensive. So we always used skim milk and glycerol for preservation of cultures. Next. Now I told you there are 3 IDAs in India. The first one was MTCC and Gene Bank, which was elevated to IDA on 4th October 2002 by the teamwork, hard work, of all the scientists in MTCC. Then the second idea was MCC Pune. And the third one is National Agricultural Microbes, which is in Mao in UP. All right. So India is very lucky and very fortunate to have three good ideas now in the country. So the take-home message is search new antibiotics so that people don't die in 2050 and conserve microbial diversity and biodiversity so that we do not lose important strains which will help us in the coming days. Thank you very much. Thank you very much.
Thank you, sir. Everyone's presence has filled our room with brilliance, among whom we like to add their unique spark to this constellation of voices. I request Kashish to introduce Dr. Preeti Prasad.
To our intelligent guest of honor, Dr. Preeti Prasad. She is an experienced teacher, researcher, presently working as an assistant professor in botany in GJ College under Patliputra University, Patna. Armed with a great gold medal during graduation with masters from Delhi University and PhD from Ranchi University, skilled in PCR, RT-PCR, agrogross gel electrophoresis, RNA and DNA isolation, SDA. She has a diverse and impressive background of education. Preeti ma'am graduated from St. Xavier's College, did her master's from University of Delhi. She has been the faculty member of Amity Institute of Competitive Examination, Class Institute, and assistant professor. I would like to enlighten us with her kind words. Ma'am, please.
A very good afternoon to everyone. Am I audible to all? Okay. A very good afternoon to everyone, honorable guests, resource persons, and the organizing team, the wonderful one. Myself, Dr. Preeti Prasad, Assistant Professor Botany from GJ College, Patliputra University, Patna. Thank you for inviting me. And as a resource person on such a wonderful occasion of Microorganisms Day, I feel so honored and blessed to be a part of this webinar. And I'm a botany person, as it is already introduced, that I'm a botany person and want to throw some light on how plants and microbes affect each other and play a significant role in our daily life through one of my studies or a project. And so my topic of discussion is plant extracts and oral health and how microbes are related to it. We'll talk about it. So let's start. I'm sharing my slides. Can you see that?
No, ma'am.
Okay, I'm trying it again.
Okay, ma'am.
Yes, this one. Can you see that?
Yes, ma'am. Now it is visible.
So my topic is plant extracts and oral health. First of all, as we know that the oral hygiene is an integral part of health of every individual, and when neglected, it results in various oral disorders. But due to high emergence of oral infections, the fast developing resistance by bacteria to antibiotics, the adverse effects of the chemical-based remedies currently used in dentistry and financial considerations, there is a need for alternative prevention and treatment options which are safe, economical, and effective. So here the oral infections which have been linked to bacterial species, especially the mutants group, as we all are celebrating Microorganisms Day, but a little bit I am just talking here the negative part of the
microbes, not to say the negative part, how they are affecting our life in another way. So unhealthy diet pattern and lack of oral hygiene promote the growth of these bacteria. And a number of methods are used to treat oral infections, but it is important to look for the role that plants play in maintenance of oral hygiene, since many of them have medicinal properties. And there is a long history of the use of plants to improve dental health and promote oral hygiene. When compared to chemical based remedies, plants always keep an upper hand, and in many Indian and African homes, even today, teeth are cleaned using stems and barks of certain plants. Here I'm just sharing a pic. Here a lady, I was talking to a lady, a tribal lady of the Tupudana area of Ranchi. I just want to discuss the interaction. That interaction was wonderful. How much the tribal people are aware of nature, its significance and their utilities. Even many of us living in the city, or we are so updated with technology, we don't know. And her knowledge, her dedication towards nature was just mind blowing. So, some plants that I considered for my work are Azadiracta indica, which is commonly known as neem, then Pongamia pinnata, Pseudium guajava or guava, Mangifera indica, then Ossimum sanctum, Mentha arvensis or mint, Punica granatum or pomegranate, Vitex negundo, Smilex prolifera, then Centella asiatica or Brahmi. And the procedure of plant extract preparation, very easy and very simple. The leaves were washed and air dried at room temperature, and then for several days until the leaves become crispy, and they were ground into fine powder using the sterile mortar and pestle, and in different solvents like ethanol, methanol, acetone, and distilled water, the plant extracts were prepared, and they were prepared using Soxylate apparatus and Rota evaporator, as you can see how they were being prepared. And bacterial strains and their revival was done it was actually done in the lab of St. Xavier's College, Ranchi, during the year 2012 to 2014. Bacterial strains which were considered for the work, and they were brought from MTCC Chandigarh. They were cured from there. Here, the Streptococcus mutans with the MTCC number, it is mentioned, Lactobacillus fermentum, Pseudomonas aeruginosa. These three were the microorganisms that we considered for our work, and in different growth media they were grown, and the revival of bacterial culture was also done in the lab itself of Xavier College, Botany Department, through a proper process. Due to the lack of time and other preoccupations, I'm going a bit faster. I'm sorry for that. An antimicrobial assay of the plant extracts that was done by disc diffusion method. As you can see, sterile disc with plant extracts were kept, and then the nutrient agar medium with bacterial growth and zone of inhibition was monitored. Like this. Diameter of the inhibition zone, including a disc diameter of 5 mm, was considered, and the different data that is generated for the different plants, like for Azadiracta indica against Streptococcus mutans in different solvents, as you can see in the acetone extract, then the ethanolic extract, methanolic extract, and water extract. In the same way for the other plants also, for against three different microorganisms for all the different plants considered for the work, and in different solvents also. It was a time-taking affair, but it was very interesting. Like this, you can see these are the data. Like this. So the effect of chlorhexidine as positive control was also considered for the work, and the results showing zone of inhibition through disc diffusion. These are the results. I'll discuss at the end about the conclusion also. Here are the results of showing zone of inhibition through disc diffusion method, as you can see, for the different solvents and for the different plants. And the graphical representation, the bar diagrams show the x-axis It's showing the concentration, the y-axis zone of inhibition in millimeters square. Here for the different colors are showing for acetone, ethanol, methanol, and water-based solvents, water-based extracts. This is for Azadirachta indica on Lactobacillus fermentum, then on Pseudomonas aeruginosa, then Pongamia pinnata on Streptococcus mutans, then Pongamia pinnata on Lactobacillus fermentum, again Pongamia pinnata on Pseudomonas aeruginosa, like this. I'm coming directly to the conclusion part. Yes, the conclusions, what we drew from the work. So all the considered extracts showed significant results against oral pathogens taken for work. From the results mentioned above, it is clear that extracts obtained from Azadirachta indica had maximum inhibitory effect against the considered oral pathogens, especially against Streptococcus mutans. Yes, neem. Every part, even all the parts of neem, is useful to us in one or the other way. Means for as we all know that neem, the leaves of neem, that is used in different for curing different diseases also. For example, during measles and all, we are using neem, especially during the months of May to July. People who are very much like they are dependent on nature, or you can see that in our daily household also, the use of utility of neem is very significant. And in the study also, it was shown very clearly. Also, the extracts prepared in the solvents other than distilled water had more efficacy. The maximum effect was shown in higher concentrations of plant extracts like 100% and 80%. Yeah, it is expected. But however, distilled water extract had shown better inhibitory effect over the ethanolic extract in case of Azadirachta indica, which was a bit strange. But other plant extracts also showed positive results against the selected oral pathogens. Azadirachta indica leaves possessed good antibacterial activity, confirming the great potential of bioactive compounds, and is useful for rationalizing the use of this plant in primary health care. It has been reported that the 70% ethanolic extract of Pongamia pinnata leaves has potent anti-inflammatory activity against different phases, acute, subacute, and chronic, of inflammation without side effect on gastric mucosa. They also observed significant antipyretic action of extract against Brewer's yeast-induced pyrexia. After Azadirachta indica, the other plant extracts which had maximum effect was Pongamia pinnata or Karanj, which was followed by Cedium guajava, as well as Smilax prolifera in its efficacy against dental caries and oral flora. It was reported that Cedium guajava leaves showed good activity, but acetone extract was highly active against Streptococcus mutans, and also reported that the aqueous extracts of Cedium guajava have caused a reduction in the addition of the early plaque settlers. The present study showed that Cedium guajava has excellent antibacterial and biofilm inhibition activity against Streptococcus mutans. Other plant extracts, which include Mangifera indica, Punica granatum, Vitax negundo, then Centella asiatica, Mentha arvensis, and Osmum sanctum, also showed positive results against all the considered oral pathogens. Also, the acetone extracts had more antibacterial effect than the methanolic one. This may be because acetone has better extracting capacity over methanol, which may be attributed to the ability to extract the natural antimicrobial compounds such as alkaloids, flavonoids, terpenoids, and phenolic compounds from the plant. So that's all. This was my work, and here I have to stop. That's all. So thank you for bearing me. And I just went very fast due to some other work also. So thank you so much for having me.
Thank you.
Thank you.
As wisdom illuminates our gathering, let us kindle the torch of dialogue by inviting Professor T. Ramesh. Before we proceed, I would like to invite Preeti to host the following event.
It gives me immense pleasure to introduce our resource person, Dr. T. Ramesh. He is working as the scientist in GS Gill Research Institute in Guru Nanak Vellachari. He has been the guest faculty in Department of Fisheries Biotechnology, Tamil Nadu in 2022. From 2016 to 2022, he was the assistant professor of Department of Biotechnology, Hindustan College of Arts and Science, Kalambakam. 2006 to 16, he was in the Faculty of Diploma in Medical Laboratory Technology, Naga Institute of Paramedical Science. He is master's, bachelor's, and PhD. He did master's and bachelor's in zoology and PhD in biotechnology. He received grant from Tamil Nadu State Council Science and Technology under Scheme 2021 to 23. He received grant from Tamil Nadu Directorate of College Education under PhD scholarship scheme. He published 17 research articles in different research magazines, etc. Editor of 2 books and an author of Book of Biotechnology with 2 other authors. He is life member of Antiviral Research Society, Indian Science Congress Association, Zoological Society of India, and Society of Indian Academy of Environmental Sciences. He is an associate fellow and executive member in Antiviral Research Society as well as state coordinator of Microbiologists Society. He has been 8 times for different categories awarded. He has participated in Indian Science Congress. He has done many social activities. As we saw, with all respect, I would like to invite him for his kind words of inspiration.
Thank you so much for your wonderful introduction about me. Can I present? Hello.
Yes, sir, you are audible.
Good evening to one and all present here. So, my topic is role of microorganism in insect pest management. So, before going to discuss about the topic, so I would like to tell what is pest and what are the deleterious effect of pest. So, pest is an organism which is harmful to all plants, animals, including humans. So, the terminology pest is derived from the Latin word which is mean by contagious disease. So, it can spread or infect from one organism to another organism. So, the meaning, literal meaning of the pest is with contagious disease. So the few examples of pest includes non-insect pest as well as insect pest. So non-insect pest, which is include animal which cause disease to other animal and humans, and some microorganisms, pathogenic microorganisms which cause disease to human, animal, and plants. So insect pest, particularly insect pest, the arthropodon small insect which cause disease to plants and which is affect the agricultural productivity is called as insect pest. So insect pest. So insect pests are living organism belongs to the phylum Arthropoda, which is harmful to agricultural productivity, which is harmful to agricultural plant, which cause damage to the productivity. So insects are responsible for significant loss of the to the world's total crop production annually. So every year you can face lot of problem because of this infection. So hence the pest control measures should be taken against to control and mitigate the action of insect in the agricultural land. So there are a lot of ways are there, lot of methods are there to control the insect pest in the agricultural land. So there are a lot different classifications are there insect pest, so based on the occurrence. Okay. So based on the occurrence, insects are classified into regular pest, occasional pest, seasonal pest, persistent pest, and sporadic pest. based on the occurrence, based on the frequency to visit the agricultural land. So this classification is made. So these are the different classification of pests. So loss due to insect pest infection. So every year we face lot of agricultural loss due to insect attack. The first category, this pie chart is clearly representing the loss due to the insect pest. So it occupies the insect which is occupied the third place to damage the agricultural productivity. So there are 26% of the agricultural productivity loss is happened due to insect pest attack. Okay, so the crop yield losses due to insect pest attack is 26% annually, facing the lot menace or issue because of insect attack. So there are lot of reason which is behind the insect pest outbreak in India. According to Indian scenario, there are a lot of reasons are play a vital role in the disease outbreak, insect pest outbreak in agricultural land. So first is anthropogenic activity. That is nothing but human activity which disturb the ecological balance and cause the insect pest outbreak in the agricultural land. So destruction of natural enemies by using the applying the chemical pesticide in the agricultural land, which is one of the first reason to cause the pest outbreak in the agricultural land. Then intensive and the extensive cultivation, agricultural practice also another reason for pest outbreak in the agricultural land. So introduction of new varieties of crops in the agriculture field is also another reason. So introduction of new pests in the new environment, that is also a reason. So accidental introduction of pests from the foreign countries. Okay, through when you importing the grains or something from the foreign countries, so that through that importing material goods, the new insect all invade inside the country. So that's also another problem for the insect pest outbreak in the agricultural land or India. So this is a chart, this pictorial representation clearly explains that how the insect get the life cycle of the insect. Okay, so the adult lay egg on the plant leaf, stem or body part of the plant. So the egg is hatched into the larva. So larva undergo the process is called as molting. Molting is nothing but shedding of the outer cuticle. So after the metamorphosis, it will be converted into pupa. So pupa is converted into adult. So this is the life cycle. So each and every stage, the larval stage is a very dangerous stage, adult and larval stage. So this stage, insect will attack the plant vigorously. So we should control the insect at the larval stage or adult stage. So that is the main aim of the pest control. So pest control method. So we should control or mitigate the action of insect in the agricultural land. Then only you can get the good yield in the agricultural land. So there are a lot of methods which is adapted to control the insect pest in the agricultural land. First one is called as physical and mechanical method. Second one is called as chemical method. Third one is called as biological method. The physical and mechanical method, there are a lot of tools and devices used to eradicate or remove the insect from the agricultural land. So you can use a light trap, a small water trap, or hand picking method. So there are a lot of methods which is adapted to control the insect in the agricultural land. Okay, that is called as physical method. Manually you can apply labors to collect or remove the insect from the agricultural land. So that is called as physical and mechanical method. So there are a lot of disadvantages are there in that method. So it will take time and labor. Huge amount of labors are required. So you have to pay that labor. So that is a first problem. This method is applicable for only in small area. In vast land, acres and acres of land, we can't apply this method. So this require repeated application also. So it will require more manpower, very cost. So these are the main disadvantages of the physical and mechanical method of pest control. So the next is chemical control. So chemical control. So after the Green Revolution, there are a lot of insecticides are introduced in India to control the insect pest in the agricultural land. So the few of the insecticides are listed in this slide. One is malathion, second is lintane, third is phenolphthalein, fourth is permethrin, carbaryl, pendicocarp, and dioxocarp. So these are the major and predominant insecticide which is applied in the field to control and kill the insect pest in the agricultural land. So it will, the effect of this insecticide is very very good, and it will it will given the good result, but at the same time it will cause deleterious effect to the environment. It will cause the, it will disturb the ecological balance. It will kill the non-target insect in the agricultural land. So it will settle in the soil and it will persist in the soil for a long time and kill the soil beneficial microorganism and kill the birds and other animals which is living in the agricultural land. And also it will enter into the food chain and it is biomagnified and uh, ill to, cause dangerous effect to the human population also. Okay, so it will cause some endocrine disturbance and it will cause the water imbalance in the human body. It will affect the circulatory system, respiratory system, reproductive system, nervous system, and excretory system. So it will get bioaccumulated in the body of animal and human and cause lot of problem in the human and animal. So, and also it will disturb the ecological balance. So that is the major disadvantages of chemical pesticide. So scientists are done research and they go for alternative way of controlling the insect pest in the agricultural plant. So that is such promising one is called as biological control. So biological control is nothing but to control the insect pest by using the biological mean is called as biological control of insect pest. Okay, so there are a lot of animals and insect and lot of living organisms are employed to control the insect pest is called as biological control of insect pest. Okay, so dragonflies, okay, that is small insect which is used to control the other unwanted harmful insects. So dragonflies are a beneficial insect which control the harmful insect which is present in the agriculture field. Okay, it will eat the eggs and larvae of other harmful insects. Then parasitoids, some birds. Birds also, it will devour the larva and pupa of some agricultural insect which is present in the field. Some nematodes, microorganisms. Okay, so microorganisms are important or small living organism which kill the insect pest. Okay, so that is bacteria, virus, and fungi. Okay, these are three predominant microorganisms which is employed to control the insect pest in the agriculture field. Okay, so the advantages of biological control method. So it is no cause no damage to the environment. It's eco-friendly. It's very clean and green. So only it's targeted the insect are eliminated. It's only very specific to the insect pest. It won't kill the other pest. Okay. So expense is very low. The production cost also very low. It's gives a sustainable outcome. So these are the major advantages of biological control of agricultural insect pest. So microbial insects. Okay. So after the emergence of biotechnology and microbiology, so researchers are producing the insecticide by using the microorganisms. Okay. So there are three different predominant microorganisms which is used to prepare the microbial insecticide. First one is bacteria, second one is fungi and virus. So these are three different microorganisms which is used to prepare the insecticide against the agriculture insect pest. First one is called as bacteria. So bacteria, as you know very well, that it is a small prokaryotic single cell organism which is living in water, air, land, okay, everywhere. So it's omnipresent, which is exist everywhere. Both kind of bacteria are there. Most of the bacteria are beneficial to the human population. Some of the bacteria cause disease to human population. That is called as pathogenic microorganism. So here, some soil bacteria are involved to produce the insecticide. Okay, so there are 90 species of bacteria is identified to pathogenic to the insect pest. Okay, so baseless there are different five different microorganisms are identified to control the insect pest very vigorously. First one is called as Bacillus thuringiensis, Bacillus papillae, Bacillus parasius, and Cocobacillus acidorum, and Serratia massicans. Okay, these are the five different bacterial species which is used for the production of microbial insecticide. Okay, so first Bacillus thuringiensis. Okay, it's called as BT. Okay, it's a slow-forming gram-positive cruciferous soil bacterium which produce a kind of toxin is called as BT toxin or cryotoxins. Okay, it's a crystalline protein toxin which kill the insect pest. Okay, so when you apply the insecticide prepared by the Bacillus thuringiensis, this bacillus thuringiensis toxins is get attached in the cuticle of insect, the outer cuticle of the insect, and it will get inside the body of the insect, okay, through the mouth, okay, and enter into the gut, okay, it will damage all gut cells which is present in the gut epithelial area. Okay, so you enter into the cell and make a pore or channel, and it will arrest the ATP synthesis inside the cell. Okay, so in such a way it will kill the insect pest. Okay, so the target insect for Bt toxins are Lepidoptera, the order Lepidoptera insects. Okay, that is moths and butterflies, flies and mosquitoes, beetles and wasps, bees, ants, and sawflies. Okay, these are the insect which is attacked by or susceptible to the Bt toxins. Okay, so these are the insects are controlled by using this Bt toxin. So the mechanism of Bt toxin. Okay, how it will get react to the insect and how it kill the insect. Okay, so when the toxins enter into the gut of the insect, okay, will damage the epithelial cell membrane. Okay, the cell which is present in the gut epithelial region, and it will make a small pore or channel in the cytoplasm. Okay, so leading to the cellular ATPase. Okay, lead to the loss of cellular ATP. Okay, and it will finally the ATP is the level of the ATP, amount of the ATP is decreases. It will finally leads to death. Okay, so it is in such a way the toxin will destroy the insect. Okay, so this is a mechanism of Bt toxins on insect. The next one is called as fungi. Okay, more than 750 species of known to infect the insect. Okay, the fungi are eukaryotic organism which body is made up of mycelium. Okay, it will harbor on the cuticle or outer body of the insect, and it will kill the insect. Okay, so mostly causing disease to insect. Most of the fungi that which is used to prepare the biopesticide will cause disease to the insect. Okay, some attack the insect through the cuticle. So as I said already, it will attach in the outer shell of the insect or the cuticle of the exoskeleton of the insect, and it will start to germinate, and it will penetrate inside the body. Highway and mycelium have penetrate inside the body, and finally it release the toxin and kill the insect. Okay, the ultimate thing is it will infect the insect and secrete the toxin and kill the insect. Okay, this is the fungal insecticide. Okay, so the predominant fungal species which is used to prepare the insecticide is called Vivaria bassiana. Okay, it is a deuteromycin fungus. Okay, it's natural occurring. It's naturally present in the soil. Okay, so it is used to produce the fungal based insecticide. Okay, so it is formulated as a vegetable powder. Okay, or it is a granule, dispersible granule, or oil based emulsifier form. It will be prepared. Okay, it will attack. It will used against the coffee berry borer insect, diamond black moth, thrips, grasshopper, and whiteflies. Okay, these are the insect which is very susceptible to the attack of Vivaria bassiana. This is the mechanism of Vivaria bassiana on insect. Okay, how it will kill the insect pest. Okay, so when you apply the Vivaria bassiana based insecticide on the field or in the body of the insect pest, so it will attach on the outer cuticle and started to germinate. Okay, so then it will slowly it spread this mycelial and it will penetrate inside the body of the insect, and it will cause disease to the insect, and finally it cause death, and again it will cause produce spore, and it is ready to attack another insect. Okay, so this is a cycle. This is the mechanism of fungal based insecticide. Okay, next viral based biobased. So some of the virus, mostly virus are pathogen. Virion means toxin, poison. Okay, the Latin word virion means toxin or poison. So virus means mostly it is dangerous to human, animal, and plant. It will cause disease to human, animal, plant. So these viruses are isolated and it is used to produce the pesticide or insecticide to kill the unwanted agricultural insect. Okay. So the virus belong to the family Baculoviridae is predominantly used to prepare the viral based biopesticides. Okay. So Baculovirus is a group of double-stranded DNA virus that infect the insect. Okay, so they infect the insect through the ingestion. Okay, ingestion. So once it's ingested by the enter into the body of the insect and it release the baculovirus particle. Okay, that is called as capsid and capsomere. The DNA get entered to the body of the insect. It started to produce its body part, viral body part. Okay, capsomere, capsid, and other part. Okay, then later it will get assembled inside the insect body, and it will kill the. Finally, it will kill the insect, and it will come out from the insect body and ready to attack another insect. Okay, so this is called as viral based biopesticide. So this is the mechanism of baculovirus based insecticide. Okay, so when the viral particle is ingested by the larva, then it dissolution of the protein capsule in the midgut and release the virions. Okay, so virions are released inside the midgut region of the larvae, and it will produce its body part, that is a capsule, capsid, capsomere, and tail fiber and DNA, everything. Okay, then it get assembled inside the insect body cells. Okay, then it will proliferate and release from the insect body. Okay, finally it cause death to the insect and it release from the insect body. Okay, so this is the mechanism of viral based insecticide. Okay, so finally advantages of microbial insecticide. So as I said already, physical method of insect control require more manpower. It is very limited, very limited. Since we are using to the very limited area, it is applicable. It's a very vast agriculture field, we can't apply the physical and mechanical method of pest control. Chemical method is very well and it's well and good, but at the same time it cause and damage the ecosystem. Okay, so that is very danger to the ecosystem and it will affect the future generation also. So we can go for alternative. As I said, we can use microbial pesticide like bacteria, virus, fungus based pesticide to control the insect pest. Okay, so these apart from that, there are a lot of advantages are there by using a microbial insecticide. First, it's a friendly to non-target species. Okay, as I said already, if you use chemical pesticide, it will kill all honeybees also and other beneficial insect also that is involved in the pollination. Okay, endomophily. So pollination, it will kill the other organism also. But this bacterial and microbial based pesticide is very specific and unique to the target pest. Okay, so it is very friendly to non-target species. Do not cause pollution. Okay, it won't cause any pollution to the environment. Okay, it's relatively cheaper. We can easily produce the bio-based pesticides. Okay, trichoderma viride, even small farmer and vendors can produce trichoderma-based pesticides. Okay, then pests never develop resistance against this microbial-based pesticide. Okay, if you apply the chemical pesticide, it will develop resistance against that particular chemical. Okay, then you have to increase the dosage of the insecticide. Okay, so that will be another very big menace a danger to the environment. Okay, but in the case of microbial insecticide, so you do not that the insect do not develop any resistance against that particular um, insecticide. Okay, so these are the major advantages of microbial insecticide. So this in concluding remark, I will tell you that these microbial insecticides are much promising kind of insecticide to control the insect pest population in agriculture field. Okay, so thank you so much for providing this opportunity. Very much thankful to all organizers who organize this wonderful international webinar. Thank you so much once again. Thank you. Thank you so much.
Thank you, Ramesh. Now, in the tapestry of our gathering today, let us weave the threads of wisdom from another gifted soul. I request Anjali to introduce our esteemed resource person, Professor Tanzima Yasmin ma'am.
With great honor, let's introduce our next resource person, Professor Tanzima Yasmin, Emeritus Chairman of Department of Biochemistry and Molecular Biology. She has qualified BSc Honors and MSc with first class in biochemistry and also did PhD in biochemistry from Rajshahi University, Bangladesh. Besides these, she has done diploma in biochemistry from the Tokyo Institute of Technology She has the great contribution in the field of teaching from being professor in the Department of Biochemistry and Molecular Biology to being the director and professor in the Institute of Biological Science in the University of Rajshahi. Ma'am have utilized her knowledge and time in attending special institute programs and courses which also includes training program of NITUV, INASP, scientific workshops at international level. She has completed various dissertations on topics like biodegradation of organic waste and many more. Possessing the ocean of knowledge, she has done more than 25 scientific paper presentations at international and national seminar and conferences. Despite of these, she has been the supervisor of around 10 scientific research projects, in some of which she has worked as the director and chief investigator as well. Also, she is still guiding many research projects of PhD and MPhil. Ma'am's contribution in the field of science is almost infinite because she has co-authored more than 60 publications, is the member of more than 5 editorial boards, and have affiliated as life member at many societies including the international also. For her immense contribution and knowledge, she has been honored with many awards, including the UNESCO and Munbosho Award of Tokyo Institute of Technology. Apart from these, she has also visited countries like USA, Germany, Mauritius, and many more. Now, without waiting for a second, let's auspiciously welcome Professor Tanzima Yasmin ma'am.
Hello, good afternoon. Good afternoon.
Good afternoon, ma'am.
But I can't upload my slide. I'm trying.
Ma'am, should I present your presentation?
Yeah.
Ma'am, have you sent us a
No,
PPT?
you didn't ask me.
Ma'am, please send it to the email ID. I will present it.
Okay, just a moment. So, good afternoon, everyone. Thank you for inviting me and this special occasion, World Microbiology Day. Where is that slide? So, should I send to Rajeshwari? The email ID? Which one? Which address?
Microbiome Society, Jharkhand. Ma'am, just a minute. My Microbiome Society, Jharkhand @gmail.com.
Biology.
Microbiome Society, Jharkhand.
Microbiome Society.
Jharkhand @gmail.com.
Jharkhand spelling?
And JHAR
JSAR
KHAND.
KHAND.
Yes, ma'am.
At Gmail.
@gmail.com, ma'am.
.com. You got it?
No, ma'am, right now I don't.
Now? Where is it?
No, ma'am. Just let me check my spam.
Sorry?
No, ma'am, I hadn't got.
The message is given, it's sended.
Just a minute, ma'am, I'm checking.
Microbiome Society, Jharkhand.
Yes, ma'am.
It's sent. Could you text in my WhatsApp?
Okay, ma'am, I'm texting you. Give me a minute.
Okay, sure. Okay, I think I may continue. First, I would like to thank Professor A.M. Deshmukh. He introduced me in the Microbial Society, India, in Maharashtra. And today, I'm interested about to share my some experience to do work in a methylresistant Staphylococcus, a substantial public health problem that is in not only Bangladesh, it is all over the world. I think I didn't get any message. You sent me.
Ma'am, can I know your number, ma'am? As in, I haven't.
Just now, I got message this one.
It's a high message.
46083. Last one.
Ma'am, is the message high?
Hi, yes.
Yes, ma'am. It's my number. Send it to me, ma'am. I will present it.
Okay, but your email ID.
Okay, ma'am. I'm giving. Just give a minute.
Sorry for the trouble. Because I'm trying to upload the file. It's not.
I'm sending.
Okay, okay. Preeti Kumari.
Yes,
Is
ma'am.
it you? Yes,
Yes, ma'am.
Yes, Preeti Kumari.
Yes, ma'am.
Preet. Uh, 0705. Received?
No, ma'am.
Preeti Kumari, Dev. 0705.
Yes, ma'am, but I haven't received it. No. And send me the link in the WhatsApp. It can also open in WhatsApp.
Now, you received?
No, ma'am. No, ma'am.
Yeah, but the message it shows send it to you.
Sorry, ma'am, but it not didn't come to me. I'm refreshing it again, but no, there's no message. Email. Ma'am, if it possible, you can send me in WhatsApp.
I'm trying.
Okay, ma'am.
Okay, just a minute.
Yes, ma'am.
Yeah. Now you receive?
मैम आपने व्हाट्सएप पे भेजा है या मेल पे?
Mail and WhatsApp both.
WhatsApp? No, ma'am. In WhatsApp, I also don't get. I think there is some issue.
Just a
WhatsApp.
minute.
Okay, ma'am.
You check one more mail.
Ma'am, mail.
Mail. That's WhatsApp. It doesn't sending from mail. I've sent twice. Preeti Kumari. Adab.
No, ma'am.
Even Microbiology Society Jharkhand.
No, I mean both email I hadn't got.
But this is showing that it already been sent to you.
Ma'am, do you have Rajeshwari's email?
Yes, I have sent it. Rajeshwari email should I send?
Yes, ma'am. Send it.
Rajeshwari Pradhan? Patwardhan?
Rajeshwari, are you present?
Rajeshwari Patwardhan, her name?
Just a minute, ma'am.
S. Rajeshwari Raj. S. Rajeshwari Raj.
Hello, Kriti. The size of the file may be large, so tell ma'am to share through the Google Drive.
Ma'am?
Ma'am,
Yeah?
please share through the Google Drive. The size of your presentation may be more than 25 MB.
It's only
So you'll
15
have
or
to
16 slide.
15, 16 slides also will make it big enough. So may not be able to mail. You can share using your Google
But
Drive.
it said it's sent mail. Email has been sent.
I didn't receive it. Don't know why. Yes, ma'am. Now I get it.
Yeah,
Now.
you get it?
Now
You
I
got
get
it?
it. Yes,
Okay,
ma'am.
thank you. I think
Presenting
it is time.
Network issue हो सकता है, ma'am.
Yeah, maybe.
Yes.
Okay, shall I start? Sorry for delay.
Yes, ma'am. Give me a minute, ma'am. I'm presenting it.
Okay, sure. Yes, thank you. Sorry for the trouble. Today, I want to share some experience to work with methicillin-resistant Staphylococcus. This is highly resistant, and we have worked with the antibiotic not only for that antibiotic is used only for a human, but for animal also. And veterinary, this is also one type of zoonotic disorder from animal to human. It could be transmitted. because for this study, we studied that pet animals, usually we keep in our house dogs, different birds, animals, rabbit, and those are worker with the animals, and even for chicken, and we get it from the market. So all type of food and animal, and even in the patient who are admitted in the hospital or outdoor patient, we collect the sample from different sources, and from there we found that methicillin-resistant Staphylococcus aureus MRSA is a substantial public health problem all over the not all over the world. It is also concerned in Bangladesh, like our developing countries. And what we have been found that Bangladesh is a very highly dense population country, and we found from our study that most of the hospital admitted patient or outdoor patient, there are almost 94% people are Staphylococcus aureus methicillin resistant. For this, we isolated. You can go for the next slide. Next slide. So we collect a sample from different sources, pet, animal, nasal source. From nasal, we collected that swab, and chicken that from slaughterhouse, that chicken, and we just frozen chicken we collected and arranged with the water, and the sample are collected from the supermarket, and then we use for isolation identification with the conventional culture method, and after that we grow that isolated sample from different culture condition, and then go for DNA extraction and PCR analysis, and culture sensitivity test also done. Next slide. These are the conventional method, and these are the protocol we used for isolation and identification of bacteria. Then next slide. Next slide, please. Can you move the slide? Okay. So previous one. So we collected from different source that mice, guinea pig, rabbit, pet dog, frozen chicken, even fresh raw cow milk, and the laboratory workers who work with the different animal or mice or pet dogs, rabbit. Those are the worker, or even in the chicken and farms, and some outdoor patient and indoor patient also. In a tertiary care hospital, we collect sample from nasal, trachea, and different umbilical or even wound infection, and total among them that Staphylococcus sample were present among 753 sample to 708. Almost prevalence is 94% prevalence are Staphylococcus positive sample. Next slide, please. These are the culture plate, and first one is the mannitol agar culture and fermentation plate, and this is the yellow colony, and black show that the fermentation of yellow color. And this is the blood agar, that is the hemolysis on the 5% sheep blood agar formation of hemolytic colony. This is the indication of the culture. And these are the gram staining of Streptococcus aureus. Next slide, please. And it is a positive cocci. And culture characteristics for catalase test, and these are the slides, and Streptococcus species. Those are isolated from different sources, and that we have seen there are a couple of bubbles, and that bubbling, no bubbling, that is no coagulation forms, and these are the negative control we use. So this is the cultural characteristics of Streptococcus species. Next one. Next slide, please. So almost whatever we isolated Staphylococcus from different sources, and we go for sensitive test and resistant test. So methicillin resistance, almost maximum sample we can here the slide shows that resistant most of the from 87 sample, this is a 62 sample were methicillin resistant and sensitive only. And vancomycin, this is the sensitive, almost all are sensitive to vancomycins, and these are not resistant. So most of them are methicillin resistance. Next one. Next slide, please. And from chicken sample, previous one, sorry. The chicken sample, the Staphylococcus aureus infected sample, and the antibiotic resistance profile from chicken range sample. So this is the bar diagram we have presented. These are the antibiotic. Those are the resistance with the Staphylococcus, and this is the percentage of chicken sample. These are infected with the Staphylococcus aureus, and this is a coagulase positive and coagulase negative test. So most of the antibiotics are resistant. We have found from the chicken sample. Next one. And this is the these are the hospital record. Oh, we have collected this patients. Those are in the outdoor patient in a tertiary care hospital from Bangladesh. So I have presented only 3 years data over there. It is from 2013, 2014, and 2015. But the recent data we don't disclose due to there are. Because we cannot mention the hospital name right now, and we use the back data. This is not the current one. And this is the sensitive pattern of Staphylococcus patient. Those are attained in the tertiary care hospital outdoor patient. So, and this figure belongs to the antibiotic resistant pattern, and most of the antibiotics are resistant and maximum antibiotic resistance, and it's a huge problem nowadays. Next slide, please. And whatever we have found that from pet dog nasal swab sample and infected with the Staphylococcus aureus, these are the coagulase negative and antibiotic sensitive pattern of Staphylococcus suggested that these are the resistant to penicillin. Among the 95.83% of frozen chicken sample were found infected with the Staphylococcus aureus. Among them, 68%, more than 68% sample were coagulase positive Staphylococcus, and antibiotic sensitive pattern of Staphylococcus isolated. That is the antibiotic ampicillin, amoxicillin, cephalexin, ciprofloxacin, erythromycin, gentamicin, doxycycline, and oxytetracycline. These samples are resistant and more are antibiotic sensitive. And there are 100% resistant to ampicillin, and 80% were oxytetracycline and doxycycline. Hydrochloride amoxicillin, ciprofloxacin, this is showed 77.5%, and cephalexin 38.33%, and gentamicin, this is the least resistant, only 13.33. And from raw milk also we have found these are the resistant to penicillin and erythromycin 75%, amoxicillin also 100%. But the isolated are sensitive to ciprofloxacin and oxacillin and cyclooxacillin and neomycin. And the broad spectrum antibiotic ciprofloxacin, this is the increase the resistant pattern. And so we know that drinking of raw milk and raw milk product, we don't suggested to drink or intake raw milk. And this could be a risk for human infection. And those patients are admitted in the hospital So these are the indoor patient. Those are the admitted patients. So they showed that these are the Staphylococcus positive, and these are the methicillin resistant anti-Streptococcus aureus resistant. And among them, 94% were resistant to cotrimoxazole, and 84% are resistant to erythromycin, and 71% to nalidixoxin acids, and 49% are doxycycline, ciprofloxacin, and vancomycin. This is the sensitive, and ciprofloxacin 21%, and the penicillin. These are the least resistance. So what? Next slide, please. So this is the our sensitive and resistant pattern. So this is the graphical presentation of the methicillin resistant and vancomycin sensitivity to the mice, guinea pig, rabbit, and human. And this is the methicillin pattern, resistant patterns of the outdoor patients in a tertiary care hospital. Next slide, please. And we have isolate DNA, and we go for HDS space. These are the methicillin resistant Streptococcus aureus, and these are line 1. It is the low range of HDS space marker, and there are 12 line, and show that the different isolate of emerge is one. And 4 types of C types, and D type, and E type E7, E per E, and 7 type F8, and G type G9. So these are the different HDS space we observed from the methicillin resistant Staphylococcus isolate. And next one, we go for next slide, please. This is the molecular detection, and we have applied for the Streptococcus aureus. And here the PCR product which are amplified 270 base pair gene, and this is the specific fragment of Streptococcus aureus. This is the shows on the agarose gel 1 to 7. These are the positive line and the positive control, and this is the line NC. This is the negative control, and these are the amplification of new gene. And from Streptococcus aureus, this is 100 base pair ladder, and lane 1 to 4. This is the amplified new gene, and from the Streptococcus aureus. And these are the next slide, please. And from the present study, we have included that dog, rabbit, mice, guinea pig, poultry, and these are the exotic species, both as a cause of infection and a healthy carrier. Also, we identified that methicillin resistant Staphylococcus aureus from various species, and it may be we are consuming also type of milk and chicken and the pet animals. So those are the we are a close contact, and maybe the workers who are involved with that animals from there, this type of interaction with the human, with the animal, this could be a possible source for human infection. And some are working very closely to the animals. These are the veterinarians and the laboratory workers, even in the people who are taking care of this type of animals. And people are involved with that cooking process. They were not maintaining the all health and hygiene. And so these are the possible source of transmission, and from animal and to the food materials, raw materials that we are consuming. So this could be a potential source of methicillin resistant Streptococcus aureus from animal to human infection. And for the public health concern, so Bangladesh, we have studied within that Bangladeshi context, and we have found from the study this is a great deal with the antibiotic resistant patterns of Streptococcus aureus with that methicillin resistance. And from the previous lecture, whatever we have found that 2015, 50, this would be a very crucial time for us for antibiotic resistant, and we have to develop new antibiotic. For this approach, we go for isolated pure novel antibiotic. Those could be solution from this type of problem. So we are hopeful that for use of abuse of antibiotic or misuse of antibiotic, because nowadays it is a global health problem, not only for Bangladesh, from other country also. So from this is my brief introduction or brief presentation for the World Microbiology Day. And so this is the day we are come in contact with other people, other scientists, and from this whole day seminar. So it is a great opportunity for me to exchange or share some words, and I am improving myself for this type of platform. We are communicating each other. Thank you very much for giving me this opportunity, and for it's a great opportunity to share my work with you. Thank you very much once again, and sorry for the trouble, whatever that for that reason I take some more time. Thank you very much for
Thank you, ma'am.
and introducing me. It's a great honor for me. Again, I would like to my gratitude to Professor A.M. Deshmukh and the St. Xavier College of Jharkhand. Thank you very much. See you soon.
Thank you, ma'am. Moving further, our stage is open and our hearts are eager to receive the eloquence of another voice. We would like to step forward and paint the canvas of our conversation with their words. I request Mubashara to introduce our esteemed resource person, Mr. Javed Iqbal sir.
Good evening to all. It's a great pleasure.
Sorry to interrupt, Mubashara, your voice is not clearly audible.
Good evening to all. It's a great
Yes.
pleasure that we introduce our esteemed resource person for today's webinar on the International Microorganism Day, Mr. Javed Iqbal. Javed sir completed his BSc in Biotechnology from Bangalore University, Bangalore. And after that, he pursued his MSc and PhD in Marine Biology from Pondicherry University, Andaman Islands. His expertise are marine ecology, biodiversity monitoring, environmental impact assessment, habitat mapping monitoring, and assessment. Currently, Javed sir serves as a marine biologist in the research institutes and the environmental services industry. Javed sir has a great research experiences such as marine ecology at Greenfield Environmental Consultancy, Al Khobar KSA, in which he have done marine ecosystem monitoring and assessment and general environmental consultant. Also, project scientist first at National Center for Coastal Research, Ministry of Earth Sciences in India. Again, Javed sir has done marine ecologist freelance at Ajaz Environment Consultant, Dubai, UAE. Work like writing proposals and method statement under underground water sound analysis, spectrogram generation, and interpretation. Also, senior biologist instructor at UNICEF Bihar, India, and doctoral research at Department of Ocean Studies and Marine Biology, Pondicherry University. Talking about his achievements and awards, he has got Rotary Youth Leadership Award in 2011 by Rotary Club at Port Blair, and Best Research Paper Award for an excellent paper presentation by Andaman Science Association. At last but not least, Maulana Azad National Fellowship by the Ministry of Minority Affairs, Government of India. Please give a warm and cheerful welcome to Dr. Javed Iqbal sir.
We welcome Mr. Javed Iqbal sir to step forward and illuminate us with their words. Javed sir, am I audible to you? Javed sir, am I audible to you? I request Rajeshwari to please call Javed sir.
Yeah, you just.
Hello everyone. Just let me know if all the people can hear my voice.
Yes, sir.
Anybody? Okay. Sorry for the being late here. Actually, I was in another meeting. Okay, so thank you for this opportunity, and I would like to mention especially the College for inviting me as being a resource person, and I thank to all to attend this program. And let me introduce myself. My name is Javed Iqbal, and right now I'm in Saudi Arabia, Al Khobar, working as an environmental consultant. And I did a PhD in marine biology from Pondicherry University, and I am actually a benthic ecologist. So today I will be talking about the macrobenthos rather than microorganism. So let me share my slides and PPT. From where I should share. Okay. Yeah, so can anyone say like my screen is shared?
Yes, sir, it is.
Okay, fine, thank you. So today I am going to present and talk about biotic indices, which is based on macrobenthic organism, and it include all the invertebrates which abode and home to the benthic soft bottom habitats. So my topic is biotic indices, monitoring and assessment of ecosystem health. When we are talking about, I am talking about the ecosystem health, then I mean of marine benthic ecosystem health by using macrobenthic community and their responses to different kind of disturbances. Especially, I would like to point that their responses are basically with the organic matter enrichment and organic matter influences by the sewage discharges, industrialization, and human population growth along the coastline. So, what is the purpose of my presentation? Because as we all know that like world ecosystem, in particularly marine ecosystem, is uh, uh, human threats, like continuously under constant human pressure, and these pressure are actually making the ecosystem services and goods they provide are exponentially declining. So, because of United Nations, they have set almost 17 sustainable development goals to achieve by 2030 and extended to 2050. So most of the environmental assessment and sustainable development goals are run around the biodiversity-based monitoring and assessment of environmental degradation program. So these are the very common practices adopted at the world level, and most of the world oceanic ecosystem. For example, we have Water Framework Directive 2000, and it was the common program adopted by the European Union. And before that, we have OSPAR program, which look after the Atlantic, Northeast Atlantic Ocean, and look after the marine pollution and sustainable development. So, just similar to this kind of initiative and programs in India, we have COMAPS program, and it is running and operating since 1992. And the similar program is monitoring of coastal pollution along the east and west coastline of Indian coastline. And before this, it was run by National Center for Coastal Research, and yeah, 6 months before I was working there, and I was involved in the same monitoring of coastal pollution program, that was the seawater quality monitoring program. In that monitoring program, we had to monitor and sample the entire coastline, and in that we have to collect the sample from starting from microbiology and planktonic community, including phytoplankton, zooplankton, as well as macrobenthos, including all those essential oceanic variables, including basic temperature, salinity, water depth, and all. And so these are the program which is being taken care by these Waterfront Directive and OSPAR, as well as COMAS program in India. So these are the real purpose of doing the assessment and assessing the ecological quality health of the marine benthic ecosystem. And another best reason to assess the degradation level of our ecosystem health, because this ecosystem provides ecosystem services, and it goes from supporting, provisioning, regulating, and culture, and all these together making it easier for well-being of human. So this is the reason why we have to assess the ecosystem health under the threats of human pressure. So today we are going to talk about this sustainable development, UN United Nations Sustainable Development Goals, and we will talk about and we will discuss about the life below water and sustainable goal number 14. And as well as next we are going to talk about these kind of organism rather than microorganism. And if you look around, this organism mostly are invertebrates and mostly marine rhyel. So if we think of this clinoid and ostracode, these are the very microscopic level of organism. So we don't include in macrobenthos, rather than we include macrobenthic organism as which sizes range from 500 micron to up to 100 centimeter and 1000 mm. So these ranges of sizes of any animal or organism living on the benthic soft bottom and shallow water benthic habitat, we call it macrobenthos, and they constitute a biotop of particular soft bottom sediment. So, so our objective of this presentation is what are the ecological indices I have applied in order to check these ecological indices into Indian water, and what would be the drawback and constraint of using these ecological indices into ecosystem health assessment in context to Indian water and Indian scenario. So, if there is any strain, then we can use some of the other indices which are giving ecological response rather than giving ecological response. They are giving functional response because functional responses are more accurate and more numeric in terms of assessing the ecosystem health because they give the functional aspect of the ecosystem, and because of knowing the functional aspect of the ecosystem we can assume and we can predict what could be the capacity of the ecosystem to be resilient in the future if we proceed with the some measures to protect and conserve the ecosystem and restore the particular ecosystem. So our main objective is my main objective is here to apply these ecological indices, marine-based ecological indices into Indian water. In particularly, I have applied these indices data collected from the Andaman and Nicobar Island. And beside this application, there are several authors we have applied who has applied these indices into Indian mainland scenario from the east coast as well as the west coastline. So, for considering the disturbances, what could be the possible aspects of degradation of ecosystem health? So, once these are the pressures which actually acting upon the ecosystem health and ecosystem services. So one major cause of the, since I am talking about the organic enrichment, so we have taken the major source from where these organic matter enrichment could have happened or could happen to the marine ecosystem, particularly it associated with population growth, human population growth, and ultimately leading to the discharge of domestic and industrial sewage and direct discharges of the sewage into the marine environment. And these are leading to the organic matter enrichment as well as phytoplankton blooms. And after that blooming, that organic matter also settle down into the bottom and leading to the hypoxia or anoxia condition, and together making the environment organic enriched. So after having this kind of pressure, that ecosystem come to into a particular state where we can observe the anaerobic degradation and lowering of dissolved oxygen as well as sediment hypoxia and anoxia. So what could be the responses after this state reaching the this state of the particular system? So responses, particularly we have seen into the microbenthic community and their structure, composition, and abundance, as well as their diversity, they change accordingly. So we know that in the real ecosystem or in near real nature, we have two kind of strategy, biological strategy, which mostly the organism or animals they adopt. One is either they will be sensitive or tolerant. Third kind of behavior and ecological responses are opportunistic. So once if some disturbances happen, then all the K strategies and T strategies like tolerant and sensitive species they vanished, and those opportunities are optimized by the opportunistic species. So based on this model, actually lots of indices apply and developed into the European countries, mostly the temperate countries, and because their ecosystem was somewhat very stagnant, and we see seasonal stratification and leading to lots of disturbances under the water surface, mostly in the benthic environment. So that the reason why, and because of this kind of responses of microbenthic community, um, most of the European Union countries, they have developed their indices to assess the benthic quality status. So the usefulness of developing benthic biotic indices is it gives us very concrete and numerical values, so that we can simply tell based on the numerical values, we can simply tell that management to what to do and what not to do from the scale from 0 to 1 or 0 to infinity, depending upon the rationale and depending on the algorithm of formula. So this way we actually develop benthic biotic indices, particularly in marine environment, and this can be happen, and this has been already developed and adopted in the freshwater ecosystem as well. So I will just like to introduce some of the biotic indices which are being used in the freshwater ecosystem. So these are the very common benthic indices which are being used in the freshwater ecosystem. Let it be the lotic or littoral ecosystem, or lotic ecosystem and pond ecosystem. So these are the basic indices which are being commonly used in the environment, and they adopt the same process, same theory of the based on the behavior and ecological and physiological responses of macrobenthic community. So once we see these kind of organism, then we define their affinity and also define their indicator values based on their response. Either they are sensitive, tolerant, or highly tolerant, or like giving some based on this tolerance level, we actually derive scores of the indices, and based on that scoring of indices, we give the qualitative analysis, a qualitative scoring of particular habitat and particular samples. So, for example, we are getting a very high abundance and high good diversity of freshwater benthic community, then we are able to say based on the score, these are the good water quality. Similarly, if we consider the marine benthic biotic indices, then these are the very common marine benthic biotic indices applied into the almost globally, we can say from tropics to temperate region and polar region, and very common indices. And in reality, these indices are not very site specific but universal. Instead of being universal, also there are lots of constraints which act upon the local affinities and local scenario. So that we will discuss later. So these are the common indices, marine benthic biotic indices being used. So these are the organisms we can assume they are some sensitive and indifferent tolerant. So basically we have divided these organisms into 5 different categories based on their ecological responses. So under the scenario of disturbances, in particular organic matter enrichment, how these organisms behave, and regarding to their adaptation with the biological life cycle and their abundance, as well as their strategy for their larval disbursement and larval dispersal, as well as like growth and growth in their abundance, as well as maintaining the particular biodiversity. So this way they actually adopt particular disturbed condition. If sensitive species represent high benthic quality, and most of the amphipods and some groups of polychaetes like organate and telenids belongs to that sensitive species. Coming to this group, that is capitalid group, which is very common biological indicator of high organic enrichment, and this is indicator of bad benthic quality and hypoxia and anoxia condition of the benthic environment. So, so basically, this from this graph we can understood that how benthic community actually behave under the stress gradient or disturbance gradient. So once we move from left to right direction, then there is an increase in stress, and so this way, like if normal condition, then we assume that species richness and biomass will be high and abundance will be low. But in intermediate level of disturbances, we can see this kind of model responses of benthic organism. So species richness though high because of intermediate disturbances, letting all the mixture of all the species come together and like optimize the particular condition. But their biomass increases. But this is the case, and this is a scenario they differ, particularly in the stressed environment, where we can see high abundance become very high, and this abundance contribution is maybe particularly for one particular species. If they become very tolerant and first order and second order opportunistic, so they become very highly abundant and optimize the particular situation and condition, leading the exclusion of other organism, other species, as well as their biomasses also reduces because of their high abundance, and mostly their juvenile and life form strategy changes accordingly. So these are the model actually which works on that theoretical and algorithm and formulation of developing biotic indices, in particularly marine environment. So these are the some literature survey we can see here. So globally, that Borja was the pioneer among all who has made lots of contribution in developing these indices, particularly MY and Bentix MY and Bentix indices. In Indian water, most of the authors they have simply used already developed biotic indices in the Indian scenario, and most of the study objective was like how much and how far these indices are being can be adopted into Indian scenario or Indian marine environment, and by developing by assessing the ecological status. So, but most of these indices are being also applied in the interdiated zone, and including my work also. So, if we see here in the next slide, this slide, if you can see, I have taken it from one of my publication, and it is publication
Sir,
21.
sorry to
Yes.
interrupt you, sir, but your slides are not changing.
Okay.
Yes, sir.
Can you see now? Is it
No,
changing?
no, sir. Still not changing.
This is the first one only.
Sir, click to the slides to option.
Can you change it now?
No.
Same?
Yes.
Can you see any changes? Different slides?
No, sir. Sir, there is a slideshow option in the.
Yeah. Tell me.
Now it is moving.
Can you see?
Yes, it can move. Try to make it full screen.
Okay. Now it's okay. Anybody?
Yes, sir.
So yeah. So
Just a minute, sir. Salesha, can you please turn off your mic? Yeah. Yes, sir. You can continue, sir.
Okay, but I cannot change my slide from here. Should I do it again? Now it's changing. Can now?
Sir, there's an option in the upper side, slideshow option written in the orange color.
Slideshow. No, it cannot help me. It's not helping at all.
So there is an icon just above the home tab. Is it
Yes.
visible like a TV icon? Click on it, sir. It will.
Yeah.
Still not changing the slide. So you can do one thing.
This call is open to anyone. Is it that orange color you're talking?
Yes, sir.
Okay.
There is an icon like TV icon. From अपर साइड में होम टैब के जस्ट ऊपर में से एक टीवी आइकन बना हुआ उस पर क्लिक करेंगे तो वो स्लाइड शो खुल जाएगा सर स्लाइड शो होने लगेगा
Not helping at all.
Okay, sir, no problem, sir. You can continue with your speech, sir. No problem.
Okay,
sir.
Okay, fine. So, okay, do one thing. Should I forward my slides to your WhatsApp?
Yes, sir. No problem. You can send me, sir.
Yeah, kindly get that. I have shared with 630 number that kindly get that.
So is the last number 630? Give me a minute, sir. That
you got?
Sir, it's Rajeshwari's number. I think he is not present in the meeting. He's
okay. Then I will share on the common number. Okay.
So you will share in the team webinar team, WhatsApp group of webinar team. Yeah, now can you get? So you have sent in WhatsApp, right?
Yeah, I have sent on WhatsApp number.
So in my number?
I have sent on both the numbers.
No, sir, I haven't got it. Give me a minute, sir, okay?
Just check with that common number 17 September webinar team, that group also you can check.
So I didn't got a presentation in that group.
What we have to do? That is taking time.
I think you can continue with your speech. No problem, sir.
So this in this slide we have given one relationship between the different indices because these indices are complementary to each other. So what was the maximum agreement between the two indices? So this way we can adopt particular indices which can be adopted or universalized in Indian scenario. So we can see here this AMBI versus BOPA was showing very high agreement between on the classifying the sample into same categories. So either good or bad categories. Similarly, if we see then about around 63% agreement was found between the AMBI and AMBI indices. So this way in one of my paper, we were able to identify the very potential indices which can be adopted and which were performing well into the Indian scenario. Next, we also adopted in the next paper. I just tried to find out the how these indices behave with different seasonality in particularly tropical conditions. So in this slide, you can see we have compared two abundance and biomass indices and compared two different scenario. So right hand scenario W1 W2, you can see these are the pristine and reference conditions, and right left hand scenario you can see P1 to P6 representing first year and second year 3 seasons. So P1 P2 P3 the pre-monsoon, monsoon and post-monsoon season. So here we can have comparative analysis and we can see the biomass was high across the location in all the season at reference conditions. And in the disturbed condition, that Phoenix Bay area, P1 stands for Phoenix Bay, in the Port Blair area, it was nearby the city, so that condition gave very bad and very low quality of benthic indices and benthic condition. And in this way, we see the high abundance in the red line. You can see very high abundance compared to the biomass. So this is the one way we can compare the abundance biomass ratio as well, and that indices was also suitable for our environment. Similarly, we have also applied other indices like benthic opportunity, that BOPA indices, benthic index, S-time marine biotech index, and multivariate AMBI. And these all gave the seasonality pattern, and responses were very similar to those kind of appeared and observed in the temperate region. So in this, most of the conditions, once we consider the Vandu National Park, then all the indices they behave according to the seasonality. But in the Phoenix Bay area and Jellyguard Bay, you see lots of consistency in the bar graph. So it's showing like there was no differences within the seasonality was not observed actually in this particular area. But in natural condition, we can observe the seasonality well, and this was like mirrored by the indices as well. So here we see all the indices in Vandoor National Park shows some kind of seasonality. So in northeast monsoon and dry season, what we call post monsoon, and showing almost cyclic pattern for the first year and second year. So this way we were able to find the seasonality pattern in indices as well. So in my next last paper from my PhD work, it was published in environmental monitoring assessment. In this paper, we tried to find the any gradient defined by these indices. So here we applied into different environment. Previous study we applied into mud flat area. This study we applied into sandy beaches area, and including the reference condition as well. So here we can see these two. C stand for carbon cough, and A stand for Aberdeen Bay. So here we see these samples are coming together and showing one kind of disturbance, and this segregation of samples are separated from other sample group, showing some stressful environment. And this blue square, you can see it representing the pristine environment. And this was PCO was done to check the gradient analysis, and this was also superimposed with canonical analysis. So here we try to correlate with what kind of indices actually were more determining in separating the different samples. So this way we were also able to check the application of these indices into sedimentary habitats, in particularly sandy beaches and sand flat habitats. And this AMBI group was most robust, appeared most robust in tailing the particular habitat situation. So this was the final result of that agreement between the how many percentage of samples was categorized into 5 different classes. So that classes was based on bad, poor, moderate, good, and high quality. And this qualitative classification was based on AMBI scorings and BOPA and all those indices scorings. So, in the last, meanwhile, in last we checked in what was the species actually showed highest correlation with the organic enrichment. So we found lots of species, they showed positive correlation, but high correlation was observed with Orbinia species, which are actually sensitive species for the European countries and for temperate countries. But in Indian scenario, this Albinia species become second order opportunist and tolerant in organic matter enrichment condition. And they were almost equivalent to Capitella singularis, which is very common and a global biological indicator and very renowned in the worldwide. Representing the organic enrichment, but species are different. That for temperate region, most of the high organic enrichment representative were Capitella capitata. But in our condition, in Indian scenario, we got the Capitella singularis, which was showing more similar behavior to the sibling of European countries. Similarly, after finding the correlation between the species and organic matter enrichment and percentage of organic matter content into the sediment, I also tried to find what was the response curve of the species against the organic matter. So here we can see the response curve in a black as well as in violet color. This representing the ecological group third, mostly by Orbinia species, and ecological group fifth by the Capitella singularis. And their responses are like a unimodal with increasing the concentration of the organic enrichment. And other species responses, you see the unimodal and Gaussian distribution as well, with the increasing the concentration of organic matter, their abundances decrease, but particular organic concentration, their abundance showed highest abundance response. So this paper actually being communicated in Marine Ecology Progress Series, and I am hopeful having positive aspects of getting acceptance. So beside applying those indices into Indian scenario, these are some very common we can say the constraint and drawback and some loopholes of applying these indices. So first the point to be noticed is different species have their differential responses, and it depends upon their geography and their hydrodynamics environment as well as their which sea they are living in. Similarly, most of the indices they have been developed in particular countries, and they have one kind of regional affinity, and their formulation have been formulated in one way to suit the particular environment. Similarly, once we applied those indices which are being developed and calibrated into temperate region, they mostly lack lots of calibration and intercalibration and boundary readjustment for the suiting the indices, those indices into Indian scenario. Fourth point is most of the indices they actually generalize the underneath disturbances. So most of the indices when they generalize, we actually lack the pinpoint what was the real cause and what was the real behavior or ecological responses of macrobenthic community, either through the change in abundance, biomass, or changing in their diversity pattern. And one of the most important drawback and fallback of these indices are they sometimes they fail to distinguish between the man-made and natural kind of disturbances, and this is very obvious in the Indian scenario because we consider the seasonality and tropical setup where natural disturbances are very prominent in the form of cyclone and storm and in the form of rainwater discharge. So this way they are not very much like suitable for the Indian scenario. But however, we can actually intercalibrate the data and validate these data sets to the reference conditions, and from there we can compare and contrast the result, and finally we will be able to say we can identify and characterize the very robust index, and it can be used to address this ecosystem health assessment and biodiversity degradation and ecosystem degradation, and we can actually address the real-time scenario and real-time requirement by the government agencies and various other agencies which are looking after the protection and sustainable development of the coastal ecosystem. So these are my presentations, and thank you for your time and listening, and thank you for giving this opportunity to represent my work. Rather, I represented macrobenthic community, which was a little bit greater size than the microorganism, particularly bacteria and viruses and some other fungi and other filamentous algae and all. So thank you very much. Thank you all.
Thank you, sir.
So I expect lots of questions because it would be there because it was kind of presentation of my research work, and it is not new actually. I will say applying marine biotech indices or benthic indices is not new at all. It has been, you can trace back it to the industrialization, and we have that particular Darwin theory of evolution, that moth color changes that indicate particular industrialization of particular period. So this way we have developed from that situation and comes to this point where we can assess our ecosystem, and we can present those data and into very concrete and concise form, so politician and lawmaker and legislature, they can understand that what is the real scenario, and they can be put into a point where they can take decisions regarding the coastal protection and biodiversity protection and conservation. Yeah.
Thank you, sir. Moving further, as we bask in the warmth of our guest presence, let us extend an invitation to another professor in our constellation of friends to shine and share their thoughts with us. I request Mubashara to introduce our esteemed resource person, Dr. Swati Soren ma'am.
A very good evening, ladies and gentlemen. It is a great pleasure that we introduce our esteemed resource person for today's webinar on the International Microorganism Day, the remarkable Dr. Swati Soren. Pursuing her love for the science, Swati ma'am went on to Karachi University to complete her PhD in toxicology and pharmacology. She also shared her expertise as she trained in electron microscope from AIMS. Currently, Swati ma'am serves as a HOD of Zoology Department of Jamshedpur Cooperative College, Jamshedpur. Please give a warm and cheerful welcome to Dr. Swati ma'am.
Thank you, Mubashra. Thank you so much. Very good evening to all of you first, and to all the delegates present here first. And thank you to Dr. Ajay Kumar Srivastav, sir, the head department of botany, St. Xavier's College, Ranchi, for inviting me as the resource person, and to all the uh, organizing committee as well, and as well as the Microbiologist Society and whole of the St. Xavier's College team and the botany department especially. Thank you so much for inviting me here. And shall I share my slide first?
Yes, ma'am.
Okay. And do tell me whether it's moving or not, huh?
Yes, ma'am.
So, is it seen?
Mm, yes, ma'am.
Is it visible now?
Yes, ma'am.
Completely?
Yes, ma'am. Completely visible.
Yes, ma'am. Completely. Okay. Now, first of all, I would like to actually hear from all of you, because many of the senior my delegates are here, and most of you are the students. I guess Preeti is there on that side, right?
Yes, ma'am.
Preeti,
Yes, ma'am.
are you UG student or you're PG?
Ma'am, UG student.
UG student. Most of you all are UG students. I can see that.
Yes,
I
ma'am.
have heard from Ajay sir also. But congratulating you all, because you have done so much a great organization team that makes up all these webinar and all the work which you have done. Congratulations for that first. Now, as
Thank you, ma'am.
we have been here since morning until today is the evening. Now it's the evening now. And for this International Microorganism Day, we have explored so much the microscopic marvels today. I hope so. And leading to my slides preparation means I am been in toxicology and pharmacology. My research was based on that. So leading towards the today's topic as International Microorganism Day. So international, it is basically very new to it. Means it has been being commemorating, or you can say it has been this day has been established by the Portuguese Society for Microbiology, and it is observed by UNESCO now. And it is why it has been being commemorated, or why this day is being celebrated. I think every student should know, and I hope you all must be knowing it, because most of the student, your organizing team as well, and the students who are participating here, and my seniors, my delegates obviously knows that. So it is being celebrated as 17th of September, and one could imagine the life before the microorganisms were discovered. First, I'll go with this, then I'm going to come with the this days why it is being celebrated. So before any disease or any illness was there, because no microorganism was discovered so far that time, means many people used to see that some evil or maybe some god spirits are there leading towards all these diseases or any illness. So I hope all the students here, I hope, because all the scientists and all the senior delegates obviously knows this is for the students exactly. For obviously you must be knowing the scientist's name. Any guesses? Anybody? Still, I'm saying.
Anthony Van
Any
Leeuwenhoek.
guesses?
Anthony
Yes,
Van
obviously.
Leeuwenhoek.
Yes, and turn even hawk. It is because we need to. We are actually celebrating this day because since 2017, and this day has been chosen to celebrate it as he sent a letter to the Royal Society of London in 1683. So this was the first description what he gave, that some single-celled organism or animalcules were there in his mouth, as he observed. So this was there, and even the microscope was discovered. And without the discovery of the microscope, we would have not been so far with the any of the research. So we all know now. So far, since morning, we are here to commemorate the International Microorganism Day. So far we now know many of the microorganism like names, and I'm not going so far, and I'm not going to go further in a very descriptive manner today, but very in short I will say that without having a microscope, these unicellular or any multicellular or any cell clusters of microorganism would have not been discovered. So some major six types I have mentioned here as bacteria, archaea, fungi, protozoa, algae, viruses are there. And obviously the microorganisms have been played, they have been given, they are very essential for life exactly. So some of the functions which they are leading these days, well, not these days, means so far, is decomposition, oxygen production, nitrogen fixation, biomass control, digestion, food production. I'm not going very deeply because I'm running shortage of time. I'm having another meeting by 8 o'clock. That's why. Uh, the types of microorganism you have all seen since the morning it is going on. That's why I'm not going in each one of it or some of it exactly. So I'll be dealing with today as microbial bioremediation as well. So we know that the microbial or microorganisms are very useful. Um, it's a breakthrough only towards the world, for the world we should say, that it is breaking down the contaminants. है And you can see this slide, how it is using the contaminants, and it is using as energy source or cometabolize it with them. So cometabolism is basically, it's a process by which uh, one can say that some contaminants, or it is being degraded by any of the enzyme or cofactor which is produced by during microbial metabolism. So bioremediation can take several months to even several years to complete. But here, in case of microorganism, these basically microorganisms are used more often than the animals are being used more than the plants for bioremediation, because they can grow easily and quickly, and they are being manipulated. So this is the slide, next one I'll go with. Again, the bioremediation means, this is again, I will say that it's a process of biological organism, microorganism, to break down hazardous materials into less toxic or even the non-toxic products. Even with the bioremediation process, it is converting into water as well. So it's due to this example of bioremediation, contaminated soil, oil spill cleanup, and crime scene, even this cleaning up agent or a kind of cleaning agent as well. So that is giving, it is serving the society as well, the microorganism, but not all are harmful. That is why. So bioremediation process involves many different microbes acting, which is parallelly, or you can say in the in situ or in the ex situ condition as well. So next one, I will go with this bioremediation, microorganism bioremediation. In this one, I'm mentioning Pseudomonas putida. So this concept of environmental remediation was, firstly, it was patented in 1981. So degradation of petroleum oil by this Pseudomonas putida. Such a wonderful thing which has been happened, means without the researchers, without the scientists, it would have not happened like that time. And the other one, you can see the first microorganism which is genetically engineered for bioremediation processes. It is Pseudomonas fluorescens, HK44, named as such. So another, as the International Microorganism Day the theme which you have taken up as like microscopic marvels that shape our world. So they are shaping our microenvironments first and then to the world, because they are habiting our microhabitats, and how they are um, within you can see means many of the microbes on the earth, they are living throughout the earth, means they are a habitat to it, and how they are useful for every livings. And talking about the microenvironment, essentially the entire world is composed of the microenvironment, and within this environment, microenvironment, we can see the right side, you can see the diagram, the photo which I have taken on is halitosis, that is the bad breath. So relating means one and then, or in any every second, any of the human or any organism is living with the micro environment only. And that is the fact that we even the water in the lake or even the ocean, these are also composed of microenvironments. And such factors which is having the environment which is being given to them, the favorable condition which is growing with the microenvironments is light, nutrients, temperature, and with the microhabitats that determine its health, and halitosis as well, means it is the present in the mouth as well, in your stomach as well, inside the body as well, means everywhere it is there. So why we are so much we are into we need to study about so much of microorganism, because one can say that it is serving the ecosystem. Microorganism is serving as the ecosystem. It's they are, in another word, as written, seen in the slide which I'm sharing right now, seen as suppliers of so-called ecosystem services. So these are serving the ecosystem. And fundamentally, if you talk about the human life and activity, these microorganisms are so much involved in our day-to-day life. Next, I'm not going to go further with the microenvironment, and because in the talks of earlier resource persons, I've already mentioned many of the like relating it to biotechnology. I'm not going to repeat all these, but some slides you can see here. One of is biocar, or as you pronounce it, I call it as biocar in agricultural soil. So it affects means due to this biocar, means soil's microorganism and microfauna, means it is fertile. The soil is getting fertile, and due to that, if the soil is being fertile with that, due to the microorganism present there. And later, the another example is shown here as arctic bacteria, whose physiology is adapted to very cold temperatures. So they have therefore certain enzymes, they have it, and potential technical or industrial utility. So for food processing and in washing powders, and that efficiently work as energy saving low washing temperatures. Another one, again, indicators are there. Some means microorganism to microorganism, they are interacting. From microorganism to plant, they are interacting with that. So the whole world is being used. The whole microorganism level is being used. I mean, before my senior delegates and have already mentioned that some of them are only harmful, some of them are pathogenic, but not all, because so much. They are being used for in the pharmaceutical areas like antimicrobial, antiviral, antioxidant, anti-cancer. So these are leading technologies being used for preparing all these. Later, I'll in my this slide, I'll show you like I have read this somewhere in my earlier paper somewhere. Microorganism can create electricity from wastewater. So this was the scientist. First, I'll show the scientist's name. This is Artemis Bogosian of EPFL. He stated as we have engineered E. coli bacteria, the most extensively studied microbe to generate electricity. So his paper was published in June. You can see on the right side it's mentioning, and I have already mentioned the from where I got this article. It has been already published in the paper, this journal June. And this is EPFL. The researchers at EPFL is École Polytechnique Fédérale de Lausanne. They have generated E. coli bacteria to generate the electricity. How wonderful it is to know, na? One could obviously think about it, how they have generated the electricity. So how wonderful is the technology being they have taken us. Now E. coli, they have increased the ability to generate energy through extracellular electron transfer. So this is which allows them to metabolize a wide range of organic substrates. So the researchers have developed a complete EET pathway within the bacteria, resulting in the threefold increase in electrical cure. So how interesting to know about microbes and E. coli, especially because it is being widely used for means major of the studies. And this paper obviously gives a revolutionary strategy, or you can say it has brought about a kind of revolution for the both waste management and the energy production. Isn't that? I hope you will agree with me. Am I audible? I can't
Yes,
see
ma'am, you are.
actually you all. Am I audible?
Yes, ma'am.
Yes, you are.
Okay. Okay. Thank you. So E. coli, yeah, because it is a very commonly used in biological research, and through the mechanism known as the extracellular electron transport, they have generated the electricity, and so now it is resulting in highly efficient. We call it as electric microbes now. So it's really very interesting to know all these. Now we have already known now there are list of roles microbes play in our lives, but I'm not going further more because we have known these. So because many scientists and many of my delegates, senior delegates, had already mentioned all these. So some of them, like it maintains the soil fertility and soil tilth. Everybody knows this now. I'm not going to further go with this. Some more I wanted to show you, like first of all, we whatever students, I really wanted to mention one more thing. Now, whatever we study in my syllabus, or whether you have it in your syllabus as microbiology, so or any of the topic or any of the unit. Right now we are in concerned with the today's topic, that is microbiology. That is only I'm dealing with the microbiology right now. So we always you should have that outcome of the subject. So whatever the subject you are dealing with, they does have some outcome, so that we can have a means kind of job oriented, you can call it. So there are many career opportunities related to it. That only I wanted to deal, means some relating. I wanted to tell you all today about, from my part, means the microbiology is, the microorganisms are so much useful. Some are harmful, but not all. And as for career is concerned, so you can go with the microbiologist, you can go with the food technologist, you can go with pharmacologist, you can go with the biotechnologist, and the other one, you can immunologist, water quality scientist, medical technologist. So many opportunities you have throughout, means after getting into these fields of microbiology, and including, you can see on the slide which I'm showing right now, in the healthcare, forensic science, environmental organization, higher education, food and drink, and pharmaceuticals as well. There are varieties of it. Now in the last slide, you can see so many microbes are there on the plate, you can see here. But thank you to all of you for patience hearing, and thank you first to your microorganisms, so that we have togetherly, we have been here, I have been here for this wonderful session today. Thank you so much.
Thank you, ma'am. Moving further, in this symphony of voices, our guest melody has enchanted us. Now let's invite another professor to join the ensemble and add their unique harmony to our conversation. I request Komal to introduce Dr. M. Mahendra Kumar, sir.
Good evening, everyone. Ladies and gentlemen, it was great pleasure that we introduce our esteemed guest for today's webinar is Dr. M. Mahendra Kumar, sir. Sir is currently as an assistant professor in the Department of Biotechnology and also assistant coordinator of the internal quality assurance cells of Guru Nanak College Autonomous Velcheri, Chennai. He secured university rank and gold medal for his post graduation. He had cleared Tamil Nadu state eligibility test He had done a postgraduate diploma in intellectual property law from Tamil Nadu, Dr. Ambedkar Law University, Chennai. Completed his PhD from University of Madras. He has won Teaching Excellence Award by Lion Club International in 2022. NESA Young Scientist Award by National Environmental Science Year Academic New Delhi in 2021. Dr. APJ Abdul Kalam Award for Young Scientist in 2018 by Biologists from India. Dayawan Award by Kundar Club International in 2003. He has also been awarded by Best Paper Award and Best Poster Award. He published 20 research papers and presented more than 25 research papers in national and international conferences. He was author of 5 books, chapters in Springer's, Elizabeth Taylor's, and Francis and William Sons publisher. He also have a 5 patent to his credits, 1 German patent, 1 Australian patent, and 3 Indian patents. He is life member to many reputed scientific associations and societies. Please join us in giving a warm welcome to Dr. M. Mahendra Kumar, sir.
Thank you, Komal, for that very nice words of introduction. Am I audible?
Yes, sir, you are audible.
Thank you. So let me present my screen. Can someone tell me whether my screen is visible?
Yes, sir, it is visible.
Thank you very much. I thank St. Xavier's College Ranchi and Microbial Society India for giving me this opportunity to share my thoughts on intellectual property rights, or we can say innovations in life sciences and its protection. So I'm Dr. Mahendra Kumar from Guru Nanak College, Chennai. So before going to the presentation, I would like to throw light upon the basic difference between a discovery, invention, and innovation. Actually, this crux differentiates the entire intellectual property rights. So discovery is something which we discover or we come to know, knowingly or unknowingly, that is already existing in nature. Whereas an invention is we humans create to suit our knowledge, suit our needs. Everyone knows necessity is the mother of invention. So when we need something, we invent something. But the same invention, if it is having more than one purpose or some unique purpose or which somebody thinks cannot be done, and if you do it, it is called innovation. So intellectual property rights is a very, very, very valuable asset that results from the creation of human intellect in industrial, scientific, literary, and artistic field. And these intellectual property comes with exclusive rights called intellectual property rights that are granted to the creators or owners of the works that are resulting of their intellectual creativity. And the owner can use his intellectual property rights for commercial exploitation. And the significance of intellectual property is this avoids duplication of research. It encourages creation of many new intellectual goods. It helps industry to produce better and cheaper product by innovation. So in India, the intellectual property rights or intellectual properties are governed by the following laws: the Copyright Act 1957, the Patent Act 1970, Trademarks Act 1999, Geographical Indications Act 1999, Industrial Designs Act 2000, the Protection of Plant Varieties and Farmers' Rights Act 2001, and last but not the least, Biological Diversity Act 2002. I'm not going to tell you about all the intellectual property rights. Since we are celebrating International Microorganisms Day, I would like to stick on to the intellectual property rights that are associated with life sciences. First one is geographical indications. A geographical indication tag is granted to a product or goods that originate from a definite geographical territory, which is used to identify agricultural, natural, or manufactured goods having exclusive special characteristics from a particular region. So this GI confers a legal protection of specific goods of a region or of a state or of a region from unauthorized use of the same. GA also promotes economic prosperity to the producers of the goods in that particular geographical territory. So these are some of the geographical indications restricted to Tamil Nadu. We have such n number of GI tags across the country. Next is patent. A patent is an intellectual property right. Many of you know it is granted to an inventor by any country's government, usually for 20 years. And it is a territorial right, it is not an international right. So if you claim or if you are granted a patent in India, it is valid only in India and not in any other countries. In case you want your invention has scope in many countries, you have to apply individual countries application and you can go through PCT. This patent is the most important patent intellectual property rights among all the rights. It gives the owner exclusive monopoly to prevent others from making, using, importing, or selling the invention without permission. It is treated as a valuable property that can be inherited, gifted, sold, or licensed on agreements like any other property. In India, if you see the history of patents goes back to 1856 when India was under the British rule. The first whatever laws that were followed or that were implemented in the Britain was followed in all the colonies, including India. But the exclusive Indian Patent Designs Act was passed in the year 1911, even though when India was under the British. But this act was exclusively for India, and this was only for product and not for process. So post-independence, the revised and most suitable Indian Patent Act was enacted in the year 1970. After India joined the WTO TRIPS in 1995, the intellectual property turn got a different facelift, and we've got more intellectual property rights after that. So, as per the Patent Act 1970, the Section 3 and Section 4 clearly delineates what are all patentable inventions and what are all non-patentable inventions. So first we have to know, even when we are working with life sciences, when we are working with microorganisms or whatever it is, in case you want to apply a patent, first you have to think whether your work is novel, your invention is novel. Second step is it has an inventive step. Third one is it has industrial applicability. Fourth one is patentable subject matter. That is, even though it complies with all the above first three, if it falls under non-patentable inventions, it will not be granted patents. So what are non-patentable inventions? Non-patentable inventions or invention, regardless of satisfying the novelty, utility, and non-obviousness tests, if it falls within the criteria mentioned under Section 3 and 4 of the Indian Patent Act 1970, then they are called non-patentable inventions. First one is an invention that is frivolous or claims anything obviously contrary to the well-established fact, then it is not patentable. For example, you invent a machine which does not stop, or when you invent a machine which gives more than 100% efficiency, then it will not be granted patent. Number two, inventions that are contrary to law of mortality or injurious to public health. For example, the onco mouse or the GMO microorganisms, whatever you create for research will not be given patented if it is against the law of mortality or if it is injurious to public health. Section 3, mere discovery of scientific principle or formulation of an abstract theory or discovery of any living thing or non-living thing substance occurring in nature, it is not patentable. Section 3D, mere discovery of a new form of a known substance. You have a product, if it is already known, mere discovery of the new use or new different form will not carry any value, so it will not be granted patent. Section 3E, a substance obtained by mere admixture. That is, you just mix two compounds, it will result only in the aggregation of the properties of the two different components. Hence, it will not be considered as an invention and patent will not be given. Mere arrangement or rearrangement of known devices will not be given patent. Any method of agriculture or horticulture will not be given patent. Section 3I says any process for the medicinal, surgical, curative, prophylactic, or diagnostic, or therapeutic, or other treatment of human being will not be given patented. Section 3J, plants and animals in whole or any part thereof other than microorganisms will not be given patented. Even earlier, microorganisms were not given patented, but now microorganisms are granted patents. In section 3L says any literary, dramatic, musical, artistic, or any aesthetic creation whatsoever will not be given patented because the respective rights can be protected under Copyright Act 1957 and not under Patents Act. Section 3M, a mere scheme or rule of method of performing a mental act will not be given patented. Section 3N, any presentation of information in any manner or by method by spoken words, symbols cannot be covered under Patent Act. Section 3O, topography of ICs are not governed by Patent Act, so patent will not be given. Section 3P is if you involve traditional knowledge or which is an aggregation or duplication of known properties of traditionally known components, it will not be granted patent. Finally, section 4, it excludes any invention relating to atomic energy falling within section 21 of the Atomic Energy Act 1962 will not be given patented. So, in case you're working on any microorganisms or you're working on any research, if it falls under section 3 and section 4, it will not be granted patent. Fine. The first patent on a life form was in 1873 when Louis Pasteur was awarded the first patent on microorganism for a yeast he developed that are free from organic germs of a disease, and it was granted patent number 141072. The first patent on genetically modified or microorganisms, or we can say after this patent only we got patent for microorganisms. So this was granted to the Indo-American scientist Anand Mohan Chakrabarty for his invention Pseudomonas bacteria, which is very famously called superbug that clears up oil spills. In US, the first patent on plants was granted in the year 1931 to Henry Rosenberg for his invention of a climbing ever-blooming rose, quite this number. But in India, we don't give, we will not, the Indian government will not grant patent for plants and animals. So once you're clear with that, these are the stages of filing a patent till it grant. So first you file an application that could be either provisional or complete. What do you mean by provisional? Provisional is you have done almost 75% of the work and you are expecting certain results, but you're afraid of your competitors, or you want a control over that, then you file the provisional application. But in case your invention is complete, then you directly go for complete application. So from the date of publication, from the date of filing, as soon as you file and pay the fees, your invention will be granted an application number. So from the date of application, the process takes 18 months to get published. In case yours is a complete application and you don't want to waste time, you can apply with a condition called early publication. You may have to pay 4,000 rupees extra so that it will be processed very soon, very fast, and you need not wait for 18 months. Then once it is published, after 18 months usually, you request for examination, which will take another 30 months. That is from the date of application, it takes 48 months. And first examination report is issued, and which is issued for third-party representation. Anybody can object or anybody can come and support. So all objections of all kinds will be welcome for 12 months, and after that everything will be compiled and grant of patent will happen. You can either expect grant of patent or you can expect a rejection of the application. In case the patent is granted, the opposition party may go for an appeal. In case the patent application is rejected with the documents provided by the opponent parties, the patent owner or the patentee can go for an appeal to the appellate board, and the appellate board will thoroughly examine your application and listen to both the parties and they will pass the final orders, which could be a revocation of the patent, or could be amendment of the patent, or dismissal of the appeal. So now we'll come to the life sciences patents. Reference to such biological matters shall be made in specification with 3 months from the date of filing. You have to inform that the sources of your invention is a biological material. Further, the source and geographical origin of the biological source material should be disclosed, and you should have got approval obtained from Biodiversity Authority. There is a body called National Biodiversity Authority (NBA). You have to apply and get the approval before submitting the patent application, or within 3 months of that. Then the accession number of your submission. Then, in case you are working on DNA sequences, the DNA sequencing in the e-form shall have to be submitted. And the legal frameworks for life sciences patents across the world, there are very important agreements. The UPOV agreement, that is the International Convention on Protection of New Varieties of Plants, the Budapest Treaty on International Recognition of Deposit of Microorganisms, then Convention on Biological Diversity (CBD), and TRIPS favors the patent for life sciences. And the latest next is the Biological Diversity Act 2002. So this was enacted in 2002 with the main aim at the conservation of biological resources, managing its sustainable use, and enabling fair and equitable sharing benefits arising out of use and knowledge of biological resources with the local communities. After the implementation of this particular act, the local or the tribes were benefited to the maximum. So the Biological Diversity Act 2002 was born out of India's attempt to realize the objects enshrined in United Nations Convention on CBD. Next is varieties and farmers' rights. So in case earlier till 2001 it was not recognized, but in 2001, after this particular act was passed, it established an effective system for protection of plant varieties and the right of farmers and plant breeders to encourage develop new varieties of plants. And this made eligible for this, the diversity must be novel, distinct, possess essential characteristics under the particular act. And if a person has got this, he will be able to produce or reproduce the material, offer it for sale, sell the material, export the material, and to hold stock of the particular material. Finally, since many of you are science background, I would like to say a few at last 2 slides about Indian Patent Agent exam. This is an exam for which only science graduates and engineering graduates are qualified. This is a person who is qualified to prosecute patents will be called patent agent. That is, he can draft, file an application in the patent office. And given the fact that drafting a patent requires specific technical as well as legal knowledge, only a qualified person can do. And in India, according to Patent Act 1970, the Section 126 envisaged the qualification of becoming a patent agent. So you should be a citizen of India who have completed 21 years of age, should have obtained a science or engineering or technology degree from any university under the territory of India, and you should apply and pass in the qualifying Indian Patent Agent exam. And this exam has 2 papers: Patent Act and Rules, drafting and interpretation of patent specification, and finally the Viva. And once you clear this, you will be able to go practice before the Controller General. You will be able to prepare all documents, transact all business, and discharge such other duties for the patent prosecution. And once you do this, many of the science graduates lack this particular knowledge of patent legal process outsourcing (LPO), as the cost of preparing, filing, prosecuting, and maintaining patents have become extremely high in the Western world. It has resulted in mushrooming of several patent LPOs in India, and these LPOs provide us with huge global market to work with such very high salary. And these are the top institutes in India offering IP courses: Academy, Mumbai, Academy of Intellectual Property Studies, Mumbai, Nalsar University, Hyderabad, IGNO. Now we have MOOC courses, YM portal also. And this is the website of Indian Patent Office. Very interactive website, and it gives all the information. You can try. Thank you. In case anyone has any doubt, I'll be able to answer if the organizers permit.
Yes, sir. Is there anyone wanted to ask question from Mahendra, sir? I think no, sir.
Thank you. Thank you very much for the opportunity, organizers.
Moving further, the spotlight has graced our guest today. Now it seeks a new professor, whose words shall dance in the limit light. I request Komal to introduce our knowledgeable resource person, Dr. Kamlesh Jangid, sir.
Now our next resource person is Kamlesh Jangit sir. Sir is currently as a scientist with Bioenergy Division, Agharkar Research Institute, Government of India. He is also appointed as head of bioinformatics and manager of high media laboratories. Also scientist D of National Center for Microbial Resources and Microbial Culture Collection. He is also been visiting scientist University of Toronto, Mississauga, Canada, assistant research scientist and postdoctoral associated University of Georgia, USA. He is a president of Berkus International Society for Microbial Systematics. Appointment as academic advisor, Beijing Key Laboratory of Biodiversity, Organic Farming, China Agriculture University, China. During his postdoctoral research, he awarded as a Best Poster Award by Bergis International Society for Microbial Systematic Beijing, China. Postdoctoral Research Award nomination UGA. He is listed in Marcus Who's Who in America, USA. During his academic journey, he secured first rank in BSc Microbiology, University of Pune, second rank in MSc Microbiology, University of Pune. He completed his PhD Microbiology from National Center for Cell Science, University of Pune, India. He has also been secured first rank in Olympiad at regional level in University of Pune Chemical Societies. He has published 60 research papers with his research groups and students, some of them reasons for oil spilling pollution, generation of antibiotic resistance bacteria along the coastal line of the Arabian Sea, Bay of Bengal, and the Indian Ocean environmental development. Robust detection of cow and female buffalo DNA through real-time PCRS forensic science international genetic supplement series, biological wastewater treatment and resource recovery. Alternative therapeutics against antimicrobial resistant pathogens, microbial community during development of microbial health and metagenomic soil ecosystem. He is invited to talk as in many international conferences like Investigating Soil Microbial Community Dynamic 2010, Advanced Microbial Solution LLC Dallas USA, Understanding Long-Term Microbial Community Development Using Capillary Electrophoresis 2020, Asia Pacific Japan Virtual Genetic Solution Text Thermo Fisher Scientific. He is also invited in many national conferences. The world's biggest collection of microorganisms in 2020 Indo Science Education Trust Virtual Lecture Series Pune and many more. He had presented various papers and posters in national and international conferences. He has been a mentor and taught many courses across his career, started from 1999 to 2021. Many PhD courses, undergraduate courses, master courses, and partial courses taught as a guest lecturer, advising and mentoring various students for research field. He is also a member of various professional societies. Please join us giving a warm welcome of Kamlesh Jangit sir.
Thank you to Moria and the organizers for inviting me for this session today. I would like to take this opportunity to present something that is very dear to my heart. At the core of the microbiology, I'm basically a classical microbiologist. Over the years, I've started to move towards molecular stuff, and being the president of the Burgess International Society for Microbial Systematics, I thought it would be apt to share some of the recent developments in the classification and nomenclature of archaea and bacteria. So, with that,
a brief introduction about the Burgess Trust. We all, as microbiologists, we are aware that the Burgess Manual of Systematics of Bacteria acts as sort of an encyclopedia of what all microorganisms exist on the planet. And the Burgess Trust was established in 1936 with the vision to provide a resource for all microbiologists across the world to offer this unique encyclopedia. So the Burgess Trust decided to start a society in 2006, which finally came into fruition in 2011. And the society is called the Burgess International Society for Microbial Systematics. The website is bismis.net. And we also have a YouTube channel which can be searched with @bismis _. So if you can, if you would like, you can search. So the goal of Bismis is to promote, research in microbial systematics as well as act as a global communicator of microbiology and advocate for research efforts in the field of microbial systematics and diversity. Some of the current officers of Bismis since August 2020 include myself as the president of the society. I feel very proud in sharing that in the past, roughly 90 years of the history of the Burgess Trust, I'm the first Indian who's representing the Burgess Society and the first president of the society. Professor Wenjun Lee is the secretary of the society, and Professor William Whitman, who also happens to be my postdoctoral advisor and also the chief editor for the Burgess Manual, is the treasurer of the society. So it's a small group of microbial systematics that actually work towards developing this, branch of science and promoting this branch of science. We, as part of our outreach activities, we conduct a series called Bismis Live, which happens on the third Saturday of every month. And just yesterday, we had our 28th session. So for new participants, if you would like to register for this, meeting, I would suggest that you use this barcode, QR code. You can scan this and you can visit this website to register once and for all the sessions. These are free of cost, and you don't have to pay any money about registering. The advantage is you get to interact with the best in microbial systematics. Now, that was about the Bismis Society outreach activities. Moving back to the science, there are three integral parts of taxonomy, we know. And the three integral parts are classification, nomenclature, and identification. Identification is something that requires a lot of elaborate lectures, and a lot of details are there. So I'm not going to cover this at this moment, but I would like to point out the major developments in the field of classification and nomenclature of microorganisms. So prokaryotic classification, as we know, you'll be surprised that there is no official classification of prokaryotes that exists in microbiology. What we know as the Burgess Manual is not an official classification. The goal was not to be the authority for systematics, but to serve as a vehicle for the improvement of bacterial classification. So the manual was a work in progress, and it is still being developed by people who work in the field. As you can see, this is the first Burgess Manual of Determinative Bacteriology, which was published in 1923. And today we are in 2023, so we are actually celebrating the 100th year of the first edition of the Burgess Manual. With the publication of the Burgess Manual, obviously there are a lot of developments in the field. And as you can see, the number of prokaryotic species described per year since 1900, you can see that with the introduction of the 97% 16S ribosomal RNA gene sequence similarity threshold, the number of sequences that started to get described every year jumped up suddenly. So we were expecting anywhere around 600 to 700 species per year. And this was quite evident that the delay in publication actually led that the second edition of the Burgess Manual of Systematic Bacteriology The volume 1 was started publishing in 2001, but it could only be completed by 2012. So it took almost 11 years to publish this 5-volume second edition. And what it led to a huge dearth of information in terms of latest developments. Over these 12 years, presuming that there were about 700 microorganisms being described every year, you're talking at roughly 7,700 organisms in across those 11 years, which were not part of the last volume. So Burgess wanted to, come up with something that allows to cover up this, and that actually I call this the dynamic taxonomy. That means there are a lot of updates and additions that keep happening. To provide up-to-date description, the Burgess Manual Trust decided about a digital edition, and the digital edition that came up is called the Burgess Manual of Systematics of Archaea and Bacteria. It is no longer called at the Burgess Manual of Systematics of or Burgess Manual of Systematic Bacteriology. It is actually called as Systematics of Archaea and Bacteria, because now we are into the domains of life that we understand as archaea, bacteria, and eukarya. This is the website by which you can visit. The manual is online. The chapters are updated as and when there is a new update that happens. So you can look at this manual. Since 2015 it is available, and you can become more familiar with what's happening in the field. Now, unlike prokaryotic classification, as I said, prokaryotic nomenclature has also undergone major shifts. And unlike classification, where there is no official classification for prokaryotes, there is an official nomenclature of prokaryotes, which is a very key difference between the two. The nomenclatural field is determined by rules and regulations, which are decided by the rules of nomenclature, and a committee that oversees the implementation of all of these rules is the one that's responsible for implementation of this. So names are given to prokaryotes are regulated by the International Code of Nomenclature of Bacteria, which was originally called as the Bacteriological Code, and its successor is called the International Code of Nomenclature of Prokaryotes. Now, the body overseeing the nomenclature of prokaryotes is called the ICSP, the International Committee on Systematics of Prokaryotes, which was originally called as the International Committee on Systematic Bacteriology. So that's the key difference, that the code is called the ICNP and the committee is called as the ICSP. Now, very recently, just about in the month of May, the International Code of Nomenclature of Prokaryotes, or the Prokaryotic Code, was revised in 2022 and published in the issue of International Journal of Systematic and Evolutionary Microbiology in May 2023. This is the website. You can go over and you can read how to nomenclate or how to give names to organisms. Now, there is a key factor with the ICNP that is prohibitory in the sense for microorganisms. And I'm going to share this. There is something called as a type in microbiology. We call one particular strain number as a type genus or a type species of a particular genera or a particular family. Now, if you look at the International Code of Nomenclature of Prokaryotes, which was released in 1966, the code says the type of a species of subspecies is preferably a designated type strain, or in special cases, it may be a description, a preserved specimen, or preparation, or an illustration. So that means the code was allowing the accommodation of organisms that could not be grown in the lab. Unfortunately, the ICNP in its 2008 revision came up with this new definition of a type strain, which has to be a viable culture and it has to be deposited in at least 2 publicly accessible culture collections in different countries. Now, this is restrictive. We all know that roughly 95 to 99% of microorganisms cannot be grown in the lab, at least with the latest technologies that are available in the lab today. So, how to give names to these organisms? So, that's a problem. And that problem is only a problem of the prokaryotic system. In the nomenclatural type for the zoological code or the botanical code, a type is an element to which a name of a taxon is permanently attached. We have never been able to see live dinosaurs, but we are still able to name them. We've never been able to see organisms that have fossilized, but we are still able to name them because of the freedom that is imparted by the zoological or the botanical code. But that's not possible with the prokaryotic code. The problem is most prokaryotes have not been brought into pure culture. Most prokaryotes are fastidious and cannot be maintained in culture collections, as we all know. So, for example, this Nitrosopumilus maritimus, it was isolated in 2005 for the first time. Its genome was sequenced in 2010, but it was only available or grown in the lab in 2017. So it took almost 12 years for these researchers to grow this organism in the lab. And once this organism was grown in the lab, the researchers had to wait almost 12 years to give it a name, which was validly published. To circumvent all of these problems, and then there are a couple of other issues. As you can see, some prokaryotes cannot be transferred between countries, especially countries like India, South Africa, or Brazil. What has happened is because of the National Biodiversity Act and the Nagoya Protocol, the NBA does not allow deposition of cultures to outside culture collections for the purpose of description of novel taxa. And because of this, we cannot satisfy the requirement of the ICNP rule number 30, which stipulates that to describe a novel organism, you have to deposit it in 2 culture collections, which are not possible now. So how to circumvent all of this issue? So essentially, we are giving a secondary treatment to the uncultivated microbial majority, and also we are creating hindrances to depositing organisms in outside culture collections for research purposes. I'm not talking about commercial activities. Now, to circumvent this problem, Professor Whitman came up with this idea that why not we give genome sequences as the type material for taxonomic description of prokaryotes. This was published in 2015. And then he discussed at length about the utility of strains, that means pure cultures, versus genome sequences as nomenclature types for species. If you look at the unambiguous reference to type species, if you have a strain, it gives you an unambiguous identification. But if you also have a genome sequence which is completely determined from an organism, the genome of a high quality can give you a 100% credential to that particular genome sequence being from that organism. So the quality of usage or the usability of a strain as against a genome is high in this reference as to act as a reference for the identity of a species. In terms of experimental value, obviously a pure culture has an high experimental value, but the genome itself does not fall far behind. It gives a moderate enough confidence of identifying an organism. And the ability to scale to all taxa, that means if you have one strain of a particular genus or one species of a particular genera, you can very well identify what all biochemical characteristics the organism may be having. Similarly, the archival permanence through time, for the culture, you have to grow them. They may be lost by culture collections. They may get impure. Whereas a genome sequence of a high quality, once deposited, it acts as a digital signature. And that's why it acts as a foolproof archival unit of an organism. The accessibility of an organism is moderate with a pure culture because of their cost. One has to pay roughly 150 to 500 US dollars to ATCC to procure a culture. Within India, you can procure Indian cultures. But if an Indian culture is to be accessed by a foreigner who is not of Indian origin, it is not possible today to access that culture without paying fees and without getting necessary approvals from NBA. And that becomes restrictive for free exchange for research purpose. Obviously, the interoperability between strains and genomes is low as against high for genomes. So with all these advantages, it was genome sequences could act as type material for bacterial and archaeal taxa. Unfortunately, the ICNP voted down this proposal, and to circumvent this, a seq code was created, which is a nomenclatural code for prokaryotes described from sequence data. So today, we have two common codes of nomenclature for archaea and bacteria. So the seq code is a code of nomenclature where the type of a species is a genome sequence. Obviously, it has to be determined with high quality. This is the seq code committee, and the funding is provided by the International Society for Microbial Ecology, National Science Foundation, the National Institute of Health, and this is the website for seq code, seqco.de. You can visit this website to get more details about this code, how this works. This is the seq code registry. Basically, it tells you how to submit a name, how to get a name, how to validate a name, how to submit a genome sequence to the database, how to validate the genome sequence, and eventually how to get a name validated to be used in research articles. So this is the entire flowgram that you can see here. The beauty about this is not all microbiologists are familiar with the Latin English that forms the basis of the International Code of Nomenclature of Prokaryotes, or the ICNP. Whereas this being an online system, it has built-in 52 automated checks which check for uniqueness across multiple codes, whether it is zoological, botanical, prokaryotic code. So when you put in a name, it will cross-check whether that name already exists in another nomenclatural code, and if it doesn't, it gives you the concept. Similarity to other names, length and pronunciability, grammatical consistency, rank coordinated suffixes, and the presence of a time material in any other culture collection. This is the website. You can go to seq code registry and you can check all the things. So today what we have is we have two parallel codes. And because of these two parallel codes, there is a lot of comparison between the two codes. As you can see here, the ICNP has a viable culture as a type material, whereas seq code has a DNA sequence. The number of taxonomic ranks above genus are 9 in ICNP, whereas in seq code there are only 4 taxonomic groups which are above the genus. Type for a taxonomic rank above genus is the genus except the class, which is the order. And for a seq code, the genus is the type material for all higher taxa. It doesn't matter. So this has been effective from January 1, 2022. Now with these two codes coexisting, where do we go from here? So the cultured and uncultured taxa to be brought under a unified framework and foster the development of synergies between the microbial disciplines of molecular biology, biochemistry, physiology, systematics, environmental science, and ecology. So the best possible scenario is to merge the two codes, bring them under a unified framework so that microbial taxonomists, ecologists, and every other person who's working in the field of microbiology do not have to get confused as to which code to use. Burgess Manual Trust, the Burgess International Society for Microbial Systematics, Lippincott, Springer, and Wiley, who have been publishing the Burgess Manual, Agarkar Research Institute, DST, and thank you for paying attention. And you can visit my lab website, microbial-lab.org, for more details. You can follow me on Twitter at @JangidK_. Thank you so much. I'll be happy to take any questions if there are.
Is there any question anyone wanted to ask from Kamlesh sir?
All right. Thank you so much.
Thank you, sir. Now, moving further, as our guest wisdom illuminates our gathering, let us kindle the torch of dialogue by inviting another speaker to share their insights and enrich our conversation. I request Mubashira to introduce Ms. Raghavi R. Ma'am.
Good evening.
Good evening.
Yeah. I'm very glad and thank you for this wonderful opportunity bestowed on me. I thank Sri Ajay Srivastava sir, Head Department Botany, St. Xavier College, Ranchi, and before going to the session, I just want to tell all something. I think Shali is a student, or is she is available here? Miss Shali Shahe. So, so she was the one who came and approached me. Basically, I am from Tamil Nadu. She came and approached me, and I am from humanities department. So I was pondering what should do. So she showed me a way like you can do it on recent trends in life science. So I also thank her. I think she's not available right now. Thank you for giving this wonderful opportunity. So thank
Ma'am,
you for this.
Ma'am Shali is here.
Yeah. Thank you, Shali, for wonderful opportunity. Even I got a chance to read science. So, okay, we will quickly move on to the session. We will be seeing the recent trends in life science pertaining to humanities. So, kindly let me know, is it visible? Is it visible?
No, ma'am. No, ma'am.
Okay, 1 second. I just something. Yeah, I think I have some internet issues over here. For no issues, I will continue as usual. So, coming to the recent life trends in life science. So, before going into this, I would like to start how the life science has been evolved throughout the years in India. So we all know many of the microbiologists, scientists, and our senior colleagues and senior have been wandering over that for the whole day. So we know Charles Darwin and many other scientists. So when we go research the evolution of biological thought in India, it starts from uh, Vedic age, that is before Christ, Vedic age, the very civilization that was formed in the borders of Himalayas. So where the first ever uh, life science has been evolved under the names of Puranas and other literatures. So the first ever biological thought was formed in the period of Vedic age in India. Later, it has been spread to other regions. So, what are the evidences for this evolution? Is the first ever evolution of life science is being traced from the famous Bhagavata Purana, which we all have in our house and we used to read to. So, where there is a part called the incarnation of Lord Vishnu, that is Dasa Avadara. So, the Dasa Avadara speaks the entire evolution of a living being, right from an aquatic mammal to a terrestrial mammal
and lastly to the human being. So this was the first ever evidence that has been found. Later, many other foreign scientists and researchers took this as a part, and they have been researched on this, and they have been came out with many beautiful thoughts, biological thoughts. So these are the things that should inspire the young budding scientists to ponder over them, ponder over such evolutions in our country, and take it out to the abroad. So secondly, many other regional evidences are also there. So one such regional evidence pertaining to the Dravidian culture is the Tiruvasagam. We we Tamil people used to take it as one of the major regional texts, that is Tiruvasagam, which was being written by Manika Vasagar. He is one of the devotees of Lord Shiva. So when you people came to Tamil Nadu, came to Tamil Nadu, you might have heard that. So Siva Puranam is one such thing where the poet used to write how a living being is being evolved through the years. It will start from a grass, budding a single cellular organism. Later, it transforms to a multiple cell organism. Later, it becomes human, and later he becomes devotee of Shiva. Something like that. And one more evidence, same in this Trivasagam, you can see in the chapter 2, Tiruvanda Pagudi, we say, which is all about how the living creatures evolve themselves. So here, in one part, he says how a fetus becomes a child, how her mother should uh, should feed or should take care to make it as a good human being, good and healthy human being. So such are the evidence we have in Indian texts, because presently even the government is being focusing and pondering on such researches. So these are some researches and some evidences we can trace, from where we could trace out the evolution of biological thought in India. Later, with the invasions and cultural disparities and diaspora and paradigm shift in the culture and the economy, economical and other traditions, these things have been faded away. It is not only in Tamil, we might have in Marathi, Odia, we have lot many, uh, what do you say, the evidences for tracing the evolution of life sciences, evolution of the biological thought. So later we have some scientists like Shushutra, who have written Shushutra Samhita, and presently we follow him for the plastic surgery, silicone surgery, and things like that. Even today, we follow his pattern for the surgery. Even for cesarean, for abnormal babies, we are using his techniques, Shushruta Samhita, for the today's medical science. We are using his points or his way of medication. We are following till today, and we have Nagarjuna, who wrote the Rasajnana Samhita. It is all about the medication, something which we now we are calling it as the allopathy medication, where we use some chemicals which are used for healing. So those chemicals has been already found by Nagarjuna, and he has written in his book called the Rasajnana Samhita. When you people have an opportunity, you can go for that. And lastly, we have Bharatwaja. He was also famous. Not only he is famous for aeronautical science, he is also famous for medication. We have Siddhas, Rishis, sages, many who have written many things. Without touching a patient, they could find his problem, his health issues. They were such such a perfectionist in medications. So we, as a budding scientist, should also follow their techniques so that we can expel in sciences. So we lot many things like that. So prominently, when we say life science, we should remember the names of Sushutra and Nagarjuna. And lately, we were also personified life science as Dhanvantri, something we have in our Indian culture, lot many things. So these are all about the evolution. So now we move on to the major part of the session, which is the yes, the recent trends in life science. So these were the evolution, and later we find many uh, evolutions and innovations and things like that. Now we are on to the 21st century, where we all see artificial intelligence that are being coming up in all the industries, not only in banking or finance sector. We have in all the industries we find artificial intelligence. Now, profoundly in medical science also we find this artificial intelligence, which has been coming up with many innovation and technologies. So when we talk of the So when we talk of innovations or the recent trends, what are available in our medical science or the life science as well, we find the role of artificial intelligence that has been coming drastically. It has been coming drastically. So this artificial intelligence is all about the recent technology that has been arrived. So recent trends that has been into the life science as the coming over years, we could have robots in our hospitals and things like that. So this, as India is being populous country with crores of populations and things like that, the definition the doctor patient ratio is drastically lesser than the other countries. So such is the case of India. So the role of artificial intelligence in India might be very helpful in improvising these ratios. We have in Chennai, some of the hospitals have this robotic medications where the robots being used for mild medications, not for the casualty or things like that. So this is being termed as the intelligent automation, where we use the what to say, yeah, artificial intelligence, robotics, and things like that. And the career in these sectors is being improving a larger. So India, the presently our government, union government has been given a policy called Make in India, Startup India. We have our Innovation Council, and MHRD has been organizing the Innovation Institution Council, where these things has been improving largely. Students, even school students, are being started to improvising these things. So such as the intelligent automatic diseases, which is even more communicable. So such diseases, the people cannot be taken, the patients cannot be moved out or taken back or something like that. So in such cases, we use what is called the virtual reality. We can, the patients is being scanned, not able to come to us directly. So in such period, we had this virtual reality medications that had really helped well for curing many of the thousands of COVID patients in Chennai itself. We've been seeing that. So thirdly, we are going to see what is called the blockchain technology, robotic process automation, and games that has been a game changer during the pandemic period. Until now it has been rapidly used for uh, the cancer patients and things, patients like that. So advanced digital platforms, they're being improvising by many IT sector that has been came with the medical sector as a partners. They have started improvising this, working on this virtual reality, augmented reality, mission and artificial intelligence, and things like that. So when we analyze it in economically, in economics, when we analyze it in 2021, we have the famous RE's Global Market Research Report. It yearly it's published. It's all regarding the medical research, and it contains all types of research pertaining to medical research. It shows that 83 percentage of respondent, that is patients, uh, feeling that the advanced technology, the role of advanced technology in medications is being very helpful in curing chronic diseases, providing proper treatments, timely treatments to the patients, and things like that. So 80% of the respondents has been satisfied, and the rest of the response feel that they're being feared of using this technology. Being recent trends, they've been fearing that this technology might be harmful for them, and this increases the incident of diseases. That is, the stress on diseases. The disease is almost stress, and using technology in curing or treating the patient makes them most stressful. They feel like that. This is one of the famous research report. When you get access for this, you could go for this research report. And secondly, what we're going to see is decentralized clinical trials. So we know when the period of the pandemic, for example, when the pandemic period started, some dates around March 10 or March 14, the first ever pandemic period phase of the pandemic started. The test, when we are going for a pandemic, the COVID test, the test or the clinical trial will be done only in Mumbai on the first 10 days of the COVID period. So later, due to the emergency and the seriousness or incidence of diseases being spreading out rapidly, we started our clinical trials in our regions itself. So this was the thing. This is called as the decentralized clinical trial. That is, the clinical trial places will be available in all the region. So clinical trial involves the variety of digital technology that enables the research to be conducted at various places on a face-to-face basis. This is what called clinical trials. So a number of contact research organizations is being set up right now. So this is all because of we should thank pandemic actually. This has increased this clinical trial into a larger extent, and as a result, the clinical trials is being set up in all the region. So in 2021 itself, we got 41% of clinical trials has been decentralized in India. So still now, many has to be decentralized. Still now, Indian government, for security reason, for some good reason, the government has been holding some of the clinical trials in their hands, and others has been decentralized. So major part of clinical decentralized has been decentralized in 2021 during the pandemic period. So now private partnerships has also been increased to 60% in the clinical trials. So due to the need and the dynamic expansion of the economy in India. So next trend which has been coming on the medical or the life science is the real world evidence. So what is real world evidence is? So accelerating the clinical trials. So it is not only that when the clinical trials are decentralized, it's everything over. It's all about we should know what's happening in reality. So in actual case, what is happening? We should know. We should analyze what's happening at the real time. So this is the only way. So when we have the real world evidence, what is happening in the medical science? What is the advancement in medical science? Is the clinical trial where is improving? Is it accelerating? It is reaching to the people at a timely pace. So these things will be covered under what is called the real life evidence. So as a result, we used to say real life evidence is the only way that will accelerate the clinical trial and support the regulatory decisions such as application of new indications or medical treatments or lab expansions, etc. So or any innovative drugs or innovative medications are improvising the medical. So only when a proper clinical trial you have, you could analyze or you could come to a conclusion that the given medication is a correct one or a proper one, is an apt one. So without clinical trials, we could not judge the given medication is a proper for a patient, for any disease for the matter. So, and this real world evidence is very vital for supporting the development of clinical trial design, as I told, and the observational research. And observational research for the progression of treatment, as I already told. That is, with only implementation of clinical trial will not tell you or will not let us know how the medication is working, whether the people are reaching or people are able to avail the medications. So it is the real world evidence which will bridge the gap between the clinical trial as well as the patients. So again, here the pharmaceutical companies are playing a major role or a pragmatic or collaborative approach with the uh, medical sector. So through the platform of data, knowledge, and other strategies. So the larger IT companies, something like your Tata consultancy, and you have Deloitte, something. These companies play a business to business strategies in bringing out the real world evidence. So how this real world evidence is being collected? This real world evidence is something that is data. Whenever a patient is being admitted or whenever a patient is being treated and whenever a patient is being discharged, the data is very necessary. So these data will be managed by some of the consultancy companies, that is information consultancy companies, and these data act as a real world evidence. We do research, scientists use this for research, the humanity people will do research for research, we have the vital statistics. So these data are very, very helpful for both scientists and the people. So here the pharmaceutical company, information technology sector is playing a larger role, very vital role in this framing or bringing out the real world evidence and bridging the gap between the medical sector and the society. So next trend, what we are seeing presently is new industry partnership. So as I already been telling about the real world evidence, where the IT companies and the business sectors is being playing business to business interface strategies. So these are all known as the new industry partnership. So it dates back to during the period of industrialization, liberalization, industrialization, that is in the 1950 to 60 period, where we witnessed many huge medical expenditures due to the war and epidemics. The medical sector was a narrow line. Only medical sectors is covered by the scientists, the doctors, and other prominent clinical people and other things like that. So today, as the economy and the society has become very dynamic, and even the epidemics has been also becoming dynamic. So in such cases, we require a dynamic medical sector, very effective and very sensitive medical sector, where we should have collaboration with not only with scientists and doctors, with all the peoples of the society, inclusive of the business houses and the shareholders and the finance, everything. So today, as we are in the towards the development path with the 75 years of independence, the medical sector has been witnessed many development and is been witnessing development each and every second. So we need investments, we need fund, we need technology, we need to database management system for research and development. So medical sector started having industrial partnership outside their industries, not as within the industries as well as outside the industries. Uh, we have collaborations with biotech companies, academic institutes or the education institutes, universities, and data providers, because government needs data. So based management system is very wide, should be very prominent and effective for a medical or a life science sector, because it is only data that will play vital role in framing the medical policies for any world economy. And this was the lesson we learned during the period of COVID-19. So presently, you can see in India, we have been increased the investment, medical investment from the collaboration with industrial partnership. The Indian investment on life science has been increased from 10 billion to 15 billion dollars as on 2021. So the companies like Pfizer and BioNTech with vaccines, they produced the COVID vaccines, and they've been forecasted as the top sellers. Uh, and you have the market. It's been forecasted that the Pfizer and BioNTech and Moderna will become the market leaders in medical science by 2026. So this is being forecasted by, again, global RS research. I already told this global RS research has been a prominent research. Their forecastings are being widely observed by many researchers and many people. So the next one is we are going to see what we are going to see is innovation in supply chain management with pertaining to life sciences. So as I already told, the industrial partnerships, as I already told, the medical science has been evolving, and it requires partnerships from not only from the medical science but also from all over the all the sectors of the any economy. So in such cases, the supply chain will also maybe might change it drastically, change drastically, because we are now witnessing a dynamic changes, tremendous changes, not only in medical sectors, in every sectors we are seeing the tremendous changes. So in such cases, we need a strong supply chain management. Not, as I already told, we need strong database management. We need a strong supply chain management too. So in such cases, when we see supply chain management, we are seeing it in three forms. We need three main steps in supply chain management. So the first train is reinforcing the value stream delivery and cost performance. So that is the cost of medication and the delivery of the quality service of medication should be analyzed. That is the cost benefit analysis. So the cost benefit, whatever we spend, we should, the benefit from the service should be equated. So that is the basic tendency of human behavior for anything, not only for medical services, it's for anything. And second step is searching affordable manufacturing condition. That is like supply of raw material. This is prominently this is used for production in medical science, medical production, medicine production. That is where we, because the medicine whatever we take is not directly, it does not comes directly, rather it is being produced in a stages of process of production. So in such cases, we should the producer will be aiming at the reduction of cost and increasing of profit. So in such cases, the producer, whoever producing services or goods and services for medical science, should be think the available or affordable manufacturer, so that the cost of the product will be reduced and the burden of the cost will not be borne by the consumer also. So the producer or the producer of medical goods and services should think in both the ways. So the cost of the medical product should be nominal, because the medicine or the medical things or the medical commodities and services are very basic services, very emergent. It has been used at the time of emergency and non-emergency period, and they know the prominent, the importance of the medical products. So the producer should think that the affordable manufacturer, should choose the affordable manufacturer for producing the products, so that his cost will not be made a burden on the producer, and the incidence of buying will not be borne by the consumers. It is just the, it's not big science to be remembered, it's just the human tendency which is being covered over here. So the last step is implementation of best practice. So implementation of best practices in supply chain management is you are implementing the apt or correct and effective policies and procedures in supplying medicines, because we know the medicine is very valuable for all the people. So it should be delivered at the right time. So delivering at the right time is very important for the medical sector. So it should be supplied at the right time in an efficient way. So the producer or whoever is producing the services or the commodities pertaining to medical pharmaceuticals sector should follow a best supply chain management in order to produce, supply, and uh, bridge the gap between the produce medical services and the consumer in a effective and efficient way. So this is what we see in the supply chain management pertaining to healthcare. So the when you are using this pattern of supply chain management, this will definitely aid in streamlining the activities pertaining to the medical science. So and it will also change the trends in the supply, and you will have a very good supply chain management and very good the relationship between the medical experts and the consumer. So the very basic aim of the any medical sector producers or the service producer or anything, anybody for the matter, should give a good quality of service and commodities to their consumer. So with this aim, they should work on this aim. So whoever is producing the or giving the better services to the medical services particularly should go for a supply chain management in such a way that bridges the gap between the service provider and the consumer at the right time, which is most important. And next, what we're going to see is so next is precision in healthcare. Precision is nothing but precision. We all know being consumer-centric engagement. So precision in healthcare can be defined in one word called consumer-centric engagement. For any sector matters, not only the medical sector or the life science sector, when any sector matter, whoever is producing a commodity or a service to the consumer should aim for consumer-centric engagement. So they must focus on the consumer. They must focus on the consumer behavior. So here, that is why we call consumer as king. So companies, so here we should create the personalized product for the for services and product for the consumers. So other sectors are different. So when we look into the life science sector, we personally meet the consumer. That is the patient for the matter. So the doctors personally have a connection with the patients. So he must have a patient centric engagement. So it is in humanities we call it as consumer centric engagement. In life science we can term it as the patient or the service takers engagement. So companies pertaining to life science, they are trying to go beyond the pills. That is where the normal medicines which we take every day for a headache or for a fever and things. Today's present life science experts are advising to go beyond the pill. They are going for a well-being, patients' well-being and patients' health without taking pills. So this focuses on the treatments or the medications apart from allopathy. So that is why the Indian government presently taken an initiative called Ayush, which which is focusing on Ayurveda, Yoga, Unani, Siddha, and homeopathy. So these things are beyond the usual medication called allopathy. So these things has been right now is been trending in the life science, where the researchers beyond the pills has been going for, and also the treatments and the medication patterns has also been changed drastically from 2021. So as the life science industries has been increasingly prioritizing the satisfaction of the patient, the fish prospects and enhances the value throughout the product cycle. So throughout the medication, there is a requirement for innovation. So previously, Sri Mahendra Kumar ji has been explaining about the innovations and things like that. So more focus and more thrust is being played on the innovations and inventions. So for medical device manufacturer, we have medical devices too, like hearing impaired device and the hand sticks and things like that. The personalization has been given more priority and innovation. So Abdul Kalam, late Prime Minister Shri Abdul Kalam ji, who devised the what is it, false leg, that is for the physically challenged, which was earlier was support weighting 10 kgs something like, so 15 kgs or 20 kgs like that. And Sri Abdul Kalam ji, who reduced the weight of that physically challenged people thing, the device, to 5 to 15 kgs. So that was a drastical innovation made by Sri Abdul Kalam ji. So this was helped even children to wear it without any hesitation or pain or other negative efforts. So it was effortless. It not even the people will not even know that it's been a false thing has been given into their body. So things like that. So the personalization has been given a priority. So Shri Abdul Kalam ji is a his life incident has been a very good example for the personalizing the medical device manufacturing. So he himself took many research trials in devising the false leg, from reducing its weight from 20 kgs to 10 and 15. So later he came out with 10 kgs too. Uh, and then health monitoring and in-home diagnostic equipment. So we today have the BP checking machines and the pregnancy checking machines in our homes itself. So previously we doesn't had that things. So because of that we do had some problems also. So today we have the health monitoring equipments in our home itself. So whenever we find any emergency, we can quickly use to that and we can go for first aid immediately. So these are the life trends that have been India today, even India as well as the global economy is been witnessing today, with huge and even the investment pattern that has been increasing in life science sector. To sum up, we can to sum up, we can say that the presently we are witnessing six types of trends in the life science sector, namely intelligent automation. It's all about the artificial intelligence, machine learning, and things like that. Decentralizing clinical trials. So earlier we had authority only with some certain labs for have the clinical trials. So right now we have been decentralized. 43% of clinical trials has been decentralized in India. So next we have the next gen real world evidence, where we are having the database management system. IT sectors has been collaborating with medical sectors to have the strong database management system for future research. And we have the new industrial partnerships, which is the new collaboration within and the outside medical sectors for improving and making the medical and life science sector more viable, stronger, and dynamic. And innovation in medical supply chain management, choosing the right supply chain in order to bridge the gap between the medical service provider and the patient. Precision medicine, it's all about consumer, this patient-centric medicine, personalizing the device manufacturing, medical device manufacturing, and things like that. So as we Indian economy is being growing rapidly, all the sectors has been growing rapidly, but the life science sector, as usual, we are being pioneering in the medical sector from the Vedic age. So we are far more being developing than the other world economies in the medication techniques, even our medical tourism that has been improved for the uh, various other medication techniques that has been we are using. As I already told, the personalized technique going beyond the pill, that are being vital, that is attracting the foreigners to come and take medication in our country. Yearly we earn around 183 million dollars income from the medical tourism. So we all will wish that the on this international microorganism day, so India will all definitely will become a good medical tourist spot in future. So thank you for giving me this opportunity, and thank you so much. uh, Even I could learn many things from this opportunity being humanities. So we do have a subject called health economics where we deal with these things, but because of you all, I got very good opportunity to study about evolution of biological thought in India, how it's been evolved. And surprisingly, I came to know that the biological thought was first started in India. Later only it went to other region like Europe and other things, China, Japan, etc. So thank you so much, Shri Ajay Srivastaji. Thank you so much, and yeah, many more seminars and workshops I will be participating definitely. I got very interest towards microbiology and botany, plant science, etc. Thank you so much.
Thank you, ma'am. Just a minute. Thank you, ma'am. Now, moving further, as our guest presence has filled our room with brilliance, I would like to introduce Dr. Ajay Kumar Srivastava, sir, the convener of our webinar. Dr. Ajay Kumar Srivastava, sir, is an associate professor and the head of Department of Botany at St. Xavier's College, Ranchi. Dr. Ajay Kumar Srivastava, sir's dedication to education, research, and community engagement has positioned him as a respected authority in the field of botany. So, I would like to call Ajay Kumar Srivastava, sir, to add their unique sparkle to the conversation. I request Ajay, sir. Am I audible, Ajay, sir?
Yes, you are audible.
Yes, sir. So, can you please give us a review about this webinar? Sir? Ajay, sir? I think some network issue. I just wanted, before ending the meeting, I just want everyone to request everyone to please turn on their camera so that we can capture this beautiful moment. I request everyone to please turn on their cameras so that we can move further to vote of thanks. Thank you, everyone. Okay, you all can turn off the cameras. Moving further, I would request Rashika Agarwal ma'am to conclude the event by vote of thanks.
Thank you, Preeti. Good evening, everyone present here. I, Rashika Agrawal, am here before you today with a heart full of gratitude as we conclude this enlightening webinar on International Microorganisms Day. On behalf of the Microbiologist Society India, St. Xavier's College, Ranchi University, Ranchi, Botany Department, St. Xavier's College, Ranchi, and IQAC, I would like to extend our heartfelt thanks to all of you for your presence and active participation in this event. First and foremost, I would like to express our deepest gratitude to our guests of honor, Mr. Ranjan Garg and Dr. Dwaratno Kavuri ma'am, who shared their invaluable knowledge and insights on the world of microorganism. I would like to thank our distinguished guests, Dr. Prasenjit Mukherjee, Dr. Prakash M. Halami, and Dr. Girish, who generously shared their knowledge and insights with us. Also, I would like to thank our chairpersons, Dr. Rajendra Kumar Roy, Dr. A.K. Srivastava, and Dr. Shama ma'am, whose presentations have illuminated the fascinating world of microorganisms. I thank all the resource persons, Mr. Sudhanshu Shekhar Pandey, Dr. Kumar Rajesh Ranjan, Dr. A. J. Nair, Dr. Sunita Borkar, Dr. Salesh R. Dave,
Professor J.S. Virdi, Dr. Swaranjeet Singh, Dr. Preeti Prasad, Dr. T. Ramesh, Mrs. Tanzima Yasmin, Mr. Javed Iqbal, Dr. Swati Soren, Dr. M. Mahendra Kumar, Dr. Kamlesh Jankit, and Ms. Raghui for inspiring and reminding us of the crucial role that microorganisms play in our daily lives from scientific research to environmental conservation. I would like to extend our appreciation to all the attendees. Your enthusiasm and contribution added depth and meaning to our discussions. A special word of thanks goes to our patron, Father Nabor Lakra, Principal, St. Xavier's College, Ranchi, for his generous support, without which this event would not have been possible. I would like to thank the convener of this webinar, Dr. Ajay Kumar Srivastav, sir. Your contributions have created this environment where we exchanged ideas and informations. Our gratitude extend to the organizing committee, volunteers, and our hardworking team behind the scene who's worked tirelessly to ensure the smooth execution of this webinar. Lastly but not the least, a big thanks to the Microbiologist Society India, St. Xavier's College Ranchi University Ranchi, Botany Department, St. Xavier's College Ranchi, and IQAC for organizing this wonderful webinar. So, as we leave here today, let us carry forward the knowledge and inspiration that we have gained from this webinar. Once again, thank you all for your presence, contribution, and commitment to celebrating this International Microorganisms Day with us. We are looking forward to seeing you again in future endeavors. Thank you.
So we wrap up our event here. Thank you everyone for making
Thank
this
you. Thank you all.
interaction. Thank you everyone.
Thanks to all of you. Good night.
Good night, sir.
Good night. Good night, everybody.